Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Welcome everyone to our Generic Drugs Forum 2023 Day 2, celebrating 10 years of GDF. My name is Ray Ford and I work with the Small Business Industry Assistance Program within the Division of Drug Information in CDER's Office of Communications. Good day, good afternoon, or good evening, as the case may be. Extremely warm and gratifying to see each of you join us again for Day 2. We certainly appreciate your commitment in attending for Day 2 from across the globe. Moving on this slide, we have QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. They will also be advertised across the screen banner during the breaks, just in case you miss it now. In our ongoing environment, this event is 100% remote in addition to being 100% virtual. As such, there may be some internet issues, but please bear with us, and if in fact we encounter those, we will try to resolve them as quickly as possible. Speaking of tech issues, here's a link for our one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. That's the same page that you used yesterday and this morning for your online access to the event. So please book that page right now. If you have any technical questions, please check there first and you will most likely find the answer. We invite you to submit your questions to us so that we have a vibrant and productive QA segment, which are so crucial to these events, and you can start submitting those questions right now. When you submit your question, please identify the individual by preferencing your question with the speaker's name as shown here, such as @Chitra. Also, during each presentation, there is a link on the right-hand side of your screen where you see the speaker's headshot, and it says download the slides. Note that this link is only active during the real-time presentation. There's also a link to the speaker evaluation, and we encourage you to take time to complete that, as your feedback is appreciated by the individual speakers. In addition to the speaker evaluation, we have our final evaluation, which will be available later this afternoon for our one-stop shop link. The final survey is a mechanism by which you will receive the certificate of attendance, which will support CE credits for RAPS, SOCRA, SQA, and ACRP. The certificate must be downloaded immediately after completing the survey, otherwise that opportunity is lost. Even if you do not want or need the certificate, please take a few minutes to complete the evaluation, as your feedback will help guide the design of our future events. Note that the evaluation and certificate are only available for 2 weeks. That takes us to April the 27th. Our thank you email, which will be sent at the end of each day's event, will also have the link for the final survey should you miss it today. As a reminder, All the sessions are being recorded. It'll take a week or so for us to process, but you will soon be able to access them from SBI's webpage. And I hope you have that webpage already bookmarked, and that is fda.gov/CEDAR-SBIA. I'm also pleased to announce once again that in addition to the certificate of attendance, this event is offering over the course of both days a total of 16 contact hours of continuing education for physicians, pharmacists, and nurses. These CEs are available to all eligible attendees who registered and attended real-time via Adobe. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than that of a single supporting company. CE faculty and speakers are required to disclose to attendees when products or procedures being discussed are off-labeled or unlabeled, not FDA approved, and any limitations on that information that's presented. We have a spectacular lineup of speakers. Once again, the planning committee, speakers, and CE consultation and accreditation team have nothing to disclose with the following exceptions. Mai Nguyen has an employee relationship with CVS Pharmacy and CVS stock received by employee benefits. And Daniel Oberzek has a relationship with Abbott Laboratories and participates in their defined benefit pension plan. All relevant financial relationships have been mitigated. Participants must attest to their attendance and complete the final activity via the CE portal. For multi-day activities, participants must attest to their attendance and complete the faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity, no exceptions. So in this case, the deadline will be April the 27th. I apologize in advance, but we are unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in loss of credit for this activity. NABP profile number should be the 6 or 7-digit number assigned by the CPE monitor, and your birth date should be in the format month, day, such as 0413. Please do not provide your pharmacy license number. Click the My Account tab and then navigate to the Edit Contact Information to verify that your information is correct. To repeat, attendees have 14 days from the last day of the activity to log in, complete the required evaluations, and attest to your attendance to claim credit. Physicians and nurses may then view and print a statement of credit. Pharmacists should log into the CPE monitor 10 weeks after the last session to obtain their CE credit. I'd like to call your attention to our upcoming events, all right, here on this slide, that are right around the corner, many of which are applicable to the generic drug industry audience. On April the 19th and 20th, we have FDA's Dosage Administration Section of Labeling Draft Guidance webinar. April the 25th, please log in for our Electronic Systems, Electronic Records, and Electronic Signatures webinar. On May the 2nd, join us for Navigating the First ICH Generic Drug Draft Guideline, M13A Bioequivalence for Immediate Release Solid Oral Dosage Forms webinar. On May the 15th, we have a Deep Dive GDUFA III Scientific Meetings webinar. And finally, on May the 16th, we've got our OTC Monograph User Fee Program, also known as a MUFA, Understanding the FY 2023 User Fees and Registration webinar. Also, a sneak preview is that on June the 5th through the 9th, we have our Ready annual conference. It's not advertised yet, but it will be coming soon. Just to remind you, it has 3 tracks, which will feature a plenary with the center directors and for CEDAR, CDRH, and CBER. Also, remember to subscribe to our listserv and LinkedIn from SBI's homepage, seen here for the upcoming events, and the new initiative and new various guidelines, etc. Now let's move into our presentation, starting with best practices for abbreviated new drug applications in GDUFA III. Our presenter in this presentation Our first presenter is Commander Chitra Mahavathan, is the Director of the Division of Bioequivalence Process Management and the Office of Bioequivalence in OGD. She leads a division of regulatory health project managers who are responsible for managing all the processes associated with bioequivalence reviews throughout the drug product life cycle for ANDAs. Our second presenter in this presentation is Captain Craig Keister. Captain Keister is the Division Director in the Office of Product Quality in Cedar. He manages the project management of staff responsible for co-leading the review of quality portion of ANDAs. In 2001, he started his career with FDA's OGD as a chemistry project manager. Within OGD, he's also held positions as a regulatory review officer and a microbiology project manager. Our second presentation is on the submission of in vitro released test data and information for topical drug products under ANDAs. Our presenter, Dr. Wei Zhang, is an assessor in the Office of Bioequivalence in OGD. She has experience in bioequivalence assessment for drug products across a wide range of therapeutic areas with different dosage forms. Hui serves as one of the division representatives in the topical product specialty focus group. Our third presentation is on the submission of in vitro permeation test data and information for topical drug products under ANDAs. Our presenter, Dr. Archana Manirekar, serves as a bioequivalence assessor in the Office of Bioequivalence within OGD. In her current role at OGD, she's responsible for collaborative assessment of ANDAs and controlled correspondences. She's also responsible for providing OB assessments of pre-ANDA submissions, including novel bioequivalence approaches for topical products, as well as industry outreach aimed at enhancing the quality of ANDA submissions for topical products. Next, we present on considerations for alternative bioequivalence in vitro study information submitted in nasal drugs. Our presenter, Dr. Vipra Kundur, is a pharmacologist in the Division of Bioequivalence 1 within OGD. She's responsible for assessing the bioequivalence of various dosage forms of generic drugs. She's also involved in addressing controlled correspondences and pre-ANDA meeting packages. She is a focal point expert for nasal and inhalation drug products within the division and conducts secondary reviews for these complex dosage forms. She's also actively involved in the review panel for product-specific guidances for nasal and inhalation drug products. Our fifth presentation is on Structured Submission and Review, Module 3. Our first presenter in this presentation is Dr. Scott Gordon. Dr. Gordon's been responsible for a range of activities to standardize data, including real-world data derived from health information technology for use in clinical research and pharmacovigilance, as well as standardization of pharmaceutical quality and manufacturing data for submission to FDA. Our second presenter in this presentation is Dr. Norman Schmuff. Dr. Schmuff joined FDA as a chemistry reviewer in 1990. Subsequently at FDA, he's been a team leader, branch chief, and associate director. He's participated in 4 ICH expert working groups, including what was then the M2 ECTD group. Currently, he's the technical lead for the PQ-CMC project. Our final presentation before the Q&A panel is on information to include with cover letters. Our presenter, Lieutenant Commander Nemi Matthews, is a regulatory project manager within the Division of Project Management and the Office of Regulatory Operations within OGD. She joined FDA in 2014 and has held positions within OGD as well as the Office of Regulatory Affairs. In addition to her current regulatory project management activities, Nemi also serves as a co-lead in GDUFA III implementation work group for Facility Not Ready Pathway. Please join me in welcoming our first presenter, Commander Chitra Mahavathan.
Hello, my name is Commander Chitrat Mahadevan, and I am the Director of the Division of Bioequivalence Process Management in the OGD Office of Bioequivalence. Thank you for the opportunity to speak today on best practices for abbreviated new drug applications in GDUFA III, a bioequivalence perspective. The objectives for today's presentation are to explain how to engage with the Office of Bioequivalence during various stages of the ANDA lifecycle, to provide an overview of bioequivalence communications during ANDA assessment cycles, and to review best practices. So first, I will touch upon ways to engage with the Office of Bioequivalence, or OB, in GDUFA III during various stages of the ANDA lifecycle through both meetings and written communication. So prior to ANDA submission, there are several pre-ANDA meetings that can be requested. For product development meetings, there has been no change from GDUFA II, and in general, these meetings provide an opportunity to discuss specific scientific issues related to a complex product, such as a proposed study design or an alternative approach. Pre-submission meetings have been expanded a bit in GDUFA III to provide a prospective applicant the opportunity to present unique or novel information that will be included in the ANDA submission. During these meetings, which typically occur approximately 6 to 8 months before ANDA submission, you can expect to engage with those from FDA who attended the product development meeting for the same product if one was held, as well as ANDA discipline assessment team members, including the OB team, that may assess the ANDA upon receipt. New meetings in GDUFA III include pre-submission product-specific guidance teleconferences and pre-submission product-specific guidance meetings. When a new or revised PSG is published and a prospective applicant or applicant has already started an in vivo bioequivalence study, a PSG teleconference may be requested to obtain feedback on the potential impact the new or revised PSG may have on the development program. If further feedback is required after a PSG teleconference, a controlled correspondence or a PSG meeting request may be submitted. And the purpose of the PSG meeting is to discuss the scientific rationale for an approach other than that recommended in the PSG. Prior to ANDA submission, OB will utilize written communication to respond to controlled correspondence. OB typically assesses and responds to inquiries related to inactive ingredients, specific drug development questions related to bioequivalence, risk evaluation and mitigation strategies with elements to assure safe use, or REMS with ETASU protocols, and when a prospective applicant requires further feedback after a pre-submission PSG teleconference. During the ANDA assessment, new in GDUFA III, you can request a post-submission PSG teleconference or post-submission PSG meeting. You can also request a mid-cycle review meeting, which is the same in GDUFA III, except for the revised name. In GDUFA II, it was called the Mid-Review Cycle Meeting, or MRCM, and now in GDUFA III, it is called the Mid-Cycle Review Meeting, or MCRM. And the purpose of the MCRM is for the applicant to ask for rationale for any deficiency listed in the Mid-Cycle Discipline Review Letter, or DRL, or to ask questions related to FDA's assessment of ANDA data or information. New data or information is not accepted for this type of meeting. An MCRM may be requested for complex products with a granted product development meeting, some eligible complex products with a pre-submission meeting only, and for eligible ANDAs designated as competitive generic therapy. In addition, you can also request an enhanced mid-cycle review meeting, or an EMCRM, and this is a new meeting type in GDUFA III. Complex generic products with a granted product development meeting and some with a granted pre-submission meeting only are eligible for an EMCRM. And the purpose of this type of meeting is to provide an ANDA applicant with the opportunity to ask questions related to a proposed scientific path to address possible deficiencies that have been identified in the mid-cycle DRL. An applicant may ask questions about potential new data or information, which FDA will discuss at the meeting. However, FDA will not provide substantive assessment at this meeting. The goal date will be extended by 60 days, and the DRL response due date will be recalculated from the EMCRM date. So for instance, if the initial DRL response due date was 30 days, now the response will be due 30 days after the date of the EMCRM. And as far as written communication, new in GDUFA III, during the ANDA assessment cycle, a control correspondence may be submitted if seeking further feedback after a post-submission PSG teleconference or to seek a covered product authorization. All other correspondence during an ANDA assessment cycle will be general correspondence. So there are several ways to engage with OB post-complete response letter or post-CRL. So an applicant can request a post-CRL clarification teleconference, which allows an applicant to seek clarification on CRL deficiencies. Non-clarification questions will not be answered. And new in GDUFA III is a post-CRL scientific meeting. The purpose of this meeting is to provide an applicant scientific advice on possible approaches to address CRL deficiencies that are related to establishing equivalence. Applicants can request this type of meeting even if there was no product development meeting. And for the post-CRL scientific meeting, questions related to both establishing equivalence as well as clarification questions may be submitted. You can also submit a post-submission PSG teleconference and post-submission PSG meeting. In addition, new in GDUFA III, a control correspondence can be submitted post-CRL. Post-approval in GDUFA III, you can now engage with OB by submitting a control correspondence. Some examples of post-approval questions we see in OB include inquiries about reference-listed drug availability and post-approval bioequivalence study recommendations. So now I will provide an overview of bioequivalence communications during ANDA assessment cycles in GDUFA III. The ANDA assessment program enhancements in GDUFA III are aimed at improving predictability and transparency, promoting efficiency and effectiveness of the review process, minimizing the number of assessment cycles needed for approval, increasing the overall rate of approval, and facilitating greater access to generic products. An information request, or an IR, is sent during the assessment cycle and is used to request further information or clarification that's needed to complete the discipline's assessment. The issuance of an IR does not stop the assessment clock. And deficiencies in IRs will be identified as major or minor, which is new in GDUFA III, and generally, these will include a response due date. Bioequivalence IRs are typically communicated by email and will come from the OB project manager. Discipline review letters, or DRLs, convey preliminary thoughts on possible deficiencies at the conclusion of a discipline's assessment. First assessment cycle DRLs will typically be issued by the midpoint and will include a response due date. And this is new in GDUFA III. Also new in GDUFA III, DRL deficiencies will be classified as major or minor. If minor DRLs are responded to by the requested due date, that response will be reviewed within the original goal date. However, for responses to major DRLs and responses to minor deficiencies which require comparable resources as major deficiencies, such as a consult, these will be classified as major amendments and the goal date will be extended. And this is to minimize the number of assessment cycles that are needed for approval. We will also issue IRs and DRLs after the midpoint in cases where one or more deficiency can be resolved through an IR or DRL, and as a result, the ANDA may be approved or tentatively approved in that current cycle. For these IRs and DRLs, major and minor deficiencies will be identified, and a due date may be assigned. The goal date may be extended by 90 days if the response is received by the due date, addresses all identified issues, and does not contain unsolicited information. And note that DRLs without a response due date may signify a forthcoming complete response letter. So now I will discuss bioequivalence best practices. For control correspondence, please submit through the CDER Direct NextGen Collaboration portal and not to individuals at FDA. Also, ensure that your inquiries are specific and contain sufficient detail. Please be sure to limit requests to one discipline to facilitate timely assessment and response. And please do not submit the same questions utilizing multiple submission routes. For inactive ingredient requests, please submit no more than 3 inactive ingredients and no more than 3 proposed levels for a drug product. So this means you can, for instance, submit 3 inactive ingredients with 1 level each, or 1 inactive ingredient with 3 proposed levels. Also, only submit inactive ingredients to be evaluated and their proposed levels. If they're not to be evaluated, please do not submit them. And please be sure to identify the reference listed drug, including the specific drug product strengths. For cover letters, please submit an outline of impacted disciplines, information on any previous communications you've had with the agency, such as related control correspondence or pre-ANDA meetings. For control correspondence cover letters, please indicate whether the inquiry is related to an ANDA. And for ANDA submissions, please indicate in the cover letter if there is an active pharmaceutical ingredient source change or submission of new bioequivalent studies conducted at a new contract research organization. For IRs and DRLs, please respond completely and by the response due date. Partial responses will not be accepted, and if responses are not received by the due date, the deficiencies may be included in the complete response letter and assessed during the next assessment cycle. Also, please be clear and concise and only submit the requested information, as unsolicited information may impact the assessment timeline and extend goal dates. If you require additional time to respond to an IR or DRL, please submit an extension request as soon as possible. And the point of contact for bioequivalents, IRs, and DRLs is the OB project manager listed on the letter. Extension requests will be reviewed on a case-by-case basis. So this slide lists the FDA contacts. For general ANDA questions, please reach out to the OGD RPM. For questions related to bioequivalents, IRs, and DRLs, please reach out to the OBPM. And for any quality-related questions, please reach out to the OPQ RBPM. So now I will go through 2 challenge questions. So for question number 1, in GDUFA III, during the ANDA assessment cycle, a control correspondence may be submitted in which of the following situations? A, after a PSG teleconference. B, when seeking a covered product authorization. C, for any clarification questions. D, A and B only. Or E, all of the above. Please take a few moments to think about the correct answer. And the correct answer is D, A and B only. So a control correspondence may be submitted during the ANDA assessment cycle if one is seeking further clarification after a PSG teleconference or when seeking a covered product authorization. Other correspondence will be considered a general correspondence. So for challenge question number 2, which of the following is true about first assessment cycle DRLs in GDUFA III? A will be issued by the assessment cycle midpoint. B, deficiencies will be classified as major or minor. C, a response due date will be included. D, responses to major DRLs will extend the goal date. Or E, all of the above. Please take a few moments to think about the correct answer. And the correct answer is E, all of the above. So first assessment cycle DRLs will be issued by the assessment cycle midpoint. They will be classified as major or minor and include a response due date. In addition, responses to major DRLs will be classified as major amendments and the goal date will be extended. So the next 2 slides include various helpful resources for you to refer to. So in conclusion, please be familiar with the expanded definitions of controlled correspondence, new meeting types, and communication enhancements in GDUFA III. In addition, follow the best practices discussed to reduce the number of bioequivalence assessment cycles. For any questions related to bioequivalence IRs and DRLs, please reach out to the OB project manager. And thank you for your time and attention today. I look forward to answering any questions during the panel session.
Good morning. My name is Captain Craig Keister. I'm a Division Director in OPQ's Office of Program and Regulatory Operations. Today I'm going to explain how best to engage with OPQ during the ANDA lifecycle, how to provide an overview of OPQ communications during and after assessment cycles, and finally, talk about some best practices and common themes we see on our end. Hopefully you'll find this information helpful. First and foremost, the RBPM is the primary point of contact for all OPQ-related inquiries. The RBPM and OPRO, and they work closely with the RPMs in OGD, as well as the assessment team in OPQ on all of your applications. Additionally, they issue all communications related to quality during the assessment cycle. If for some reason your RBPM is unresponsive, please be mindful of the layers in the organization to elevate to, starting with their branch chiefs, division directors, deputy office director, or office director, prior to reaching out to more senior FDA leadership. Next up, I'd like to talk a little bit about the information request letter, or the IR letter. It's very common and very valuable means of communication between us and industry. It's a tool that allows us to solicit for additional information or clarification that we need in order to complete the assessment cycle. The IR could be single discipline or multidiscipline. When issuing the IRs, the RBPMs will typically request a response date, and you will receive a communication via either phone, email, or letters. Next up is the discipline review letter, the DRL. And that is more comprehensive than the IR. It's the assessment team's preliminary findings, and it's issued when our assessment has been substantially completed. This is usually around the midpoint of the assessment cycle. Like the IR, you will typically be asked to respond by a certain date. Responding to IRs and DRLs. Extension requests should be made as soon as possible by contacting the RBPM. The RBPM and assessment team will work together to determine if an extension will be granted. Your extension request should be done in a manner that provides you enough time to completely respond to the IR DRL, and we will frequently not allow multiple extensions. I would also like to point out partial responses will not be accepted, and the timing or unsolicited information in either of these submissions could potentially extend the goal date of the application. Next up, we'll talk about OPQ communications, and particularly the meetings. First up would be the mid-cycle review meeting. It's for the applicant to ask for rationale for any deficiency identified in the mid-cycle DRL, and or to ask questions related to the FDA's assessment of data or information in the ANDA. An applicant may not present any new data or information at this meeting. A mid-cycle review meeting will take place within 30 days after the date the sponsor submits a meeting request. A new meeting we have is the enhanced mid-cycle review meeting. The purpose of this meeting is for the applicant to ask questions related to proposed scientific path to address possible deficiencies identified in the mid-cycle DRL. An applicant may ask questions about potential new data or information to address any possible deficiencies identified in the mid-cycle DRL. FDA will discuss the data and information, but will not provide substantive assessment of data or information provided by the applicant at that meeting. If an enhanced mid-cycle review meeting is requested, the meeting will take place within 90 days after issuance of the last mid-cycle DRL. FDA will extend the ANDA goal date by 60 days if an enhanced mid-cycle review meeting is requested. It will also extend the response due date for any relevant DRLs by recalculating the response date starting from the date of the meeting. For example, if we initially requested 30 days after the DRL, it will now be due 30 days after the enhanced mid-cycle review meeting. An applicant may submit an unsolicited amendment after enhanced mid-cycle review meeting, which could result in additional goal date extensions as well. Post-CRL teleconference. That's used to seek clarification concerning deficiencies identified in a CRL. FDA will grant appropriate requests for teleconferences requested by applicants upon receiving first cycle CRLs and upon receiving subsequent CRLs. An appropriate request is one that clearly identifies the specific deficiencies to be discussed and the reason why such deficiencies are not clear. FDA will provide a scheduled date for 90% of the post-CRL teleconferences within 14 days of the request and conduct 90% of the teleconferences, if granted, within 30 days of the written request. Controlled correspondences. So under GDUFA II, OPQ received approximately 700 controlled correspondences annually. So approximately 2,800 of those in the previous 4 years. Common themes of those were stability data and how much was needed, container closure, changes, bracketing of batches, microbiology, batch size, packaging changes, and questions. The control correspondence on the amount of stability data increased dramatically after the publication of FDA guidance, development of ANDAs during the COVID-19 pandemic, questions and answers. Under the G2 framework, correspondence seeking regulatory or scientific advice after issuance of the CR, TA, or approval letter was considered general correspondence. Under the new G3 regulations, these are now submitted as controlled correspondence. Applicants now have the option to submit controlled correspondence to seek clarification. In OPQ, we perform an IQA with a multidisciplinary team and the PMs on each application. This team, in most cases, will be assigned to address the controlled correspondence. So, to sum up, during the ANDA assessment cycle, it can be submitted if an applicant needs feedback following a product-specific guidance teleconference or to seek a covered product authorization. If it's after the ANDA assessment cycle, so following a CR, TR, or full approval, it'll be submitted as controlled correspondence. DMF unsolicited amendment issues. Poorly timed unsolicited amendments continue to adversely impact the application timelines. This can result in goal date extensions, unnecessary CR letters to the DMF and or the applicant. Frequently, unsolicited amendments are not necessary to be submitted at the time they are. And communication between the applicant and DMF holder regarding DMF submissions is the best solution. So some tips to avoid unsolicited amendments. DMF holders and applicants should share the information in a timely way. Any plan changes to the DMF, as well as action dates and priorities for the referencing applications. If it is not clear whether an unsolicited DMF will impact an application timeline, do the following. Reach out to your customers to get information on their application timelines. Reach out to DMF OGD and see if we can provide helpful guidance on the submission timing. And most importantly, understand the consequence of a poorly timed DMF submission to your customer's ANDA. It can likely result in goal date extensions depending on the date that it's received, and the ANDA goal date will be extended from the date of the DMF amendment. So if that amendment is submitted very late in an ANDA review cycle, they potentially could have a long extension to the ANDA. The length of that extension is determined by the nature of the amendment. If it's a major or a minor, it could result in them losing an exclusivity or a forfeiture date, and withdrawal of an amendment will not undo an already extended goal date. So some GDUFA III DMF enhancements. So first up is a DMF prior assessment. A DMF holder may submit a request for the assessment of an original DMF previously unreviewed 6 months prior to the planned submission date of an ANDA. And that's for an original, an amendment, or a PAS. FDA will grant and review the DMF prior to ANDA submission when the planned application submission meets certain criteria. Some of that criteria: all patents and exclusivities will expire within 12 months of the planned submission date. There are fewer than 4 approved therapeutically equivalent drug products, including RLD in the Orange Book. No blocking patents or unexpired exclusivities in the Orange Book. And the applicant is not seeking to carve out any conditions of use. In addition, submission for a drug product that could help mitigate or resolve a drug shortage and prevent future shortages, or if it's related to a public health emergency. So FDA assessment of solicited DMF amendments. FDA will assess certain solicited DMF amendments related to original ANDAs and PASs upon receipt, even if the original ANDA or PAS in which the DMF is referenced is not currently under assessment. Priority is given to those amendments related to ANDAs for which acceptability of the DMF assessment may result in a full approval on the next application cycle. Assessment comments issued to DMF holder at least in parallel with issuance of the review comments relating to the DMF that ANDA references.
Now let's discuss some common deficiencies in the DMFs. We'll start off with non-mutagenic organic impurity controls. All potential impurities need to be included in the discussion. This includes starting materials, intermediates, including in situ intermediates as appropriate, byproducts, and impurities in the starting material and intermediates, as well as their continued reactions in the process.
Common deficiencies in the DMF. Mutagenic impurity controls. Failure to use QSAR methodologies that are ICH M7 compliant in the hazard assessment. Insufficient data is provided when upstream controls are established at higher limits. Incorrect threshold of toxicological concerns is proposed. Failure to limit the total mutagenic impurities when multiple impurities are present, and failure to show the analytical method is suitable for its intended use. Drug product considerations. Extractable leachable studies for key packaging components. Utilize correct safety concern threshold for analytical evaluation thresholds. Select study conditions pertinent to specific attributes of the product. Maximum daily dose for impurity and extractable leachable qualifications. Impurity qualifications besides the pharm-tox qualification for small molecule APIs, and any device differences between generic and RLDs. Facility assessment, what we need. Please ensure that all current facilities, including all relevant DMF facilities and their responsibilities, are clearly listed on the Form 356H. Process development and bridge to commercial scale. Commercial process descriptions, process flow diagrams, and sterilization validation packages for sterile products. We would also like to note that you are now required to verify that all facilities are ready for inspection. Common manufacturing deficiencies. New batches required due to significant failures with submitted batches with no root cause of understanding provided. An incomplete facility responsibility listing on a 356H, and process parameters and in-process controls not supported by process development knowledge in the application. The cover letter. We have attached is a hyperlink for the cover letter attachments for control correspondence and ANDA submissions. We ask that you use this when identifying when preparing submission cover letters. Be sure to identify all applicable disciplines for which you're submitting information, and that's for the amendments in particularly. We'd also like to remind you that secure email is required or strongly recommended If you don't have this, you can easily request it by sending an email to secure@fda.hhs.gov. The templates and the guidance appendices will make for more efficient triage process. I'm not going to read all these off, but the next 3 slides provide links to resources that will be helpful for you for managing your applications. CEDAR guidance, GDUFA webpage, good ANDA practices, cover letter, attachments for control correspondences, ANDA submissions, and on. So in summary, please reach out to all RBPMs for all quality-related communications. Completely respond to all quality IR DRLs by the requested response date, and be familiar with your ANDA assessment timeline and all possible communications. Actively and frequently communicate with your DMF holder. And lastly, take advantage of the many new ways to communicate with the agency with the GDUFA III enhancements. Thank you very much for your time. Be familiar with your ANDA assessment timeline and all possible.
I'm from the Office of Bioequivalence in the Office of Generic Drugs. In this presentation, I will speak on the submission of in vitro release test data for topical drug products under Abbreviated New Drug Application. This presentation reflects the views of the author and should not be construed to represent FDA's views or policies. After this presentation, the audience should be able to identify the key components that constitute a high-quality IVRT submission, describe the level of detail expected for each key component, and know the appropriate module for each component to be submitted in the electronic common technical document. I will start off with a brief introduction on the in vitro release test, and then explain the agency's expectation for a high-quality IVRT submission. In the next few minutes, I will share with you some submission-related common deficiencies. FDA published the draft guidance for industry in vitro release test studies for topical drug products submitted in ANDAs in October 2022. In recent years, there has been an increasing trend of topical drug product applications that contain in vitro studies, in place of the traditional comparative clinical endpoint study to support demonstration of bioequivalence. The characterization-based approach includes in vitro release test, in vitro permeation test, and physicochemical and structural property studies. IVRT is utilized to assess the rate of drug release from a proposed generic topical product, comparing to its reference standard for the purpose of supporting a demonstration of bioequivalence. Please note, the topical products here may include gel, emulsion, lotion, cream, ointment, or other semi-solid or liquid-based dosage forms applied to the skin, but do not include the transdermal delivery system, such as patches or fumes. To develop an IVRT method and eventually use it to support a demonstration of bioequivalence, the process includes 3 phases: method development, method validation, and pivotal comparative IVRT study. In general, there are 4 major components. Usually, the applicants prepare a separate study report for each of the major components. Those study reports include stepwise details of the test procedures that enable the assessors to reconstruct in their mind how the studies were conducted. Also, in the submission, all the raw data are included so the agency may verify the study results. In the next few slides, we will discuss the agency's expectation with regard to a high-quality IVRT submission. Like other in vivo bioequivalence studies, The summary tables for each component are to be submitted in Module 2.71. Detailed study reports, protocols, relevant standard operating procedures, and other supporting documents are to be submitted in Module 5.312, except for anything pertaining to the sample analysis, the analytical method validation, and the documentations related to the study sample analysis, including the chromatograms, raw data printout, should be submitted in Module 5.314 in the respective subfolders. In this slide, we will focus on the IVRT method development. Summary information relevant to IVRT method development, including but not limited to the following information, may be submitted in tabular format in Module 2.71. Given that multiple conditions may be tested to optimize your IVRT method, it is recommended to list all the conditions tested for each parameter. A detailed IVRT method development report containing the above information as well as supporting documents should be submitted in Module 5.312. One of the common deficiencies is that applicants described all the parameters they had tested, but did not include any rationale for the selection of parameters. Another deficiency is that missing study details in the report. For example, the stepwise procedure for dose application. Please include information such as the tool you use in the dose application, whether the dosing chamber is occluded or not, is dosing staggered or sampling synchronized, or vice versa. Now let's move on to the method validation. Similar to the method development, in Module 2.71, summary table of IVRT method validation should include the general information and a method description. A report of IVRT method validation should be submitted in Module 5.312. Please refer to the IVRT guidance for the definitions of validation parameters. It is commonly seen an IVRT submission miss one of the following information, such as apparatus qualification data, receptor solution sampling qualification data, a detailed procedure for dose application, or a component and conversation table of the altered formulation, or room temperature and relative humidity logs. Just like a regular submission for in vivo bioequivalence study, you need to submit a summary table for analytical method validation in Module 2.71 and a standalone report for analytical method validation along with SOP, raw data, and the representative chromatograms in Module 5.314. I want to emphasize here, please do not forget the stability data under assay condition. Now we move on to the last key component, pivotal IVRT study. The contents in the summary tables are very similar to that in the method validation. You provide the final test parameters of the pivotal study design, as well as the general information such as study number and the test site. Please note, as a pivotal BE study, the test site is subject to FDA's inspection. In the report of pivotal IVRT study, please make sure you include the test and the reference product information, such as the batch size, the manufacturing date of the test product, and expiration date for the reference product, and the potency for both batches. The requirement for submission of sample analysis data is the same as other in vivo PKBE studies. That should include 100% of the raw data and 20% of the chromatograms. Please submit all the rejected, re-injected, and repeat analysis. The individual concentration data should be submitted in the appropriate format. To summarize, the agency often identifies missing or incomplete information in the original submission. Many of the common deficiencies described today can be avoided. Please submit all necessary information or data that enable the reconstruction of study process and the verification of results. Now it's time for the challenge question. What are the key components of an IVRT study submission? The answer is all of the above. That concludes my presentation. I would like to express my gratitude to my colleagues and supervisors for their invaluable assistance in preparing for this presentation. Now I hand it over to the next speaker.
Good morning. It's great to be here today to talk about IVPT submissions. As we know, IVPT studies may be used to characterize and compare the rate and extent of a drug's bioavailability at or near a site of action in the skin from a test topical product and the reference standard. To talk about IVPT submissions. Thus, IVPT studies are important components of the characterization-based bioequivalence or BE approach for generic drugs as outlined within product-specific guidances of several topical products. So let's jump right in and see if we can demystify high-quality submissions for IVPT studies. This presentation reflects my views and should not be construed to represent FDA's views or policies. Our learning objectives are to identify key components of a high-quality IVPT submission, understand the key submission expectations for each key component, and clarify the organization of IVPT information and data within the ANDA submission. The 5 key components include relevant to the IVPT methodology, development and optimization, referred to as IVPT method development, validation, including sensitivity and pilot studies, referred to as IVPT method validation, and IVPT pivotal evaluation based on the validated methodology. Relevant to the analytical methodology, validation of the receptor sample analysis methodology, also referred to as analytical method validation, and IVPT method validation and pivotal study sample analyses. Now, let's look at some key submission expectations for all IVPT studies, as well as a few that are specific to each of the 5 key components. Key submission expectations relevant to all IVPT studies include submissions of details relevant to each IVPT study, such as study objective, method parameters, study procedures, operators involved, and conclusion. For examples of IVPT method parameters and study procedures, please refer to Appendix B provided in the PDF of these slides. Other key expectations for all IVPT studies include the submissions of details that are relevant to each experimental IVPT run, such as diffusion cells run in parallel, identified by unique cell IDs, cells that were dosed, relevant product lot numbers, and experimental run dates. Details relevant to each product lot, such as lot number, product strength, formulation, batch size as applicable, as well as assay potency with container content uniformity if available, manufacturing or expiration dates as applicable, and for any altered formulations, rationale for the alteration made, and description of the altered manufacturing process. Details relevant to each skin section, such as donor ID, age, race and sex, supplier information, anatomical site of skin collection, and individual measurements mean and percent CV for skin thickness and barrier integrity test results. Details relevant to the handling of each skin section, such as conditions at which skin was received and subsequently stored, date and time of each retrieval from and restorage in the deep freezer, and the number of freeze-thaw cycles to which the skin was subjected. Lastly, submission of details of any protocol or SOP deviations or experimental observations that may influence the interpretation of the study results. Moving on, key expectations for IVPT method development studies include submissions of recommended details as relevant to all IVPT studies, documentation of all systematic efforts leading to the selection of a IVPT methodology suitable for demonstration of BE, complete details of the optimized IVPT methodology comprised of IVPT equipment and method parameters, acceptance criteria and study conditions, along with well-controlled study procedures documented in detailed SOPs. Another key expectation is the submission of spreadsheets containing raw concentration and PK data for method development studies showing all relevant calculations. For the preferred data format, please see Appendix A in the PDF of these slides. Lastly, it is expected that IVPT methodology that is first thoroughly optimized, then adequately validated and utilized in the pilot study is used unchanged in the subsequent pivotal study. Key expectations for IVPT method validation studies include submission of adequate information and data to support specific qualifications and controls that are related to the suitability of IVPT equipment, suitability of skin sections selected for dosing, suitability of the receptor solution, suitability of the sampling procedure, characterization of the permeation profile and range, characterization of the precision and reproducibility of results, characterization of depletion of the applied target dose over the study duration, validation of the method selectivity, consistency of ambient study conditions, suitability of the applied target dose amount and dose duration, and lastly, submission of study results that are supportive of method robustness if available. Other key expectations include submissions of recommended details that are relevant across all IVPT studies, detailed protocols and SOPs, adequate evidence to support control and consistency of study procedures, conditions, and method parameters, as well as submission of spreadsheets containing raw concentration and PK data for all validation studies showing relevant calculations. Moving on, key expectations for IVPT pivotal study include submissions of, again, recommended details that are relevant to all IVPT studies, relevant protocols and SOPs, details of randomization, blinding, and unblinding procedures, adequate evidence that supports control and consistency of study procedures, conditions, and method parameters, as well as SAS transport raw concentration PK datasets for the pivotal study. Next up, key expectations for analytical method validation studies include submissions of complete details of the sample analysis method and experimental procedures, relevant protocols and SOPs, submission of complete details of each study involved in the analytical method validation such as linearity, recovery, etc., along with adequate assessments of drug stability and dilution integrity as applicable. Last but not least, key expectations for analyses of IVPT method validation and pivotal study samples include submissions of relevant protocols and SOPs, 100% numerical raw data for all analytical runs, complete details of each analytical run or sample, rejection, reinjection, repetition, or reintegration as applicable, and submission of adequate representative chromatograms. Lastly, relevant to each analytical run, submission of details such as run ID, analyte name, dates of extraction and assay, donor ID, instrument ID, analytical run status such as accepted, rejected, re-injected, etc., and a brief run description. Now let's see how these IVPT data and information should be appropriately organized within the ANDA submission. Information and data relevant to IVPT studies, such as method development, validation, and pivotal studies, belong in Module 5312. Thus, study reports, protocols, datasets, etc. relevant to these studies should be placed in appropriate separate subfolders in this module. Now, simplified trial summaries or ts.xpt files that are relevant to the method development, validation, and pivotal studies should also be submitted in Module 5312. For further details, please refer to Appendix C in the PDF of these slides. Information and data pertaining to analytical method validation or IVPT sample analyses belong in Module 5314. Thus, all relevant reports, protocols, SOPs, etc. relevant to analytical method validation or IVPT sample analyses should be placed in appropriate separate subfolders in Module 5314. And that brings us to our challenge question. Validated IVPT methodology as used in the pilot study should be utilized unchanged in the pivotal study. a change in which of the following would not be considered as a change in the IVPT methodology between the pilot and pivotal studies? A. Study procedure. B. Acceptance criteria. C. Formulations evaluated. Or D. Equipment. Take a few seconds to think about which answer you think is right. If you selected option C, you are correct. At slide 14, we identified options A, B, and D, but not C here as components of an IVPT methodology. Accordingly, use of a third additional formulation in the pilot study as compared to the test and reference standard formulations in the pivotal study is not considered as a methodology change. In summary, providing high-quality IVPT submissions is straightforward once we have a good grasp of their A, B, and C's. That is, a good understanding of the key components, the key submission expectations, and their organization within the submission. For comprehensive information, always consult the relevant agency guidances. Now, obviously, in order to generate high-quality IVPT submissions, the underlying IVPT information and data must also be high quality. Therefore, as you work on your future IVPT projects focused on high-quality developments and optimizations, validations, and pivotal evaluations, please utilize the A, B, and C's as discussed today to provide high-quality IVPT submissions to the agency. That concludes my presentation. I will be back shortly to answer your questions, and thank you for your attention.
Good morning, everyone. My name is Vipra Kundur, and I am a pharmacologist in the Division of Bioequivalence, Office of Bioequivalence within the OGD. Today I will be speaking on considerations for morphologically directed Raman spectroscopy, study information submitted in nasal suspension ANDAs. I would like to start my presentation with a disclaimer. The views and opinions expressed in this presentation are solely from myself and do not necessarily reflect the views of the FDA. The learning objectives are to explain the alternative approaches to the comparative clinical endpoint study for locally acting nasal suspensions, considerations for a high-quality submission for an MDRS study, and to identify submission-related common deficiencies for an MDRS study. The FDA has a weight of evidence approach for demonstrating bioequivalence for the nasal spray suspension drug products. The weight of evidence approach utilizes in vitro studies to demonstrate equivalent in vitro performance, pharmacokinetic or PK studies to demonstrate systemic exposure, and comparative clinical endpoint study to further support the equivalent local delivery for a test product that has demonstrated formulation and device similarity. However, there are certain challenges in conducting a comparative clinical endpoint study, such as high variability, low sensitivity, and longer study duration. Therefore, FDA also recommends an alternative BE approach in lieu of a comparative clinical endpoint study. Historically, the FDA has recommended an in vivo comparative clinical endpoint study to ensure equivalent local delivery of drug between the test and reference products. However, with the emergence of advanced techniques such as morphology-directed Raman spectroscopy, the FDA currently recommends that the prospective applicants may submit drug particle size distribution data in lieu of an in vivo comparative clinical endpoint study to support bioequivalence of nasal suspension ANDAs. With the emergence of MDRS technique, there has been an increasing trend of nasal suspension product submissions that contains MDRS study. Many MDRS study deficiencies are related to incomplete submissions. So in this presentation, I will provide an overview of common deficiencies that are observed for an MDRS study, and I will also provide clarity on the agency's expectations on MDRS data in support of bioequivalence. In this slide, I am going to talk about what information should be submitted for an MDRS study and also in which module should the information be submitted. All the BE summary tables should be submitted in module 2.7.1. Currently, we don't have BE summary tables posted for an MDRS study. However, we recommend that you please check the fda.gov website for the most current recommendations. In module 5.3.1, please submit the method development and method validation reports, including standard operating procedures and protocols with predefined acceptance criteria that were effective at the time of the study. In the same module, please also submit the pivotal study report. For the pivotal study, please submit the 100% raw data and also the PBE analysis on D50 and SPAN. Depending on the data submitted and upon evaluation of the said data, additional information may be requested during the ANDA assessment. In the following slides, I will be discussing the most common submission-related deficiencies for an MDRS study. These submission deficiencies are related to method development, method validation, and pivotal study. The most common submission-related deficiencies for method development are observed for sample preparation and image and size characterization. Submission-related deficiencies for sample preparation are mostly related to incomplete submission of detailed procedure and validation data. Therefore, we recommend providing complete optimization data on the following. Selection of an appropriate dispersion technique. For example, wet dispersion or wet measurement technique should be used for aqueous-based suspensions. We recommend providing particle size distribution data for pre and post actuation to demonstrate control on agglomeration. And also each batch should be evaluated separately. Data should be provided to ensure that the selected sample volume is appropriate and also has no touching particles and the dispersion is uniform. Particle size distribution data for each concentration should be provided to ensure reproducibility. We recommend providing optimization of particle settling time and also the method used for de-agglomeration. If any surfactant is used, then the solubility of particles in the selected surfactant should be provided, and also the concentration of surfactant should be optimized for particle size distribution reproducibility. Since excessive pressure results in particle damage, the cover slip and its placement is very important. Therefore, particle integrity validation data should be provided. Validation data should also be provided for the selection of an appropriate sealant for preventing sample evaporation. And finally, Data to justify the selection of number of sprays for sample preparation should also be provided. Image and size characterization deficiencies are also related to incomplete submission of validation data. We recommend providing supporting data for the selected crash pixels and equivalent size for area pixel using appropriate magnification. Provide validation data to support selection of the threshold value. Optimization of Raman particle count data should also be provided. Validation data for distinguishing API and excipient particles and also agglomerates should be provided. We recommend that you specify whether the same elongation filter was used and optimized for both test and reference products. Provide data on selection of circular equivalent diameter that is appropriate for the product. Provide validation data to support the selection and optimization of Raman's correlation score. An appropriate orthogonal method should be selected to support the pivotal MDRS method. And finally, provide validation data to justify the pre-processing settings. Submission-related common deficiencies for MDRS method validation and pivotal study. Many of the deficiencies for method validation and pivotal study are related to missing study information and documents. We recommend that you submit validation data for MDRS method, for reproducibility of the method and accuracy, and also robustness of the method. For the pivotal study, please submit the 100% raw data and PBE analysis on D50 and SPAN. To summarize my presentation, the number of ANDA submissions containing an MDRS study has increased in recent years. I went over multiple common MDRS submission deficiencies that are related to method development, validation, and pivotal studies. And as you can see, many of the identified MDRS-related deficiencies could be prevented by providing supporting development and validation data. For folks who are interested in the CE credits, I have one challenge question for this topic. Which of the following statements is true? A. Potential challenges of a clinical endpoint study include high variability and longer study duration. Option B. Number of ANDA submissions containing an MDRS study has increased in the recent years. Many MDRS study deficiencies are related to incomplete submissions. Option D. All of the above. And the answer is option D, all of the above. So with this, I would like to conclude my presentation, and I hope with my talk, I gave you some helpful hints on how to improve your MDRS study submissions by avoiding unnecessary deficiencies and also reducing the number of review cycles. Thank you.
Hello, I am Scott Gordon. I'm a Senior Health Informatics Officer as part of the Data Standards staff in the Office of Strategic Programs at the Center for Drug Evaluation and Research at the FDA. I'll be continuing to talk about the topic of this session, specifically focusing on our efforts to standardize the information of pharmaceutical quality, chemical manufacturing and controls, or PQCMC. Specifically, at a high level, what is it, where are things now, and how you can learn more. Standard disclaimer, the views and opinions in this talk are mine and shouldn't be attributed to my employer, FDA, and the presentation itself should not be considered guidance or a change in guidance, nor does it necessarily reflect FDA's positions. After this talk, you should be able to explain the problems the PQCMC program intends to solve and how, identify the goals, objectives, and scope of PQCMC, describe the processes used to develop the PQCMC data standard, understand stakeholder outreach taken by PQCMC, discuss some current and future work, and recognize where you can find out more. So why is PQCMC needed? What is the problem that the PQCMC program is attempting to address? On the left-hand side is a representation of the Electronic Common Technical Document, the ECTD, which is an ICH standard that is used for packaging all applications to regulatory agencies such as the FDA. For the purposes of this talk, focuses on Module 3. Module 3 is the home of all the detailed information relating to the product quality, chemical manufacturing, and controls for the drug product in question in a given application. Some information, relevant information, also exists in Module 2 in summary form. The issue at hand is that all of Module 3 arrives to FDA as essentially electronic paper, so PDFs and things of that nature. These are inherently unstructured and non-computable data information. They're often not standardized. You can represent similar things in slightly different ways. For the sponsors, this creates a large amount of manual work just to create. And on the FDA side, there's equally large amount of manual work to transcribe and otherwise prepare this information so that they can reliably analyze this information. The net result of this is it's incredibly time-consuming, not an ideal way to be preparing data or using data that needs the kind of analysis it deserves in the 21st century. So what is the solution that the PQCMC program is offering to help address this issue? The PQCMC program is developing a data standard that supports the transmission and receipt and processing of PQCMC information that is consistent in format, uses consistent values, and most importantly is highly computable and ready for analysis. There's benefits across the board, and these are not exhaustive. From the sponsor side, having this as a standard in the future will allow some clear expectations of exactly how data should be formatted, will actually support a pre-checking of the content and the quality of the content before submission, and even support other industry side data analytics. From the FDA side, we see this as being beneficial in providing a consistent in format and values that are received. It will be able to fuel software-powered analysis and will be much faster to review. And on the FDA side, you can also check for valid content and data quality upon receipt. As a mutual benefit, this is an example of the paradigm of submitting the single kind of information, the single piece of information one time to FDA, and FDA can then use it for many purposes, as opposed to sponsors having to submit basically the same information to different places at different times. What are the core activities of the PQCMC program? We'll start with the two overarching goals. First is to determine just how to structure Module 3 information, and then to work with a standards development organization, in this case HL7, to make this an implementable data exchange standard. And the PQCMC data standard is being developed using HL7's FHIR data standard, and I'll be talking a little more about that in a moment. And as for the scope of the PQCMC program, the amount of information that this developing standard is meant to cover, it's quite a lot. It's pretty much everything that's amenable to structuring in Module 3, and as mentioned before, also some sections of 2.3. And it covers quite a lot of different types of applications, those that are pharmaceutical-oriented, those that are biologics-oriented, as well as veterinary medications. And the 3 centers listed here have been working since the beginning of the program on this effort as well. And it's also meant to cover pretty much all application types and pretty much all dosage forms. And now for a somewhat deeper dive into the information areas to be covered by the developing PQCMC data standard. The program divided this work into 2 somewhat sequential phases, the first of which covering many foundational concepts, and the second going deeper into complex manufacturing processes. So for phase 1, these were the foundational concepts: how to define every drug product and every substance within that product, the quality control tests and acceptance criteria for product substances, excipients, raw materials, formulas for making batches of the product, results of quality testing, stability, and analysis of batches of products and active pharmaceutical ingredients, as well as details on packaging and containers. And on the left, you'll see the more formal descriptions of these data domains. Phase 2 of this work then took all those foundational concepts and then dove into the description of the complex manufacturing processes for drug substances and drug products. So put very simply, how a manufacturer puts everything together to create products. Every step, every mechanism, machine, process, etc. And what steps take place at which facility, of which there are many for one product. And this phase is being done in a more iterative fashion. The drug substances, we are focusing first on small molecules, more of the pharmaceutical side, and then biologics. And on the product side, you can see we're aiming to cover a wide range of dose forms, the most common of which are things like solid orals, but there's many others, obviously liquids, and then getting into biologics and other variations. So the intent is to cover quite a bit. And also some additional coverage for things like annual lot distribution reporting and CMC changes in our annual reports. So a question I'm sometimes asked in regards to the PQCMC program is, why does it take so long to create a data standard? Well, creating a data standard from scratch is inherently an immensely time-consuming process. And this is meant to be sort of an overview of why. When there is no pre-existing structuring, an enormous amount of time is spent on determining what can be structured and how to structure it. Issues such as the level of detail, number of data elements, terminologies that may need to be used, relative cardinalities of different elements, any business rules that FDA or another organization may have, all have to come into play for hundreds and hundreds of bits of information. All of this, of course, needs to be reviewed and revised. Internally, we review these at FDA with our various center subject matter experts. We've also, and I'll get into this more in a moment, released batches of these drafted elements and terminologies to public by Federal Register notice to get industry comment. And that also leads to revisions where deemed necessary. Then all of this is taken to the Standards Development Organization, HL7 in this case, to begin the sort of designing process. How do we represent all of this information, this structuring in HL7 FHIR? And this is where we follow HL7 processes, to the letter to ultimately create a final representation in HL7 FHIR of the PQCMC data standard. The culmination of all this work at HL7 using FHIR is in the production of what is called an implementation guide. An implementation guide, put very simply, is the set of instructions for IT folks to create and implement the software, databases, etc., to allow the creation of messages and or ingestion and processing of messages that follow the rules set down in the standard, all of this work that was developed. It also supports additional narratives and other information so that non-IT folks also will have as comprehensive an understanding as possible as to how this standard will work. As a brief aside, I'm sometimes asked, why use FHIR for this work? Well, a number of reasons. First, of course, it's a consensus-based standard for public use. This is pretty much a must-have for U.S. federal government agencies. But more importantly, FHIR has an immense amount of flexibility built into it and allows us to represent the data needs that we have quite exactly. There's a lot of work to make it do that, but once it's done, it works. Further, there's much built-in support in HL7 FHIR for PQCMC definitions and concepts. Now, this is because of work that our teams at HL7, as well as the European Medical Agency and Health Canada did to put a lot of these requirements into what is about to be published, version R5 of FHIR. So that's an extra benefit. Further, our PQCMC work harmonizes well with other FHIR development in the same general data domain space. For example, the EMA work on IDMP. Now, I mentioned previously about the stage of review with industry, and this is just a blow up sort of that previous timeline. The outreach to industry and the general public is critical to getting this right. Getting the input of all stakeholders is the only way to ensure that the ultimate representation of this information in the final standard will work for everybody. So the process that we at FDA take to getting this information out into the public is the release of what are called Federal Register notices, specifically asking for comment on documents. So we prepare documents that show a set of the data elements and relevant terminology lists and ask for comment. To be clear, this is not guidance. This is purely documents for comment. And once we receive those comments, our teams look them over, and often we get very useful advice, and that helps make refinements to the data elements and terminologies going forward. And here I'd just like to present a short timeline of the industry and public outreach that has been and will continue to be ongoing. So in 2017, we released a first draft of a good portion of the phase 1 data elements and control terminologies for comment. This was followed in 2018 by a public meeting, which gave us a lot of input about the whole PQCMC initiative, as well as additional feedback on phase 1. We had in 2019 a very early proof of concept IT testing with some industry participants that gave some valuable feedback at a very technical level. We spent the subsequent couple of years doing quite intense FHIR development at HL7. So under the Biomedical Research and Regulatory Group, which is the sponsor of this work at HL7. and developing the FIRE implementation guide. In March of 2022, we released a new notice showing an expansion of the Phase 1 data elements with lots of revisions based on feedback by everything previous on this timeline, as well as a representation in a more document form of proposed representation in HL7 FIRE. And this is something we expect to be continuing now and into the future. So where are we now? Well, Phase 1 structuring is complete, and Phase 2 structuring is well underway at this point. An early draft of the HL7 FIRE implementation guide with Phase 1 information is in development. This is a clickable link. Please go ahead and take a look. It is a draft implementation guide. It is information only as part of the HL7 PQCMC project. To be clear, it is not final. We expect revisions to it, nor is it binding in any way. It is purely for informational purposes. And revisions are expected as Phase 2 structuring keeps moving forward and after public comment gets represented as FIRE in this implementation guide. Where are we heading? Well, we're certainly heading to the completion of Phase 2 structuring. We'll be making decisions on staging, in other words, what parts of PQCMC to implement when. We definitely anticipate further rounds of industry participation in pilot testing. And as mentioned previously, we intend to continue outreach efforts and capture public comments in order to ensure we capture as much stakeholder input as possible to be reflected in the PQCMC standard. And finally, how can you learn more? We have a project page at fda.gov, and we plan to keep updating this periodically. We, as mentioned previously, are intending to have continued releases of more structured parts of PQCMC for public comment. There is a FIRE project page for PQCMC at hl7.org. This is a clickable link. And as mentioned previously, this is done under the auspices of the Biomedical Research and Regulatory Work Group at HL7. And finally, we do have a standing email address, pq-cmc@fda.hhs.gov, which you can use for any and all questions you may have about the PQCMC initiative. And that brings me to the end of my presentation. I'd like to thank you all very much for your attention.
Let's now turn our attention to the future of application submission and application review. After this presentation, you should be able to describe the PQCMC vision, explain what's been done so far, describe the role of these two standards organizations, explain our ongoing work and its challenges, and describe what you folks might do to prepare for the future of structured applications. So the current model could be said to be a narrative submission going to a narrative review or assessment from which data such as component and composition is manually entered into a FDA database. Then the PDF documents are stored in our electronic document room or the document repository. What might be called a future database model of submission would involve both structured Module 3 documents along with some unstructured narrative. For example, how it is that you arrived at the wet granulation model as compared to direct compression. The structured documents, as before, would be stored in our electronic document repository. but structured data elements such as components and composition would be automatically extracted into an FDA knowledge base for multiple uses, including to pre-populate CASA template review templates and to populate our facility risk ranking algorithm for inspection purposes. An important feature here is the 2-year publication timeline for when PQCMC would be a mandatory format. In this slide, I hope to clarify the distinction between PQCMC and CASA. Briefly stated, PQCMC is about standardizing submissions and CASA is about standardizing assessments with templates pre-populated from data that's retrieved from PQCMC submissions. So PQCMC's goal is to standardize and structure the quality information in the ECTD using data elements, data standards such as XML, JSON, and HL7 FHIR. CASA, on the other hand, is a system of internal knowledge management involving pre-population of review templates, risk ranking algorithms, and data analytics that will facilitate lifecycle management in comparison to previously submitted documents. In the end, implementation of PQCMC will enhance the current CASA system, which relies largely on manual population of the CASA template. So the important takeaway message is that PQCMC is about submission and CASA is about assessments. Here's a summary of where we are, having assembled a diverse range of subject matter experts across the 3 centers. We've created data models for the listed elements and standardized terms and definitions, relying first on existing standards, for example, from FDA, ICH, IUPAC, USP, and EMA. We have had a proof of concept with 5 pharma companies where we tested the FHIR instance could be successfully sent through our electronic gateway and that it did adequately represent the data prior to its transformation. We have worked diligently to harmonize with the CASA group, although initially we worked separately. We're also collaborating with internal FDA standards groups, which I'll mention subsequently. So what we've published is 2 Federal Register notices in 2017, which laid out our Phase 1 terms and definitions. The 2 main comments were related to expanding PQCMC beyond FDA efforts, which are described on the next slide, and conforming to the ISO identification of medicinal product standards, for which we created a document with a term-by-term mapping. And we also held a Zoom with pharma to engage in an interactive discussion. Later in my talk, I'll discuss some of these challenges with IDMP. And here are some references to IDMP and to our Federal Register notice, and a reference to our FDA webpage that contains PQCMC information. Along with Health Canada, we proposed an ICH topic on structured data quality. This proposal was accepted as a new topic called Structured Product Quality Submission to start after progress is made on the CTD-Q revision by the M4Q working group, which is ably chaired by Lawrence Yu. We anticipate next year as the start date for the SPQS group. But we'll continue with our efforts in PQCMC because we need them to support the CASA initiative. The history of the IDMP standards is that they were spun out of ICH pharmacovigilance groups in the early 2000s. Subsequently, the scope has expanded to cover exchange of all medicinal product data, for example, by creating a global unique identifier for a medicinal product known as PHPID. The 5 standards listed cover substance, dosage form, routes of administration, units of measure, and other medicinal product information. What conformance to IDMP means is not so straightforward. Does it mean having the same data model? Can there be the same data model with different terms? The EU, for example, mandates the use of IDMP, and yet the EMA used different term lists for ingredient roles. An effort to map dosage forms with EDQM resulted in only a 30% overlap of terms, and medicinal product ID is left to a regional implementation, and no other country, for example, uses our NDC code. And FDA requires USP dosage form labeling, for example, and even within FDA, we have several different dosage form lists that are in use. So the conclusion is that we'll try to align where possible with IDMP, but the conformance is just frequently not possible. The necessity for standard terms and definitions is to eliminate confusion about synonyms and the exact meaning of the same term used in different contexts. For example, until recently, the term active ingredient had different meanings within two sections of the Food, Drug, and Cosmetic Act. But standardization will not only facilitate a ontology or relationship between terms, but it will facilitate data analytics as given in the example below. It also will facilitate risk ranking. For example, does wet granulation give a better content uniformity than direct compression for low-dose drugs? So for us, we use these control vocabularies to create drop-down lists. For example, ingredient role would be comprised of these three items. To provide you with a specific example, here's what we did for drug product manufacture. By creating a model based on unit operations, what are those elements associated with the unit operation? They are category and subcategory, for which we had a SUPAC IR equipment addendum, which I'll mention was a great collaborative effort between FDA and industry, and for which there was a Hammer Award. So we also associated with a unit operation these elements like equipment data, process parameters, established conditions, in-process control, step ID, and material input. We had discussed adding established conditions, but because of the newness of this concept and the current work of the M4Q group, we decided to defer it and to not include it in the current PQCMC model. So here's an idea of what we do in our PQCMC collaboration with the CASA group, in which we look at common terms, different terms with the same meaning, and come up with a consensus on a given term, define it, and decide which group will make that change. Our current and future work involves these many external collaborators. here, and here. And many standards groups within FDA. We're pushing ahead into other sections. We have a JSON-based model for drug synthesis that renders a graphical synthetic scheme which builds in chemical intelligence and is not simply a picture. We focused so far on solid oral dosage forms, but we're now expanding into liquids, including biotech products such as monoclonals, with input from our Office of Biotech Product subject matter experts. And finally, late next year, we plan to publish a PQCMC draft guidance that will likely focus on specification, components, and composition. There are IDMP challenges in the road ahead, including its initial focus on pharmacovigilance, a different specification model, and use case, many undefined terms, and I would note that our contractors, however, required us to come up with a definition for every term we used, and every regional control vocabulary list. We had to have terms for each of those. And then there are many PQCMC related terms that are not in IDMP, which really had no focus on drug manufacturing. So some of those are shown in this list. Other challenges we face include too many standards and too few standards. Too many in some of the listed examples, and too few as listed in the gaps. Developing these data models, definitions, and ontologies is time-consuming and resource-intensive process when done in a systematic way. Another issue is that we have yet to see any commercial vendors supporting FHIR for regulatory submissions, although there's a lot of FHIR support in the EHR, electronic health record arena, and in the clinical area. And of course, we need to build database structures to support these incoming submissions. So in conclusion, PQCMC will substantially change things. It will result in significant changes for FDA and applicants. There'll be a need for new business processes and infrastructure. And we hope that PQCMC will enable alignment and be embraced by IDMP. And as for the future, 745A mandates 2 years after the final guidance. And remember, we haven't published a draft guidance yet. And the Step 2, there's an uncertain time frame for any Step 2 document from SPQS. So because these changes will implement years in the future, there's still time for you to prepare. And about preparing, some suggestions that I have are to maintain current awareness of these efforts through things like Google Alerts, looking at YouTube, which has a lot of videos from FDA, EMA, and Unicom, which is a European project involving IDMP, and of course look to the FDA webpage. Other things you can do are apply the master data concept of a database with a single source of truth for a term. Group your data according to IDMP concepts. You might create a new field, administrable dose form, which is an IDMP concept, and just do data cleansing and curation. So here are a couple challenge questions. Which of the following statements is true? PQCMC is primarily about application assessment. CASA is primarily about application submission and their assessment. PQCMC and CASA are primarily about application submission and their assessments, or PQCMC will provide the structured data needed for CASA assessments. So I'll give you about 5 seconds to think about this. And the answer is PQCMC will provide the structured data for the CASA assessments. Of course, PQCMC is not about application assessment. Okay, moving to our next challenge question. Which of the following is not true? PQCMC submission format will become mandatory in 2025. PQCMC attempts first to use existing data standards. PQCMC is now an FDA-only project. PQCMC has published 2 Federal Register notices. I'll give you a couple minutes to think about this. And the answer is PQCMC will become mandatory in 2025 is not correct because we only anticipate publishing a draft PQCMC guidance next year, and that will be followed sometime later with a final guidance, and the final guidance then will take 2 years. So thanks for your attention, and we'll handle some questions at the end of the session.
Good morning, everyone. My name is Lieutenant Commander Nimi Matthews. I'm a Regulatory Project Manager with the Division of Project Management in the Office of Generic Drugs. As a project manager, I review incoming submissions daily, and a significant part of that process is our triaging based on the information provided in the accompanying cover letters. Today, I would like to share with you all a few suggestions regarding the information to include with your cover letters. Our learning objectives today are the following: to understand the purpose of a cover letter and the cover letter attachment, to examine FDA-issued guidance as it pertains to both these documents, to evaluate pertinent information to include in the cover letter based on the submission, to discuss the resources available to help create an effective cover letter for the submission's purpose. At this time, I also wanted to point out that these recommendations and suggestions shared today are consistently being updated via new guidance and maps, so please refer to those for the most current information. To begin our discussion, I wanted to go over the purpose of cover letters. So what is the purpose of a cover letter? The cover letter provides us the initial snapshot into the submission. It helps provide a summary of contents, identify the purpose, such as a response to an IR, resubmission after an action. It highlights the key elements included in the submission. It provides applicable regulatory statements, such as the MMA verification. And lastly, it helps FDA route and manage these submissions effectively. Similarly, let's define the purpose of a cover letter attachment. The cover letter attachment serves as a guide to help prepare the cover letter and submission. So it's very different than the actual cover letter. It serves as a tool to help prepare the cover letter and the submission. It is designed as a checklist for the common types of information applicants include in their cover letter. The goal is that this checklist format will help ensure that all relevant information outlined in the checklist is addressed in the corresponding cover letter. As mentioned earlier, this tool also helps FDA triage and manage the submission without unexpected delays. A sample of this checklist is provided in the draft guidance titled Cover Letter Attachments for Controlled Correspondence and Candidate Submissions, a link to which is provided at the end of this presentation. The use of this tool or its submission to FDA is not required, but it is recommended to help create a complete and accurate cover letter. Next, I want to share a few guidance for industry available on this topic. Our first one is the one for ANDA submission, content, and format, which was finalized in June 2019. As the name suggests, this document contains non-binding recommendations of the contents and format of an ANDA submission. As an applicant, you can always use an alternative approach to what is outlined in this guidance, as long as it satisfies all the requirements of the applicable statutes and regulations. What is really helpful in this guidance is the suggested cover letter template included in the appendix of this guidance, as well as the recommendations it provides on what should be generally included in the cover letter. On page 4 of this guidance, it provides some information about the cover letters, which is included in Module 1, with the other administrative information of an electronic common technical document submission. It also provides a non-exhaustive list of some of the major changes to an application that should be included in the header of the cover letter in order to avoid any triaging delays. Some of these include new strength amendments, changes to a dosage form, etc. The appendix I mentioned earlier provides a template for the cover letter. Not all the paragraphs recommended in this template need to be included in your cover letter. Only pick the applicable ones for that specific submission. It is highly recommended that you adapt the cover letter to meet your needs and submission type. You could also create your own template from the provided template and modify it for each of your submissions. The next guidance I wanted to share today is the one titled Cover Letter Attachments for Controlled Correspondence and ANDA Submissions. The draft version of this guidance was released December 2021. Appendix 2 in this guidance provides a cover letter attachment template, which is, as with the other templates, it is highly recommended that you modify the template to meet your submission needs. Of note, the cover letter attachment does not replace the cover letter itself and should be used as an add-on or a guide to create the perfect cover letter. The last guidance I wanted to bring your attention to today is the one titled ANDA Submissions, Amendments to Abbreviated New Drug Applications under GDUFA, which was finalized in July 2018. On page 15 of this guidance, it includes useful information regarding the contents to include in the cover letter, which we will cover in further detail in the next few slides. As with the other guidance, use the template and the information provided in the guidance and tailor it to meet your needs and your submission needs. Additional information to include in the cover letter can also be found in the specific guidance for that submission type or a specific map. FDA also has a quick access page listed here on this slide for all the available guidance and sections of the FDA maps that you may need to refer to. Our next topic of discussion are the components of a cover letter. The points I discuss on the next few slides are recommended for all submission types. First one is it is recommended that the cover letter be issued on the company letterhead. It should indicate the type of submission, such as a CR response, an IR, or a DRL response. It should include the date of the submission and reference at a minimum the ANDA number, the name and strength of the drug product, as well as the sequence number of the submission. Continuing on with our recommendations for all cover letters, the cover letters should additionally include a statement on how the documents were submitted and the file structure, the name, signature, and contact information of the person submitting this information, a regulatory and technical point of contact for the submission, including an email address, and reference, if any, to a relevant FDA action letters, emails, or correspondence. Of note, if the cover letter is signed by someone other than the responsible official or the U.S. agent, it should still include the name and contact person listed as a U.S. agent or the responsible official on the 356H form. Continuing on with our recommendation for all submissions, the cover letters should include a regulatory description of the submission, including an appropriate regulatory information, and any desired hyperlinks to the submitted information. It should include a MMA or verification statement. It should include a technical description of the submission, including the approximate size of the submission, as well as a statement stating that the submission is virus-free with a description of the software used to check the files for that virus. Moving on to recommendations based on the submission type, please always refer to the applicable guidance for that specific submission type. Additionally, some submissions, like withdrawal requests, require that you reference the aspect of the CFR that is the basis of the submission in the cover letter. Provided here are two examples of how to use the header and references on a cover letter. Note how all the pertinent information is provided upfront, which is tremendously helpful during the triaging process. Now that we have covered a lot of information regarding what needs to be included in the cover letter, this is a good time to pivot and share some of our frequently observed errors or omissions. First up is missing the MMA verification statement as required in 21 CFR 314.96 which requires amendments to an ANDA to contain the appropriate patent certification, recertification, or statement if the amendment contains new indication, new strength, changes to product formulation, or a change in the physical form or structure of the active ingredient. If the amendment does not contain any of these, the applicant should include a verification statement noting that the amendment does not include the changes I just mentioned. But one or the other is required for most types of amendments. To avoid delays and to err on the side of caution, I recommend adding that verification statement to every amendment. Next one is missing the priority request on every resubmission, even after it was granted. Resubmissions open up new projects in our database, and noting that a priority was requested and granted on the cover letter helps project managers ensure that the correct goal dates are assigned. Sometimes project managers assigned to the ANDA change, and this information upfront on the cover letter help avoid delays and errors during triaging. Next, highlighting any unsolicited information or major amendments in the cover letter upfront allows FDA to accurately assign a goal date upon the receipt of the amendment and reduces the later changes in classification when an unsolicited information or major information is discovered during the review of the submission. Some examples of this include gratuitous labeling information that is necessary for an approval. Next up on our list is not noting new or revised patent certifications, litigations, or carve-out updates. An additional point here is when including carve-outs in a cover letter, I recommend bolding them, having them in bolded font so they are easily identifiable and addressed accordingly. Following that is tucking in major changes or significant information on latter pages of the cover letter versus having them up front and on the first page, such as including a new strength amendment on page 10 of a lengthy cover letter versus having it right up front in the header of the cover letter where it's easily identified. And last on our list is not noting major amendment information upfront. A few examples of major amendments are new batch studies in response to deficiencies, changes in manufacturing sites. So the changes are made on the 356H or in the quality section, but it's not noted on the cover letter for us to triage that amendment appropriately. Any changes to a DMF or changes that would require an additional filing review, like a new strength amendment, but it's not noted on the cover letter. Instead, it's just noted in the body of the actual submission. For a full list of major amendments that are considered major. A full list is available in the guidance and the submissions amendments to abbreviated new drug applications under GDUFA, and it's in Appendix B of that guidance. Please refer to that guidance, and if any of your submissions fall under one of those categories, please remember to note and tag that cover letter with a major amendment notification clearly identified in the header. Now that we've covered some of the most common errors, let's move on to best practice. First up on our list of best practices is to include all new or major changes, including any labeling updates, right in the header or reference if you are combining submissions. Oftentimes, labeling updates are included in submissions along with other information, but not identified on the cover letter, and this can lead to delays in labeling information being reviewed. Always remember this. If the information submitted can in any way change that goal date, please put that on the header or reference section to be on the safe side. We highly recommend you create your own cover letter and cover letter attachment template to include all the information typically included in your submissions. It does two things for you. It reduces chance of leaving out any pertinent information required for most submissions, such as an MMA or a verification statement. It also helps you take out the inapplicable information and tailor that template or that cover letter for that specific submission. Next up is highlight any significant elements of your submission right at the beginning of your cover letter, as well as order your changes or order your submission from major to minor changes. So all the major changes noted up front and not lost in the latter pages of cover letter, as we mentioned earlier. Always remember this. If the information that is included can change the goal date, please put it in the heading or the reference section of your cover letter. Highlighting any unsolicited information or major amendments in that cover letter up front allows FDA to accurately assign the goal dates upon the receipt of that amendment, and it reduces the later changes in classification where we identify unsolicited information or major information during the course of the review. Continuing on with our best practices, please prominently identify or bold in your cover letter if a labeling carve-out is part of that submission. Separate each item in a paragraph of its own. Be concise, especially if more detail is provided within other modules. And use keywords rather than a vague description of the content. For example, using the keyword reformulation versus stating changes to composition of the product. Get familiar with the types of changes or information that can affect your review process and ensure that those changes are always noted on your cover letter body and header. For example, having keywords such as a new strength amendment or clearly stating that one of your facilities is not ready for inspection helps us triage that amendment accordingly. Bold the text of any administrative requests that are combined in with the data submitted for review. For example, if there is a request for reconsideration submitted along with the CR letter, please bold those so those are easily identifiable to the reviewers as well as the project managers. Use a cover letter and a cover letter attachment when combining submission types, so you don't have a 3-page cover letter, and if the project manager reviews the cover letter attachment, you can easily see all the different types of submissions that are combined into that one amendment. This is new for GDUFA III. So when requesting a change in CR classification for facilities only CR, using the correct keyword is just as important. So for facilities major to minor requests, include the keyword facility only or facility-based reclassification. We recommend putting this in the header as well as the body of the letter. And you can do that with the other recommendations I've mentioned earlier as well. Put those key words in the header as well as in the body. And this new process is applicable only to CR responses and should be included with the CR response to be considered. Now that we've come to the end of our presentation, let's do a quick knowledge check. Challenge question 1. Which module of the ECTD submission is the cover letter contained in? Module B, Module 2, Module A, or Module 1? And the right answer is Module 1. The next challenge question is a true or false statement. The FDA cover letter template provides information that is required for each submission. A, true, B, false. And the answer is false. This is because the FDA provides a general recommendation of everything that needs to be included in a cover letter, but it is up to the applicant to modify that template and to adapt it to meet your needs and to only include pertinent information to that specific submission. Our challenge question number 3 is another true or false question. The FDA cover letter attachment can be used instead of the cover letter for the submissions. True or false? And the answer is false. As stated before, the cover letter attachment does not replace the need for a cover letter to accompany each submission. In summary, the applicant should use the cover letters and the cover letter attachment to help FDA identify the content and purpose of the submission. While FDA provides some guidance for cover letter and the attachment, the applicant should tailor their cover letter to meet their specific needs and the regulatory requirements. The cover letter should help guide the FDA on how to route your submission for an appropriate review. The cover letter should clearly state any significant changes to the application, such as formulation change or new strength amendments, right in the heading and the body of the cover letter. Cover letter attachment is an excellent tool to ensure all pertinent information is addressed in the cover letter and the submission. I wanted to share a few of our resources today with you, and these links listed on this slide will take you to our landing page for guidances, maps, the ECTD guides, as well as the CFRs. And listed here are the direct links to the guidances we talked about today. And as always, if you have any questions regarding the cover letters or the templates during the course of your submission, please do not hesitate to reach out to the regulatory project manager assigned to your ANDA. That brings us to the end of my presentation. Thank you all for your time and attendance today. I hope the information I shared with you is helpful and useful to you.
Thanks so much for all the great presentations. We'll now move into the Q&A panel session. And please remember, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. And as another reminder, please remember to indicate at the beginning of your question the presenter that you wish to respond to your question. We'd like to welcome back to the Q&A panel Lieutenant Commander Malik Imam, who's the Deputy Director of the Office of Regulatory Operations within OGD, and Commander Andrew Fine, who is a Senior Advisor in OGD's Office of Safety and Clinical Evaluation, Division of Clinical Review. Looks like we have some questions coming in right now, and the first questions will be addressed to Commander Mahavathan. And here's the first question for Commander Havathan. Can we submit other related documents and the DRL response impacted due to the query along with documents requested by the agency in order to avoid further queries related to the same?
Thank you so much for that question. In general, please respond with the requested information only, which will include any related documents that may help answer the DRL deficiencies. Note that if you submit any information that's not requested by the agency, the goal date may be extended. Thank you.
Thank you for responding to that question. We do have another question for Commander, and here is the question. What if I have a question for the agency and if it fits in multiple submission categories like a controlled correspondence or meeting, how do I know which submission path to choose?
Thank you. That's a really great question. So if you're unsure of the submission category, it's recommended to select what you think is the best pathway. If the agency determines that a different pathway is more suitable, we will suggest that to the applicant rather than just denying the request. And as a reminder, please do not submit the same question using multiple submission pathways. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a couple of questions that came in from Captain Keister. And this is a long one, so I can certainly read it if we need to. And here we go. If an establishment has been submitted in sequence as 0000, it is assumed that the answer to the question would be yes for is the establishment new to the application, and pending for what is the status of the establishment. But after the original sequence, if an amendment to an IR is filed as sequence 0002, still an unapproved ANDA, and if there are no changes to the establishments in the application, how is the circled question to be answered in this sequence? That is, for the question, is the establishment new to the application? Should the question be answered no? And the second question, what is the status of the establishment, remain as pending? Captain Keister, we're having problems hearing you. We're going to move on to the next panelist and let our tech team reach out to you to work with the audio. Moving on to our next panelist, we do have a few questions that came in for Hui Zhang, and here is the first question. Is it acceptable to use the test product in IVRT method validation?
Thank you for the question. Yes, it is acceptable. During the method validation, both the test and the reference products can be used. In addition, the applicants need to manufacture an altered formulation to use it to demonstrate the supplemental selectivity of the IVRT method. Thank you.
Thank you for responding to that question. We do have one more question for you, Hui Zhang, and here is the question. Should the applicant submit separate 2.7 tables for method development, validation, and pivotal study in ANDA?
Thank you for the question. Yes, please submit separate summary tables for the method validation, development, and the pivotal studies. Thank you.
Moving on to our next panelist, we've got a couple questions that came in for our Chana Manriker. And here is the first question. Would you please share the general experimental conditions that can be used to conduct IVPT robustness, as the general guidance does not include this information?
Yes, thank you for the question. As discussed in the IVPT General Guidance, inherent variability in the permeability of human skin, whether in vivo or in vitro, is incompatible with the primary assumption related to robustness testing, which is that the test system performs consistently when all system variables such as temperature, stirring rate, etc. are at nominal settings. Therefore, while recommendations for alterations to study conditions for the purpose of demonstrating method robustness can be provided for IVRT studies, the same cannot be provided for IVPT. Nevertheless, over the course of your IVPT method development, if you note that the permeation profiles or IVPT endpoints are robust to certain variations in method parameters, such as, for example, stirring rate or the applied dose amount, then complete details of those findings should be reported in support of method robustness. Thank you.
Thank you for responding to that question. We do have another question that came in for Chana Manarikar. And here is the question. Can you clarify what evidence should be submitted to support consistency of study procedures and conditions and control method of parameters across the method validation and pivotal studies?
Thank you. This is a great question. As noted during my presentation, scientifically justified and optimized IVPT methodology, including suitable method parameters, study conditions, and study procedures that ensure precise control of method parameters, should be established during method development. Thereafter, these well-controlled study procedures should be meticulously documented in SOPs for use in the subsequent validation and pivotal studies. Accordingly, across these validation and pivotal studies, IVPT method parameters and environmental conditions, such as those described in Appendix B in my presentation, they should be monitored over the IVPT study duration, and evidence demonstrating their control within adequately justified and pre-specified tolerances should be submitted. Now, adequate control of method parameters over the study duration across each cell and each IVPT experimental run can be achieved when the corresponding study procedures are also similarly well-controlled and consistent. Thus, demonstrated control of IVPT method parameters along with detailed descriptions of how each study procedure will be performed and controlled can support a conclusion that the corresponding study procedures are also well-controlled and consistent. So to give you an example, relevant to validation and pivotal studies, evidence of adequate precision and accuracy of the applied dose, the actual applied dose, which is basically dispensed dose minus any dose that is lost during the application process, along with granular description of the dosing procedure and how it is controlled in the relevant SOPs, together can be supportive of the consistency and control of the corresponding procedure, which is the dosing procedure. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we've got a couple of questions that came in for Vipra Akundor. And here is the first question. Do you know when the BE summary tables for the MDRS study will be posted?
Thank you for the question. Currently, we are working on developing the BE summary tables for the MDRS study. However, we don't have a timeline for when they will be finalized. So please note that the tables, when finalized, will be posted on the fda.gov website. Therefore, we strongly encourage you to check the FDA website for the most current recommendations. Thank you.
Thank you for responding to that question. We do have one more question for Vipra Kundur, and here is the question. Can you elaborate on what validation information should be submitted for the selection of circular equivalent diameter data?
Thank you for the question. For the CE diameter, we recommend that you provide validation data to justify the use of CE diameter that is not blind to significant % of the API particles. We also suggest to provide specificity, accuracy, precision, and robustness of the API particles using the selected CE diameter. Thank you.
Thank you for responding to those questions. We're going to check back in with Captain Keister and see if the audio works out. And here's the question that we previously asked, but we're going to ask it again since it's so long. If an establishment has been submitted in sequence 0000, is it assumed that the answer to the question would be yes for is the establishment new to the application, and pending for what is the status of the establishment? But after the original sequence, if an amendment to an IR is filed as sequence 0002, still an unapproved ANDA, and if there are no changes to the establishments in the application, how is the circled question to be answered in this sequence? That is, for the question, is the establishment new to the application? Should the question now be answered no? And the second question, what is the status of the establishment, remain as pending?
Thanks, Ray, and sorry to have to make you read that one twice. But in short, yes, what the questioner suggested would be correct. On initial submission, it would be new and it would be pending. Assuming there are no other changes to that application, to that facility rather than establishment, future submissions, you should not list it as new. That would, because it's previously been submitted to that application. And yes, it would still be a pending status.
Thank you for responding to that question. We heard you loud and clear, so we'll try one more question for you, Captain Keister. And here is the question. If I need clarification of deficiencies received in our CR, should I submit a post-CR scientific meeting request and a controlled correspondence?
Thanks for that question, Ray. For clarification questions, you can submit a request for a post-CRL clarification TCON or a controlled correspondence, but you should not submit both. If you also have scientific questions related to the equivalence, you can submit both these questions and a clarification question as part of a post-CRL scientific meeting.
Thank you for responding to that question. Moving down the line to our next panelist, we have a few questions that came in for Scott Gordon. And here's the first question. For HL7 FHIR, is the idea to eventually reduce the amount of PDFs submitted in Module 3 of applications? Dr. Gordon, we're having difficulty hearing audio. We're going to let you reach out to our tech team, let them check in with you, and then we'll check back in a few moments. We're going to move on to our next panelist. And we have a couple more questions that came in for Norman Schmuff, and here is the question. It's a little bit longer, so I'll try to read it slowly. I could understand how a description of a manufacturing process could be developed such that reviewers can have a complete understanding. However, that the document would not be like any existing master batch record. Is there a vision for how that regulatory document would relate to the master batch record that is on the manufacturing floor, driving the steps of the chemical operators and recording the relevant information in the executed batch record?
Yeah, it appears now that that information that we had structured in the manufacturing section would really have to be manually input into some application tool. In the future, for particularly as it relates to electronic master batch records and electronic batch records generally, we would anticipate that there would be an automated process where those items that would be submitted in the application, in the structured application, would be extracted in an automated way from the master batch record. But right now, and probably in the near future, it would require manual input and then an additional PDF file of the master batch record.
Thank you for responding to that question. We do have one more question for Dr. Schmuff, and here is the question. For PQ-CMC, whether the current ECTD publisher needs to modify their publishing tool, or is this needed for are any additional software tools needed?
Well, the initial, the ECTD publishers have to be updated for version 4 of the new ECTD. But to get to this point of whether new ECTD tools will have to be available for structured submission, it may be that there is some back-end tool that's not actually part of the current ECTD ECTD tool that does the direct submission. For example, many of these files will just be, we anticipate they'll be XML and JSON files. So XML and JSON files presumably will be handled by any existing ECTD system that will handle either version 3 or the newer version of the ECTD system. So I don't anticipate that there'll be changes to that ECTD system per se, but you'll need some additional back-end tools.
Thank you for responding to that question. We're going to check back in with Scott Gordon to see if the mic and everything is working okay. And here's the question. I'm going to reread it. For HL7 FHIR, is the idea to eventually reduce the amount of PDFs submitted in Module 3 of applications?
The goal isn't to reduce PDFs per se, but to reduce to reduce the amount of unstructured, non-computable data coming in Module 3. So the goal really is to increase the amount of computable and structured data. So it's not about any one specific format, it's about whether it's computable and structured. Thank you.
Thank you for responding to that question, Dr. Gordon. We do have another question for you. We're able to hear you loud and clear. Here's the question. How can we learn more about the FHIR development of PQ-CMC?
That's actually a really good question. It is I think I mentioned, an official project at HL7 under the auspices of the Biomedical Research and Regulatory Work Group. In fact, I think if you go to slide 17 in my slide presentation, if you download that, there's a link to the PQCMC FIRE project page. This is the HL7 project. And that actually brings you to the Confluence page, which will give you information on how to join weekly, if you'd like, meetings of the Biomedical Research and Regulatory Work Group at HL7. Just a note, do look at the agendas. There's many projects at that work group. But on occasion, we'll be discussing things like PQCMC. It is a public forum, so obviously we would welcome all sorts of new participants. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We've got a couple of questions that came in for Commander Anemi Matthews, and here's the first question for Commander Matthews. Changes in dosage form. Did you mean to say changes in formulation? And the questioner provided a note, usually a change in dosage form would require an approved suitability condition, a suitability petition, and a new ANDA.
Thank you for the question, Commander. And you are correct. I meant to say changes in formulation and not a change in dosage form. So thank you for the very attentive listener and the question.
Thank you for responding to that question. We do have another question for Commander Matthews, and here's the question. If a labeling update is made due to changes requested in deficiency, is the labeling update considered unsolicited information?
So any new information outside of what was requested in the deficiency letter is considered unsolicited.
Thank you for responding to that question. We do have another question for Commander Matthews. It seems it'll be short, so here's the question. What is the MMA acronym meaning?
The MMA acronym stands for the Medicare Prescription Drug Improvement and Modernization Act of 2003.
Thank you for responding to that question. Moving back up to the beginning of our panel, I have another question that just came in for Commander Chitra Mahivathan, and here is the first question. Clarify in what instances is it more appropriate to submit a controlled correspondence versus a meeting request post-complete response letter, please?
Thank you so much for that question. So a controlled correspondence may be submitted for clarification questions or any questions that may be considered out of scope for a meeting. You can also submit a controlled correspondence after a teleconference if you're seeking further clarification or if you have any new or additional questions rather than requesting a meeting. And also post-complete response letter, you can submit a controlled correspondence to seek regulatory or scientific advice or to request evaluation of alternative BE approaches. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a question that just came in for Hui Zhang, and here is the question. For IVRT in a post-approval supplement, SUPAC-SS, do the same recommendations apply regarding information and reports to provide and placement in the ECTD structure?
Thank you for the question. The information presented today is applicable for IVRT studies with the purpose of establishing bioequivalence. If you submit IVRT data in the post-approval supplement for the purpose of drug product quality control, then the information should be submitted in Module 3. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a new question that just came in for Vipra Kundur. And here is the question. Can an in vitro study be an alternative to BE for nasal sprays?
Thank you for the question. For nasal sprays, the applicants may submit an alternative study proposal to demonstrate bioequivalence of their drug product. However, we strongly encourage that the applicants submit the specific alternative study plan to the agency prior to submitting an ANDA via appropriate pathways. Thank you.
Thank you for responding to that question. We have a couple more questions that came in for Chana Manarikar. And here is the first question. What information is expected to adequately support the qualification of receptor solution sampling?
Thank you so much for the question. As discussed in the presentation, the expectation here is that the suitability of the receptor solution sampling procedure is adequately qualified. So specifically, relevant to the IVPT validation and pivotal studies, adequate qualification or evidence should be supported in support of the following key aspects. So first is that the sample aliquot volume withdrawn at each sampling event is precise and accurate. 2, the sampling procedure is appropriate. And 3, the optimized IVPT test procedures ensure adequate sampling time precision from cell to cell within each experimental IVPT run. Thank you.
Well, that's all the time we have for questions in this session. A huge thank you to our presenters and panelists for the great presentations and answering numerous questions that came in. We'll now enter into our break for 10 minutes, returning at 11 a.m. Eastern Time. Enjoy your break. Welcome back from the break. We hope you had time to refresh your coffee to prepare you for our next presentation. First, we'll present on GDUFA III labeling updates and tips. This presentation will highlight GDUFA III updates that impact ANDA labeling and provide tips on ANDA labeling submissions. Our first presenter, Commander Julie Nishiwa, is a supervisor in the Division of Labeling Review in the Office of Regulatory Operations within OGD. Our next presenter in this session is Dr. Kimi Odesina, who's a senior labeling reviewer in the Division of Labeling Review in ORO and OGD. She joined CEDAR in 2014. Our final presenter in this presentation, will be Lieutenant Commander Cody Ochiozo, who serves as a labeling reviewer in OGD. She plays a critical role in advancing FDA's public health mission by mitigating errors on medication labeling to promote the safe and effective use of generic drugs. Our next presentation is an overview of major quality deficiencies and approaches available in GDUFA III. Our presenter, Karen Ireland, serves as a senior regulatory business process manager in OPQ's Office of Programs and Regulatory Operations. She's been to the FDA with OPRO since 2017. She has expertise in supporting the quality review of pre-market ANDAs and has represented OPRO in working groups for guidance and changes in the GDUFA III program. Our final presentation before the Q&A panel is on drug product quality tips, drug device combination products. Our presenter, Dr. Kwok, is a senior pharmaceutical quality assessor in the Division of Liquid-Based Drug Products. In this role, he acts as the application technical lead for integrated quality assessment of generic parenteral, ophthalmic, topical, oral, and inhalation solution drug products. For the past 7 years, he's been reviewing ANDA, bio-IND, and pre-ANDA meeting packages involving complex drug products. Also, he served as an FDA liaison to the USP Packaging and Distribution Expert Committee for developing USP packaging chapters and standards as a member for the development of FDA guidance for drug delivery performance of drug device combination products. Now, please join me in welcoming our first presenter, Commander Nishiwa.
Welcome, everyone. I am Commander Neshwat, and I am excited to start the presentation on GDUFA III labeling updates and tips. The objectives of this presentation are to discuss generic drug user fee amendments, or GDUFA III updates that impact Abbreviated New Drug Application, or ANDA labeling. We will also discuss best practices for ANDA labeling submissions, including best practices specific to ANDA supplements. GDUFA was signed into law on July 9, 2012. GDUFA agreements are negotiated by FDA and representatives of the generic drug industry to address regulatory challenges. GDUFA is authorized every 5 years, with the most recent reauthorization being for fiscal years 2023 to 2027, also known as GDUFA III. Labeling is an important component of GDUFA III and the generic drug program. The Division of Labeling Review, or DLR, within the Office of Generic Drugs, ensures that the generic drug labeling is the same as the approved labeling for the product's reference-listed drug, or RLD, except for differences allowed under Section 505J 2A5 of the Act and 21 CFR 314.94 A84. DLR also ensures that the labels and labeling accurately reflect the drug product information and provide sufficient information for safe and effective use of the drug product. DLR has several commitments under GDUFA III. In the first assessment cycle and after receiving an ANDA for assessment, DLR will determine whether a consult needs to be issued to another review discipline and then initiate such consults. DLR will strive to issue the labeling discipline review letter, or DRL, at months 6 to 7 for ANDAs with a 10-month goal date, and at months 5 to 6 for ANDAs with an 8-month goal date. DLR will limit the labeling assessment to one information request, or IR, or one DRL if other disciplines will not be acceptable during the first cycle. DLR will continue to assess labeling to enable an action within the assessment cycle if other disciplines are acceptable. Issuance of complete response letters that contain only labeling deficiencies will be minimized by utilizing later cycle IRs and DRLs, as well as the imminent action process. Now we'll go over best practices as it relates to GDUFA III timelines and labeling. As mentioned previously, one commitment under GDUFA III is to determine whether a consult needs to be issued to another review discipline and to initiate such consults. An example of a potential situation that could lead to a consult includes labeling where the applicant has proposed to carve out information protected by patents and/or exclusivities. To help facilitate identifying possible consults and addressing these carve-out issues earlier in the assessment cycle, we request that applicants prominently identify, e.g., use bold font to indicate labeling carve-outs in cover letters. Other tips include addressing all patents and/or exclusivities listed for the RLD, and also ensuring the proposed labeling carve-out is consistent with patent certifications and exclusivity statements. We also recommend monitoring available labeling resources and making necessary revisions to labeling. Drugs at FDA is the official source for approved labeling, so we recommend checking Drugs at FDA routinely and prior to submitting labeling amendments to ensure that your proposed labeling is the same as the last approved labeling for the RLD except for differences outlined in Section 505J of the Act and the CFR. We recommend monitoring the Orange Book for any updates to the RLD's patents and exclusivities. As soon as you identify updates to the RLD's patents and exclusivities, we recommend being proactive in addressing the new patents and exclusivities and submitting labeling that is consistent with the new patents, patent certifications, and exclusivity statements as applicable. We also recommend checking the United States Pharmacopoeia National Formulary for updates to drug product monographs and USP general chapters. In any of these cases where applicable information is posted on Drugs at FDA, Orange Book, or USPNF, we encourage applicants to go ahead and submit labeling as appropriate to address the updated information in these resources. Please do not wait to receive a DRL to address these issues. Be proactive and identify and address these updates as soon as possible to allow timely review of your labeling. Now we have our first challenge question. Which resource should be used to find the RLD's last approved labeling? Is it A, DailyMed, B, Orange Book, C, Drugs at FDA, or D, USPNF? I will give you a few seconds to think about the correct answer. For those that pick C, Drugs at FDA, you are correct. I want to emphasize that Drugs at FDA is the official source for approved labeling, so please use Drugs at FDA when finding the RLD's last approved labeling. I did also want to note that while DailyMed does post labeling on their website, it may not always be the last approved labeling, and therefore, Drugs at FDA should be the primary resource for finding the RLD's last approved labeling. Now, I will hand it over to Lieutenant Commander Ichozo for the next part of the presentation.
Thank you, Commander Neshwat, for the introduction. During this portion of the presentation, I'll be sharing best practices for ANDA labeling submissions. If you're looking for golden nuggets, I'd encourage you to continue to stay tuned in. As a quick highlight, over the next few slides, I'll discuss the importance of ensuring consistent labeling submissions, correct editorial and grammatical statements, proper formatting of the limitation statement and title, the importance of submitting consistent drug product name and manufacturing statements, adequately differentiating related products and/or product strengths, the recommended format for expiration dates, ensuring the appropriate use of labeling statements, confirming a sufficient number of medication guides are provided, compliance with standards for ferrule and cap overseal for injectable products, child-resistant packaging, CRP, verification statements, and lastly, submitting consistent CRP statements. So we have a lot to cover, so let's dive in. Have you seen anyone walking around with two different colored socks? Perhaps if your answer is yes, your mind had probably guessed that the person was most likely in a rush or made an honest mistake. In a similar vein, inconsistent labeling submissions is an error that can easily be avoided. When submitting the Microsoft Word and PDF text versions of your labeling, it's important to ensure that the information is the same and consistent in your submissions. Additionally, the submitted document should align with the latest RLD labeling found at Drugs at FDA. We strongly advise that you conduct a thorough review of your submitted labeling pieces for spelling, spacing, typographical, grammatical, and/or data errors. Editorial and grammatical errors in labeling may be misinterpreted by the end user and can lead to medication errors that ultimately result in patient harm. So this is something we do take seriously. For example, trailing zeros or missing zeros can lead to a dosing error. Examples of other errors are shown on the slide. For prescribing information labeling following the physician labeling rule format, in the highlights section of the labeling, ensure that the limitation statement and title are formatted correctly. As shown on the slide, we recommend that the name of the drug product be presented in uppercase letters to improve its prominence. One of the concepts we would like to drive in today is the importance of consistency. Throughout the PI, the references to the drug substance and drug product should be consistent. For example, if your drug product name is ABC tablets, do not simply reference the drug product in some places as ABC, while in other places as ABC tablets. Use the established name, including the dosage form, when referencing the drug product. So in our example, references to the drug product should be consistently presented as ABC tablets. When naming the place of business of a manufacturer, packager, or distributor, include one of the qualifying statements found in the CFR listed on the slide. And make sure this information is consistent across all labeling pieces where it's listed and/or required. In July of 2006, the Institute of Medicine published a report titled Preventing Medication Errors. The report cited labeling and packaging issues as the cause of 33% of medication errors and an astounding 30% of fatalities from medication errors. It emphasized that the product naming, labeling, and packaging should be designed for the end user, that is, the provider in the clinical environment and/or the consumer. One of the ways to create safer labels is to adequately distinguish and differentiate different strengths of the drug product and/or proposed product labels from your other related products. This can be achieved through boxing, contrasting colors, and/or some other means as demonstrated on the slide. Although there are various ways to write an expiration date, we have found that the use of 2-letter months and 2-digit year abbreviations for expiration dates has led to confusion, misinterpretation, and sometimes delay in treatment because the abbreviation was interpreted incorrectly. For example, the abbreviation MA could mean the month of March or May, whereas the number 12 could represent the day, month, or year. Therefore, we recommend using a 4-digit year. Also include a hyphen or / to separate the portions of the expiration date. The alphabetical character abbreviations shown on the slide are the preferred format for the months as well as the expression of the expiration date. When warning or cautionary statements are needed for the drug product, it is pertinent that the correct labeling statement is utilized. Non-affirmative warning or cautionary statements have been misinterpreted. Therefore, affirmative statements should be used. For example, for intravenous use only, fatal if given by any other route, or must dilute before use. These are examples of statements that are more easily understood. Let's go through the examples shown on the slide. For injectable single-dose products, include the discard statement on the container label and other labeling as appropriate and as space permits. For example, include the statement, discard unused portion. For injectable pharmacy bulk packages, add a prominent box declaration on the principal display panel reading, pharmacy bulk package, not for direct infusion. For products without gluten, the term gluten-free is not acceptable. Instead, we recommend the following statement: contains no ingredient made from a gluten-containing grain, wheat, barley, or rye. The warning statement for rubber, latex-free, is not acceptable. We recommend the statement, this specify the component you're referencing is not made with natural rubber latex. The CFR uses the term natural rubber to include natural rubber latex, dry natural rubber, and synthetic latex or synthetic rubber that contains natural rubber in its formulation. When any of these are included in the product, add the caution statement, caution, this product contains natural rubber latex which may cause allergic reactions. Lastly, there are special labeling statements and regulatory requirements needed for certain inactive ingredients. Specifically, tartrazine, aspartame, and sulfite are a few. Although this is not an exhaustive list, we wanted to point out some examples. Please reference the CFR listed on the slide for additional information. Thank you for your undivided attention thus far. I'd like to turn the floor back over to Commander Neshwat to wrap up the remaining content for this section.
Thank you, Lieutenant Commander Ochozo. I will continue with presenting on best practices for ANDA labeling submissions. Another topic we want to highlight is confirming a sufficient number of medication guides when applicable. We want to ensure that a sufficient number of medication guides are available for dispensing and distribution of your drug product per 21 CFR 208.24b. We request that applicants provide a statement that a sufficient number of medication guides are available for dispensing and distribution for your drug product in Module 1.14.1 and or the question-based review, or QBR. If this information is not provided in the submission, a comment will be issued asking the applicant to confirm this information. Another area we are going to discuss is complying with standards of ferrules and cap overseals for injectable products. We request that applicants provide a comment in Module 3.2.p.7, Container Closure System, as to whether text appears on your product's cap and ferrule overseal, and to also comment on the color of your product's cap. This is very important to include in your submission, as we want to ensure that your proposed cap and ferrule overseal are in compliance with the requirements of the USP General Chapter 7, labeling for ferrules and cap overseals. Per USP standards, a cautionary statement is intended to prevent an imminent, life-threatening situation and may include instructional statements that provide potency or other safety-related instructions if warranted. If no cautionary statement is necessary, the ferrule and cap overseal must remain blank. Again, we request commenting in Module 3.2.p.7 as to whether text appears on your cap and ferrule overseal, and also to comment on the color of your cap, as only potassium chloride for injection concentrate can have a black cap. Per USP standards, only cautionary statements may appear on the top or circle surface of the ferrule and cap overseal. If a cautionary statement is necessary for your drug product, we recommend providing a high-resolution image of your ferrule and cap overseal in Module 3.2.p.7, Container Closure System, and ensure there is adequate contrast between the proposed text and color of the ferrule and cap overseal, as we want to ensure the cautionary statement is easily read. Now we'll move on to child-resistant packaging, or CRP, verification statements. Ensure that your proposed packaging meets the requirements of the Poison Prevention Packaging Act, or PPPA. We encourage including a written verification statement in Module 3.2.p.7 that your product's packaging meets the U.S. Consumer Product Safety Commission's, or CPSC's, child-resistant packaging standards. An example of a written verification statement may be, we verify in this submission that the following package or packages meet U.S. Consumer Product Safety Commission's, CPSC's, standards under 16 CFR 1700. If you choose to include CRP statements in your labeling to indicate that your product packaging is child-resistant, please ensure they are consistent across all labeling pieces. If including CRP information in the PI, the patient labeling, it should appear in the How Supplied Storage and Handling section of the PI and the How Should I Store Drug X section of the patient labeling. Again, the CRP statements in the PI and patient labeling should be consistent. If choosing to include CRP statements on prescription drug container labels and carton labeling, display the CRP statement on the side panel in close proximity to the storage information. For a non-prescription or OTC product, display the CRP statement in the drug facts labeling under the subheading Other Information. We refer you to the child-resistant packaging statements and drug product labeling guidance for industry for additional details. Now we have our second challenge question. Which of the following statements is true? A. Cautionary statements may appear on the side surface of the ferrule and cap overseal. B. Consistent Microsoft Word document and PDF text version should be submitted for PI and patient labeling pieces. C. Gluten-free is an acceptable labeling statement. Or D. Different strengths of the proposed drug product should not be adequately differentiated, e.g., boxing, contrasting colors, and/or some other means. Please take a few seconds to think about which statement is true. For those that selected B, you are correct. Consistent Microsoft Word document and PDF text version should be submitted for the PI and patient labeling pieces. The other three statements are false. Now I will hand over the next part of the presentation over to Dr. Odeshina.
Thank you, Commander Neshawat. In this portion of the presentation, we will be discussing best practices for ANDA labeling supplements. We will discuss the importance of having a detailed cover letter, ensuring a complete submission, patents and exclusivities, side-by-side comparisons, electronic patient labeling, URLs, and lastly, FDA's safety labeling changes. Detailed cover letter. Clearly and accurately state the proposed changes in the cover letter. Failure to do so may lead to a CBE being denied to a PAS at a later date. For CBE 0 RLT updates, state the NDA number, supplement number, and the date of approval of the reference supplement in the format shown on the slide. Ensure complete submission. Note that DLR does not issue acknowledgement letters for CBE zeros. If we request for an amendment to a specific supplement, please do not submit a new supplement. Submit an amendment to the referenced supplement. For ANDAs with shared prescribing information, provide submissions for each ANDA. For example, if ANDA A and B share an insert, if a CBE 0 is submitted to ANDA A for labeling updates to be in accordance with the reference listed drug, the applicant should submit a CBE 0 for ANDA B as well. Patents and exclusivities. Ensure that the proposed labeling is consistent with patent certification and exclusivity statements. For labeling carve-outs to align with an agency-issued BPCA template, also address the new exclusivities listed in the Orange Book. New labeling carve-outs due to exclusivities should be submitted as a prior approval supplement, not as a CBE. Submissions to align with an agency-issued BPCA template should be submitted as a CBE. Side-by-side comparisons. Provide a side-by-side comparison of either the approved RLD labeling with the proposed ANDA labeling, or the previously approved ANDA labeling with the currently proposed ANDA labeling. For amendments, note and submit what was requested in the issued letter. When submitting a prior approval supplement, also provide a side-by-side comparison with the previously approved labeling, the RLD labeling, if applicable, and the proposed labeling. Electronic patient labeling, URLs. Do not replace the standard medication guide statement with a URL on container and carton labeling. A URL may be placed in addition to the standard statement, provided that it meets the requirements of 21 CFR 610.60. Add a statement to the top of the medication guide to alert dispensers that a medication guide will need to be printed and dispensed. The medication guide dispensing statement should be included on all labeling pieces to include the container and carton labeling at the bottom of the prescribing information and at the top of the medication guide. Note the URL link should be simple, non-promotional, and not false or misleading. Ensure consistency of the URL link in all proposed labeling pieces. Patient information leaflets can also be supplied electronically and submitted as a CBE 0. For DAS safety labeling changes, proposed labeling should include language identical to what is delineated in the SLC notification letter. Submit a side-by-side comparison of the previously approved labeling with the new SLC language. This concludes our presentation. In summary, we would like to reiterate that adherence to ANDA labeling best practices facilitates the labeling review process and enables the fulfillment of GDUFA III labeling commitments. This ultimately results in the approval of safe and effective generic drugs for the American public. We have several resources available to assist you, and we would like to thank you very much. We look forward to welcoming your questions in the question and answer session. Thank you.
Thank you, and I'm happy to be here today providing you an overview of major quality deficiencies and approaches available in GDUFA III. Learning objectives. First, I will go over some of the most commonly seen major deficiency themes for pharmaceutical quality. Second, I'll discuss how the agency will communicate those deficiencies to ANDA applicants for their applications. And finally, I'll talk about how ANDA applicants can ask questions of the agency on ANDA major deficiencies, including some new approaches that may be available as part of our GDUFA III commitment. Some disclaimers. Please note the major deficiency themes I'll be discussing are not an exhaustive list of the quality major themes. A more thorough, though again not exhaustive, list can be found in Appendix A of the ANDA Amendments Guidance for Industry. Additionally, not all of the new approaches in GDUFA III that will be discussed are available for all ANDA drug products. First, major quality deficiency themes. When we look at drug substance, one of the most commonly seen major deficiencies is for the referenced drug master file, or DMF, for the active pharmaceutical ingredient, or API. While the agency will tell ANDA applicants their reference DMF has major issues, the specific issues will only be disclosed to the DMF holder. For more complex products, such as peptides, adequate demonstration of API sameness to the reference listed drug is key. Additionally, when toxicological studies are needed to qualify an unqualified impurity, this requires additional expertise outside of the quality assessment team, so this is considered a major deficiency. As we move into drug product, you can see just like the need for toxicological studies to evaluate an unqualified impurity for drug substance is considered a major deficiency, this can also be an issue for drug product. Major deficiencies around extractables and leachables could be due to a need for a safety assessment, an inadequate assessment, or a new unsolicited submission that includes extractable and leachable assessment. All of these would be considered major deficiencies. On unacceptable analytical methods, this may mean the proposed procedure is not stability indicating or is not discriminating enough to address product quality. Next, the major deficiency themes for manufacturing can encompass either facility or process deficiencies. The one we see the most is the major deficiency due to an inadequate facility, whether it's because of a pre-approval or surveillance inspection. For process, inadequate control of critical process parameters, inadequate justification of the scale-up strategy, or the need for new batches due to stability failures are commonly seen major deficiencies. When it comes to the major deficiencies for microbiology, adequate validation data is key. For example, product release or stability testing methods such as antibacterial endotoxin testing, sterility testing, or container closure integrity testing are important. Terminally sterilized products must provide adequate sterilization validation data. Aseptically filled products must show their manufacturing process supports the sterilization of the equipment and components used to produce the drug product. And for multi-dose products, they must provide evidence the product remains sterile dose to dose through their antimicrobial effectiveness testing. Finally, biopharmaceutics major deficiencies may be due to the need for new in vitro dissolution analytical methods because the proposed method is found inadequate, the data to support the proposed in vitro release acceptance criteria is inadequate, or a new in vivo bioequivalence study is required to support the proposed post-approval change. Now for our first challenge question. FDA will tell an ANDA applicant why their referenced Type 2 DMF is considered major. Is this A, true, or B, false? Please take a second to consider which answer is correct. And the correct answer is false. As noted earlier, an ANDA applicant will be informed their referenced DMF is major, but not the nature of the deficiencies sent to the DMF holder. It is up to the ANDA applicant to discuss with the DMF holder as needed to evaluate the impact on the referencing ANDA. For the next two sections of this presentation, I will only be addressing how the agency works with unapproved ANDA projects and will not address post-approval submissions. I will start with how major deficiencies will be communicated to applicants by the agency. First, information requests. Generally, quality will not issue major information requests. There may be instances when we do, but it is unlikely to be a regular occurrence. Applicants may see major IRs from the clinical discipline for a combination product at the midpoint of the first review cycle, which could impact quality as well. If a major IR is issued from quality and there is no due date, that may indicate a forthcoming complete response letter. Next, discipline review letters, or DRLs. Major quality deficiencies will be sent in the quality discipline review letter, though this will exclude facility deficiencies. Facility comments, such as notification that a PAI was conducted and may not have an acceptable outcome, or a travel restriction may be communicated in a DRL, but those are not considered deficiencies. If ANDA applicants respond to their major DRL by the due date, the goal date will be extended to allow time for review of the DRL response. Note that due date includes any agreed upon extension that is confirmed via email, and the goal date extension will be for a major amendment to allow time to review the response. Finally, complete response letters, or CRLs. Major quality deficiencies will be sent in CRLs, and this will include facility deficiencies. Any remaining deficiencies from consults that were not sent in a DRL will be communicated in this letter. An example would be if an applicant saw pending consult language in their DRL, the deficiencies from the consult, as applicable, will be provided in the CR letter. Please note, even if an ANDA received a minor DRL, it is still possible a major CR letter is sent, depending on the assessment of the DRL response and any remaining deficiencies that were not sent in the DRL, such as those related to an inadequate facility. Now, for our next challenge question. Which letter type is when an applicant with an unapproved ANDA most likely will first learn of a major deficiency for microbiology? Will that be A, an information request letter, B, a discipline review letter, or C, a complete response letter? I'll give you a couple seconds to consider which answer is correct. The correct answer is B, discipline review letter. As I mentioned earlier, quality generally will not be sending major IRs, so the first time an ANDA applicant may learn of a major microbiology deficiency would be in the mid-cycle DRL in their first review cycle. Now, I will talk about how applicants can engage with the agency on questions they have around major quality deficiencies. For meetings, there are two main types of questions, clarifying and scientific, that may be asked by applicants. Clarifying questions may be asked in mid-cycle review meetings, or MCRMs, or in post-CRL teleconferences. Competitive generic therapy drug products and complex drug products are those potentially eligible for a mid-cycle review meeting. As you might have learned during yesterday's sessions, several new meeting types are now available to ANDA applicants in GDUFA III to address scientific questions. This includes an enhanced mid-cycle review meeting or a post-CRL scientific meeting, which are both only eligible for complex drug products in general. ANDA applicants will be notified of their eligibility to have a mid-cycle review meeting or an enhanced mid-cycle review meeting, either in the filing acknowledgement letter or in the CGT granted letter, whichever comes later. Additionally, FDA may grant the post-CRL scientific meeting if it is for a complex generic product or, in FDA's judgment, the request raises issues that are best addressed via this meeting process and cannot be adequately addressed through controlled correspondences. Speaking of controlled correspondences, you might have learned in yesterday's session about new avenues in GDUFA III for ANDA applicants to ask questions of the agency regarding their post-CRL or post-TA applications via controls. Post-CRL questions for major deficiencies can be submitted via a controlled correspondence, and that control may be considered level 1 or level 2, depending on the complexity of the major deficiency and the offices involved. Finally, our last challenge question. Which category of ANDA drug products are eligible for meetings to address scientific questions regarding major deficiencies? A, all drug products, B, CGT drug products, or C, complex drug products? Please take a second to consider which answer is correct. The correct answer is C, complex drug products. There may be rare cases where FDA will grant non-complex drug product a post-CRL scientific meeting when the agency determines the issues raised cannot be adequately addressed by a controlled correspondence. So, in summary, major deficiencies can span any of the quality subdisciplines, and as is also noted in Appendix A of the ANDA Amendments Guidance for Industry, what's been discussed in this presentation is not an exhaustive list of major deficiency themes, and ANDA applicants will see others that are outside of those discussed today. Quality major deficiencies will most likely be communicated either in a DRL or CRL. In some instances, quality will communicate major deficiencies in an information request, but that will not happen with much frequency. And finally, ANDA applicants now have new avenues to address their questions regarding major deficiencies in GDUFA III. I've provided the links to the ANDA Amendments Guidance for Industry, as well as some of the updated guidance for industry based upon our GDUFA III changes. Additionally, I would like to highlight some other resources, including the GDUFA III commitment letter and the updated map regarding issuance of IRs and DRLs for unapproved ANDAs that include additional information related to this presentation. I'd like to thank Heidi Lee and Craig Keister, my supervisor and division director, for their guidance as I've developed this presentation. And this concludes my presentation. I will address any questions you have in the upcoming Q&A panel. Thank you.
Hello everyone, welcome to my talk. As we receive more and more combination product applications, I would like to take the opportunity here to present some helpful tips on how to ensure that your submission will meet the drug product quality requirements, hopefully leading to your ANDA approval in the first review cycle. For my talk, the learning objectives are to define drug-like combination product, describe the framework for quality assessment, discuss product development study to demonstrate suitability for use, discuss quality control and stability program. By definition, a combination product is a product composed of two or more different types of medical products, drug, device, and biological product per 21 CFR plus 3. Also, it is subject to CGMP requirements for combination products with the final guidance published in 2017. Generally speaking, combination product can include a single entity, co-packaged, and cross-labeled. Currently, there are 3 pathways for pre-marketing review of combination products: device-led combination products, drug-led combination products, biologic-led combination products. For drug-led combination products, you can submit NDA or ANDA. The focus on my talk is ANDA. So how do we determine if it's a drug-led combination product? Well, the assignment is based on which constituent part provides the primary mode of action. PMOA is the single mode of action that provides the most important therapeutic action of the combination product. For drug-led combination product, drug is the most important therapeutic action. CDER is the lead center, main point of contact that will have primary jurisdiction for its pre-market review and regulation. Based on this criteria, you should be able to determine whether you can submit an ANDA or not. Let me give you some examples of drug-led combination products. They can range from injectable, oral syringe, and cup, like the picture on the right-hand side that can measure dose, ophthalmic eyedropper, nasal spray, metered dose inhaler to transdermal delivery system, and vaginal system. Today, I will discuss some key considerations for drug product quality when submitting an ANDA for prefilled syringe and injector. First and foremost, let's look at the drug product quality framework we have already in place for combination products per ICH guidance. We started out with M4Q published in 2001, talking about reproducibility of the dose delivery. ICH Q1A, Q6A on functionality testing, to the most recent ICH Q12, which has an appendix dedicated to the combination product for post-approval CMC changes. Also, it's essential to apply the principles of ICH Q8, Q9, and Q10 during the drug product life cycle to assure drug product quality. Equally important for drug product quality framework, FDA guidance on container closure system was published in 1999, talking about performance of assembled packaging system. Subsequently, more guidance was published to provide recommendations to industry on the CMC and quality requirements of combination products. So what happens when you submit an original ANDA to FDA? Basically, there are 2 main teams who will assess your submission. OPQ team will assess all quality aspects of an ANDA, whereas OGD team will assess bioequivalency, comparative threshold analysis for the user interface, and labeling. Note that for labeling, drug product review team will assess description and house supply in the package insert, so you will most likely see this type of labeling deficiency in the drug product quality DRL, or CRL. Since combination product can have a high-risk profile, like complex device constituent part auto-injector, and for emergency use, we will initiate CDRH consult through ICCR process, inter-center consult request. One for device engineering performance, evaluate essential performance requirement, drug delivery function, and so forth. The other consult for device quality system regulation. So you may receive deficiency from CDRH if the submission was found inadequate by CDRH. Let's go right into the section of your submission that are critical for device constituent part of the combination product. In P2, you should establish a quality target product profile, QTPP, to ensure desired drug product quality. The table on the right is an example of QTPP element for an auto-injector ender. In addition, we will provide rationale for the selection or design of your CCS, including the device constituent part, and you can add that to the table. As a drug product quality tip, you can add device functional property requirement to the table as well to make your table complete. In P2 during drug product development, you need to identify critical quality attributes, CQAs, that should be within an appropriate limit. Use prior knowledge and risk assessment to identify material attributes and process parameters that can affect CQAs. You can also modify the CQA as more data is available. Finally, CQAs can include more product-specific aspects based on the route of administration for your combination product. To qualify the CCS, you need to demonstrate suitability for its intended use, protection, compatibility, safety, and functionality. The last point is the key consideration for the combination product. For compatibility, a dosage form should not interact with the packaging components. Compatibility study can include glass delamination study per USP 1660 and in usability study per labeling. All contact materials should be considered. One compatibility example is that the API can react chemically with a compound leached out from the adhesive of a stake-in needle prefilled syringe, forming a new drug product impurity. This brings us to our next important topic. The packaging component should not leach undesirable amounts of substance. In my last example, the compound leached out from the glue of the prefilled syringe reacts with the API. Control of this impurity in the drug product spec may be needed. Therefore, we recommend you to conduct per USP 1663 and 1664. Assess per AET threshold. If your leachables are above AET threshold, identify and provide toxicological assessment. Common deficiencies are incorrect calculation of AET and omission of categorization and tox assessment of leachables above AET. Here is another tip. Based on our current FDA thinking, for injectable chronic use, you can use SCT of 1.5 microgram per day, and for non-chronic use, you can use 5 microgram per day. The last point that demonstrates suitability for use is performance and functionality. You should demonstrate the device can deliver the product in an accurate and reproducible way, such as dose, test conditions to simulate the use of the drug product closely for labeling. Here we have a prefilled syringe. For performance, prefilled syringe can be tested for delivered volume accuracy, break-loose force, glide force. For auto-injector with prefilled syringe or cartridge assembled into auto-injector, it can be tested for dose accuracy, cap removal force, activation force, extended needle length, and injection time. For a case study, a drug product is co-packaged with 14 syringes as 14-day kit with the labeled instruction to inject 0.2 ml daily for 14 days. To simulate the use of the drug product per labeling, you should provide accuracy study data of each dose from each of the 14 syringes for a total of 14 doses from one single vial. This data is essential to show your drug product can meet the labeled claim requirement. Moving along to P5 control of combo drug product. Control strategies should ensure consistency of drug product quality. You want to develop control strategy based on product risk profile, such as complexity of design and manufacturing. For example, product and process control can be simplified as product complexity decreases. The specification for performance should be set based on the dosage form, route administration, and design feature. Moreover, the test method should follow the appropriate standard. For example, for auto-injector, the test method should comply with the applicable ISO 11608 series, which are recognized by CDRH. I provide a link here. You can check to see which standards are recognized by FDA. In your drug product formulation spec, you want to include the quality attribute of the drug product per USP1 and test them at release and or stability as shown in this example here. In addition, we recommend you to include the device performance attribute of an auto-injector and test them as shown in the table here. As another tip, acceptance criteria along with justifications should be provided. Some common deficiencies are omission of description for integrity of device constituent part and device performance acceptance criteria in the drug product specs. That's all I have for P5. In P7, description of the container closure system should include enough information for quality assessment, such as material construction, coating treatment like Borotec and for plunger, lubricants such as silicone oil for barrel and needle, suitable QC specification and test procedure, technical drawing, certificates of analysis from both supplier and drug product manufacturer, USP compliance, and any other relevant technical information for the packaging components. This information is critical for qualification of container closure system. We are at the last section, P8. To support expiry, you should package, store, and test your stability samples per ICH Q1A and Q1E. Here's another tip. For the stability program of a pen injector, as an example, one full primary batch should be completely filled into cartridges, entirely assembled into pen injectors, and placed in cartons. The other two should provide sufficient fully assembled and packaged products for drug product quality and performance stability testing. For the last tip, you can use one position for post-approval stability testing if there's no worst-case position. Here's the end of my talk. To see how much you understand the material, we have two challenge questions. Challenge question number 1. Which of the following product is not a drug-led combination product? A. Drug in IV plastic containers. B. Drug in bottles with child-resistant closures. C. Drug in glass vial with empty syringes. D. Drug in aluminum tube with vaginal applicators. I will give you 15 seconds. The answer is B. Drug in bottles with child-resistant closure. Second challenge question. Which of the following statement is not true? Auto-injector must comply with CGMP requirements for combination products. CDRX assesses aging, stability data, and specification for essential performance requirement of auto-injector. Threshold of toxicological concern of 120 microgram per day can be used to calculate AET for assessing extractable and leachable of auto-injector due to treatment duration of less than 1 month. D. Stability data should demonstrate that the performance of auto-injector is maintained during shelf life. I will give you 15 seconds. The answer is C. C is not true statement. In summary, I have covered the drug product quality considerations for generic combination products in terms of suitability for use, QC control, and stability requirements. With advancement in complexity of combination products for better patient care, we will continue to develop more guidance to address a regulatory submission. Stay tuned. Here, I want to acknowledge the following people from OLDP. Thank you for their support and guidance. Thank you all for your attention.
Thanks so much for the great presentations. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like we have a few questions already coming in for Commander Neshua, and here is the first question. In the case of a discontinued RLD, if RLD labeling is available on drugs at FDA, is it recommended to show a comparison against RLD labeling and the original ANDA submission?
Thank you for that question. If the RLD labeling is available on drugs at FDA, even if it is discontinued, we recommend showing the labeling comparison against the RLD labeling in the original ANDA submission. Thank you.
Thank you for responding to that question. We do have another question from Commander Neshirwa, and here is the question. What differences in labeling can be accepted between the RLD versus the ANDA application?
Thank you for that question. Permissible differences can include differences approved under a suitability petition, other differences because the drug product and the reference listed drug are produced or distributed by different manufacturers. And so this would include due to omission of an indication or other aspect of labeling protected by patent or exclusivity. So those are some of the permissible differences. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We've got a few questions that came in for Kimi Odesina. And here's the first question. Can the ANDA applicant have additional information in labeling with justification?
Thank you for that excellent question. So generally speaking, ANDA labeling should be the same as the reference listed drug. However, for supplements, the regulatory pathway for providing additional information with justification would be a prior approval supplement. And during the review process, we will determine the acceptability of that additional information. Thank you.
Thank you for responding to that question. We do have another question that came in for Kimi Odesina, and here is the question. Is it a best practice to denote a space for lot number and expiration date in the draft container labels in the original ANDA?
Thank you once again for that question. And absolutely, yes, that would be considered a best practice to denote a space for lot number and expiration date in the draft container labels. Thank you.
Moving on to our next panelist, we've got a couple of questions that came in for Karen Ireland, and here is the first question. In regard to peptides, is the sameness in terms of structural conformation or mass or sequence?
Thank you for that question. All of those can be factors when it comes to peptide sameness. I would refer you to the draft guidance that was published in November 2022 for additional information. That guidance is sameness evaluations in an ANDA active ingredients. If you find that after you read that and work on developing your product, you still have questions for the agency, for your complex product, I would recommend you look at our final guidance published in October 22 for formal meetings between FDA and ANDA applicants of complex products under GDUFA to see if you qualify for one of those meeting types to answer your additional questions. Thank you.
Thank you for responding to that question. We have another question that came in from Karen Ireland, and here's the question. And they're regretting that it's not clear to the questioner, but what is the difference between a CRL and a DRL? Also, if there is no response time specified in the agency's notification letter, what are the general deadline expectations?
Thank you for the question. So a CRL, or complete response letter, it would contain all the deficiencies across multiple disciplines, so labeling, bioequivalence, or quality, and closes out your review cycle. A DRL, or discipline review letter, would be individual to a discipline. For example, you might get a quality DRL or a bioequivalence DRL. If there is no response time specified in the letter, for example, a DRL with no due date, that might indicate that you are getting an upcoming CRL. And we would ask that you either wait for the CRL and respond once you are able to respond to the full complete response letter, or you respond to the DRL as soon as you're able to fully respond to all deficiencies included. The general response time expected for a CRL is within 1 year of issuance of that CRL. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We have a few questions that came in for Dr. Kai Kwok, and here's the first question for Kai Kwok. Where can sponsors find FDA guidance on EPRs?
Hello, everyone. For EPR, currently, we don't have a guidance out there for you to take a look. It's under development. In the future, we may have guidance or may not have guidance coming out for EPR. Thank you.
Thank you for responding to that question. We have another question for Kai Kwok. And here's the question. Can the other SCT be used apart from what was mentioned in your presentation by following ICH M7?
Thank you for the question. Currently, we do not accept the TTC from ICH M7 based on the duration of treatment to calculate your AET. So we have to use SCT, as I mentioned earlier, to calculate your AET for leachable and extractable assessment. Thank you.
Thank you for responding to those questions. Moving up to the beginning of our panel, we have a couple more questions that came in for Commander Neshua, and here is the first question. When should we include drug product established name, including the dosage form and the labeling? And is it mandatory to add the USP designation to the drug substance and drug product established name?
Thank you for that question. So when we mentioned the drug product and established name, this would include the dosage form. So if the RLD labeling is referring to the drug product, the established name with the dosage form should be used in the ANDA labeling. Whereas if the RLD labeling is referring to the drug substance without the dosage form would be used in the ANDA labeling. And with regards to the USP designation or descriptor in labeling, use of the USP designation is not mandatory. When the USP descriptor is used, we recommend limiting the use of the USP descriptor to the dosage forms and strengths, the description, and the how supplied sections of the prescribing information. Thank you.
Thank you for responding to that question. We do have another question for Commander Neshua, and here's the question. What if the cap overseal needs to be texted with a cautionary statement, same as the RLD, but in case if it's kept blank, will it be considered a change in labeling?
Thank you for that question. If the RLD has a cautionary statement on the cap overseal and the ANDA's cap overseal is kept blank, this would lead to a labeling deficiency, as the ANDA should be the same as the RLD and have that cautionary statement on the cap overseal. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few more questions that came in for Kimmy Odesina, and here is the first question. What type of supplement should be submitted for addition of a QR code and electronic medication guide in URL statements?
Thank you. That is a great question. So these types of additions should be submitted as a CBE 0. Thank you.
Thank you for responding to that question. We do have another question for Kimi Odesina, and here is the question. What is the process for incorporating a new proprietary name in labeling?
Thank you again. That is an excellent question. So a new proposed proprietary name should be submitted as a prior approval supplement. Please note that these new names need to be submitted and approved by the Division of Medication Error Prevention and Analysis, also known as DMEPA, before it can be approved for use in labeling. Thank you.
Moving on to our next panelist, we've got a couple more questions that came in for Karen Ireland. And here is the first question. Can a product directly get a CRL without any IR, DRL during review cycle?
Thank you for the question. It depends on which review cycle we are in. For the first review cycle of an ANDA, generally an applicant will receive at least one DRL during around the midpoint of the review cycle. However, once we are in subsequent review cycles after, for example, a CR, those reviews may happen without any IR or DRL being sent out. Thank you.
Thank you for responding to that question. We do have one more question for Karen Ireland, and here's the question. In what instances might quality send a major IR?
Thank you for that question. If the assessment team determines early on in a major amendment or an original and a review cycle that there is information missing that they would be considered major, we may send a major IR at that time. Additionally, we do look at the prioritization for that drug product, such as if it's for a declared public health emergency, to try and get that major IR sent to get to approval as promptly as possible. Thank you.
Thank you for responding to that question. We're moving on to our next panelist. We've got about 13 minutes left. We do have a couple questions just came in for Kai Kwok, and here is the first question. Is the food grade certification mandated for all drug device combination products?
That's a good question. Basically, for combination product, for your non-combination product, your requirement to qualify your CCS is almost the same. So what we're looking for, if it's the syringe on your barrel or stopper, will be combined with USP. So that is come before the food grade certification. We probably would not need those because it's combined with USP. But for other stuff, maybe non-USP, we don't have USP chapter to assure quality, then we may look for food grade certification. I hope that answers your question. Thank you.
Thank you for responding to that question. We've got a couple more questions for Kai Kwok, and here is the next question. Can both drug formulation specification and device performance specification be on the same specification sheet in P5?
Another good question. Yes, you can have it separate or be put in the same specification for your drug product. Thank you.
Thank you for responding to that question. We have one more question for Kai Kwok, and here's the question. Can you please confirm that the SCT given on slide 15 is applicable for a parenteral drug?
Yes. So yes, the slide 15, the chronic duration of use, you use SCT of 1.5 microgram per day, and less than chronic use will be 5 microgram per day. You can use that for parenteral drug product. Thank you.
Thank you for responding to that question. Moving up to the beginning of our panel, we do have a few more questions that came in for Commander Neshua, and here's the next question. When should USP be used with the drug product name in the prescribing information?
Thank you for that question. If your drug product is subject to a USP monograph, we recommend only using USP with the drug product name in the dosage forms and strengths, description, and how supplied sections of the prescribing information. Thank you.
Thank you for responding to that question. We have another question for Commander Nishikawa. And here's the question. If we already certified to a patent with use codes and a new use code is listed for that patent, do we need to submit anything for the new use code listed?
Thanks for that question. So yes, you would need to address all new information, and this would include any new use codes listed in the Orange Book for the reference listed drug. Thank you.
Thank you for responding to that question. We do have another question that just came in for Karen Ireland, and here is the question. If I received and responded to a minor DRL, why did I just receive a major CRL?
Thank you for the question. So applicants might receive a major CRL after they've gotten a minor DRL for several reasons. It could be that the information they submitted in their DRL resulted in new major deficiencies. There is also the possibility that there was a pending consult at the time of the minor DRL, and the consult was finalized with major deficiencies, which would result in a major CR. Finally, the CRL could include major facility deficiencies that we do not include in our quality DRL, and any of those instances could result in a major CR after the applicant received a minor DRL. Thank you.
Thank you for responding to that question. We do have another question that just came in from Caroline Ireland, and here is the question. Is it mandatory to issue a DRL during mid-cycle review?
Thank you for the question. The agency, per the GDUFA III commitment letter, has noted that we will send for each discipline either an IR or DRL at or around the mid-cycle of the first review cycle. So generally, yes, we will be sending those around the mid-cycle for the first review cycle. Thank you.
Thank you for responding to that question. Moving on to our next panelist. Got a few more questions that came in for Kai Kwok. And here is the first question. For a combination product kit containing a lyophilized powder vial, a diluent vial, syringes, and needles, how many batches of diluent are required for stability testing?
Good question. Basically, if you manufacture both a lyophilized powder vial and a diluent vial, you will need to provide stability data from at least 3 exhibit batches, including both the lyophilized powder and diluent vials per ICXQ1A. Even the diluent is a sterile water for injection. And also, a drug product supplied with a reconstitution diluent should include a separate section, 3.2.P, with the diluent information. Thank you.
Thank you for responding to that question. We have another question for Kai Kwok. And here's the question. What are some of the pump performance functionality data that should be provided in a submission for a multiple dose metered dose pump dispenser for topical use?
Thank you for the question. It will depend on the design of your metered dose pump dispenser and labeling. And some of the performance tests such as number of pump to prime, dose uniformity, mean dose weight, meaning the dispensing amount per actuation, total number of doses, actuation force should be considered to demonstrate suitability of use for your proposed combination product CCS. Thank you.
Thank you for responding to that question. We're checking our queue, and it looks like we've worked through all the questions at this time that are relevant to the presentations that were provided. We're going to give a huge thank you to our presenters and panelists for the great presentations and for answering numerous questions that came in. We'll now enter into our lunch break until 1 p.m. Eastern Time. Please enjoy your lunch. Welcome back from the lunch break. We hope you're able to enjoy a refreshing lunch. We do have a few housekeeping reminders regarding CE information to share while you're finishing your lunch. As a reminder, in addition to the speaker evaluation, we have our final evaluation, which will be available later this afternoon from our one-stop shop link. The final survey is a mechanism by which you will receive the certificate of attendance, which will support CE credits for RAPS, SOCRA, SQA, and ACRP. Please remember to download the certificate immediately upon completing the survey. Otherwise, you'll miss the opportunity to do so. Even if you do not want or need the certificate, please take a few moments to complete the conference evaluation, as your feedback will help guide the design of our future events. Note that the evaluation and certificate are only available for 2 weeks, and that takes us to April the 27th. To summarize, for our physicians, pharmacists, and nurses claiming the continuing education credits, CEs are only available if you registered via Adobe Connect and attend in real time. SBIA will issue the CE claim code and instructions to all attendees later today. So be sure to complete the CE session evaluation on or before April 27th. I apologize in advance, but we are unable to make exceptions to that deadline. Attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to their attendance to claim credit. Today's email will have both the claim code and instructions together with the survey link. As a reminder, all sessions are being recorded. It'll take us about 5 to 7 business days to process the recordings, but you'll soon be able to access them from SBIA's webpage. And if you don't already have that bookmarked, here's an opportunity. It's at fda.gov/CEDAR-SBIA-Learn. It's our pleasure to move into our next session of presentations. First, presenting on a change in API supplier, drug substance quality tips. Our presenter, Dr. Kiduo Kion, is a senior reviewer in the Division of Lifecycle API, Office of New Drug Products. Kiduo has experience reviewing drug substances in support of various types of FDA applications. She is a relied upon leader in the review of complex APIs, particularly oligosaccharides and iron-containing macromolecular complexes. Our next presentation is on a change in API supplier drug product quality tips. The first presenter for this presentation is Dr. Rajiv Paul, who is a Senior Pharmaceutical Quality Assessor for the Division of Post-Marketing Activities 2, which is responsible for post-marketing activities for generic drugs, including the assessment of ANDA supplements in CEDAR. Dr. Paul has been at FDA since 2014, starting as a CMC assessor for ANDAs and ANDA supplements. He also serves on multiple working groups to facilitate the drug product review process. Our final presenter in this presentation is Dr. Bo Jiang, who joined the FDA in 2013 as a chemistry reviewer in OGD, reviewing CMC sections of ANDA applications for various dosage forms. In 2015, Bo transferred to OPF within OPQ as a process reviewer to focus on assessment of manufacturing process and facilities. In 2016, Bo started as acting QAL in Branch 1 of DPMA 1 and has worked as a senior pharmaceutical quality assessor since November 2021. Bo's been involved in multiple work groups, which include and are related to data integrity of OPMA data integrity, OPQ facility withdrawal working group, OPQ AIP MAP working group, and OPQ DI MAP working group. Next, we present on managing quality post-approval. Our first presenter in this presentation is Dr. Paul Swartz. Dr. Swartz is the Director of the Division of Pharmaceutical Scientists in the Office of Pharmaceutical Quality, responsible for evaluating manufacturing changes for approved generic drugs. In this capacity, he also works closely with the drug shortage staff at CDER in helping alleviate or prevent drug shortages for critical medical necessary drugs. Our final presenter in this presentation is Dr. Gabinga Okabadeyo, who is currently the Division Director for Post-Marketing Activities in the Office of Program and Regulatory Operations, Office of Pharmaceutical Quality. He joined FDA in 2013 as a Regulatory Business Process Manager in OPRO and subsequently held the positions of Acting Quality Assessment Lead and Branch Chief. Our final presentation before the Q&A panel is on inspection tips, best practices for remote interactive evaluations, and other alternative inspection approaches. Our first presenter in this presentation is Dr. Lane Christensen, who's currently a branch chief in CDER's OPQ, Office of Pharmaceutical Manufacturing Assessment. He joined the FDA in 2009 and has also held positions with CDRH, Office of Compliance, CDER OGD, and the OC, Office of Global Policy and Strategy, completing international assignments and positions with FDA's India and China offices. Our final presenter for this presentation is Michael Chasey, who's the Director of the Division of Pharmaceutical Quality Programs within ORA. He provides leadership and oversight for the coordination of domestic and international inspectional activities related to pharmaceutical products, including a mutual reliance agreement activities. Prior to this role, he served as a branch chief overseeing the Foreign Inspection Program and the Foreign Dedicated Drug Cadre. He has played an instrumental role in the development of various alternative tool policies used to supplement on-site inspections during the public health emergency. Please join me in welcoming our first presenter, Dr. Kiduo Kian.
Thanks for the introduction. Hello everyone. Today I'd like to share with you on the keys surrounding the change in API supplier, focusing on the drug substance risk assessment perspectives. First, I will briefly talk about the pre-approval changes to the drug substances. In December 2000, FDA has published guidance for industry entitled Alternate Source of the Active Pharmaceutical Ingredient in Pending ANDAs. This guidance focuses on the use of alternate sources of the API in unapproved ANDAs. To substitute an alternative source of the API, the following circumstances should exist. First, the original API source is not being withdrawn due to deficiencies specifically relating to that API, such as the lack of adequate controls, evidence of alteration, or evidence of falsification of data. If any of these situations apply, then a new acceptable bioequivalence batches may need to be submitted to support the alternate source of the API. Second, the previous bioequivalence batches and bioequivalence studies should be acceptable, and also the specifications of the alternate source API and the test data should be essentially the same as the original source API, so that the quality can be ensured. The applicants should submit an amendment to their pending application requesting the withdrawal of the primary API supplier, with the supporting information to add the new supplier, just as expected for any supplier of an API. Most commonly, the ANDA applicants and DMF holders found a necessity to report the changes towards the drug substance in the application's post-approval stage. In April of 2004, FDA has published the final guidance for industry entitled Changes to an Approved NDA or ANDA. It covers recommended reporting categories for post-approval changes for drugs. Recently, in September of 2018, fulfilling the commitment of GDUFA II, FDA has published the draft guidance for industry entitled Post-Approval Changes to Drug Substances. It focuses on changes to drug substance manufacturing during the drug product application's post-approval period. Specifically, it addresses how the risk of one or more changes to the drug substance should be assessed and outlines recommended documentations to support the change. Please note that FDA's guidance or draft guidance do not establish legally enforceable responsibility. It only describes the agency's current thinking on a topic and should be viewed as recommendation. The draft guidance applies to synthetic drug substances and the synthetic steps involved in preparing semi-synthetic drug substances. It covers the following changes. First, the facility scale and equipment changes associated with all steps of the drug substance manufacturing. It discusses the specification changes to starting materials, raw materials, intermediate, and unfinished and final drug substances. It discusses the synthetic manufacturing process changes and changes to the source of the drug substance, also changes to the container closure system for the drug substance. Facility changes generally do not include modifications to the synthetic pathway. The applicant or DMF holder is responsible for ensuring that any new manufacturing facility is operating in accordance with cGMP regulations. Here I would like to clarify that a change from one drug substance manufacturer to another is considered a change in the source of the drug substance instead of a change of facility. In this regard, it is just likely a new API supplier with a new DMF. Please ensure that the LOA to reference the new DMF is on file. The involved facility in this new supplier are included in your 356H, and the new DMF will undergo regular risk assessment if it's not already found adequate. When your current existing referenced master file submits a change of facility, especially adding a new manufacturing site, we sometimes encounter a situation called hidden facilities, which are those recommended facilities that are in the DMF but do not appear on the NDA or ANDA's 356H form. The facilities that are recommended to be included are listed here. The final drug substance manufacturing, micronization, sterilization, and testing sites, sites for intermediates addressed by ICHQ7, facilities used for storing or warehousing drug substance prior to a disposition decision. The critical intermediate site is the most common hidden facility that is not included in the applicant's 356H. Determination of critical intermediate is made on a case-by-case basis, using principles as outlined in ICHQ 11. List all recommended DMF facilities in the application will greatly facilitate the agency's assessment, so please communicate all required facilities and their responsibilities early to your applicants. Whether or not the DMF facility is listed in the application will not change the determination whether it needs a comprehensive evaluation. If a hidden facility is identified, the following comment will be sent to the ANDA applicant, and there may be an extension of the performance goal day. So please make sure you include all the recommended facility in your 356H form. Specification change usually happens when a USP monograph becomes available or is updated for a drug substance. There are also other situations that a specification may have to be modified due to the impurity profile non-equivalency, usually because of a manufacturing process change. While the drug substance specification should be updated to comply with the compendial standards, if impurities that are listed in the compendium cannot be formed in your process, then they do not need to be included in the specification. However, you should provide a footnote to the specification and COA of the final drug substance to state that the impurity is just not possible based on your ROS. Please remember to provide appropriate method verification or validation reports as applicable as listed here. Also, if an in-house method is used for assay or related substance, when a compendium method is available, please remember to provide method equivalency report to demonstrate the in-house method is comparable or better than the compendium method. We recommend using a crude API sample or a sample spiked with the specified impurities at the specification limits for the method equivalency purposes. Please avoid using a very clean drug substance that show every impurity to be not detected. Manufacturing process changes, especially changes in route of synthesis, are considered to have a moderate to high potential to adversely affect the impurity profile of the drug substance. The manufacturing process should be validated using the new route of synthesis. Impurity carryover studies and spec purge studies should be conducted as appropriate, and control of mutagenic impurities in or expected to be in the final drug substance should be evaluated according to SHM7. Any modification to drug substance manufacturing carries some risk of causing an adverse impact on quality. Each drug substance manufacturer should assess the particular proposed modification to their drug substance to determine the risk associated with the change. Certain modifications are viewed as less likely to result in an adverse impact than others, and late-stage changes usually has a greater adverse impact on the quality of the drug substance and consequently on the drug product. A central principle underlying the risk assessment is that a change in the drug substance manufacturing process can be adequately assessed by comparing 3 consecutive pilot or commercial scale batches of pre- and post-modification material to determine if the quality of the post-modification material is equivalent to the pre-change material. Evaluation may include, but is not limited to, a comparison of impurities in pre- and post-modification intermediates, unfinished drug substance, or the final drug substance. A comparison of drug substance physical properties before after modification, and drug substance stability data. If the impurity profile of an isolated material following the change is equivalent to that of pre-change material, the drug substance impurity profile is considered unaffected by the modification. It is important to determine the stage in the manufacturing process at which impurities of the isolated material should be evaluated. After the proper intermediate or drug substance is identified for comparison purposes, the level of existing and new organic impurities, residual solvents, and elemental impurities should be compared per the related SH guidelines. It is also very important to determine the potential for the formation of mutagenic and unusually toxic impurities, including nitrosamines per the SHM7 and the FDA guidance on control of nitrosamine impurities in human drugs. The adequacy of the analytical procedures should be established to support their usage. If the manufacturing modification occurs at an upstream step before the final intermediate is produced, and the equivalence cannot be demonstrated for the intermediate isolated immediately following the change, then the impurity search should be extended to the next downstream intermediate. Please note that the impurity search should also be expanded to include appropriate downstream impurities that may be formed based on your process's chemistry. The evaluation and comparison should be repeated downstream further if necessary. In case that you are not aware of, the reauthorization of the GDUFA III program features a number of enhancements, including a mechanism to enable assessment of DMFs in advance of certain ANDA and PAI submissions. Pursuant to commitments under GDUFA III, FDA issued a draft guidance for industry entitled Review of Drug Master Files in advance of certain ANDA submissions under GDUFA. It describes instances when an early assessment, or so-called DMF prior assessment, could be requested by a DMF holder, and the circumstances under which FDA would commence an early assessment of Type 2 API DMFs, 6 months prior to an ANDA or PAI submission referencing the DMF. Such circumstances mainly involve products that could help mitigate or resolve a drug shortage issue, or product that could help address a public health emergency. The draft guidance provides recommendations for such DMF holders when making a request. Please refer to the guidance for details for eligibility. The DMF prior oral assessment is not available for the review of drug substance information submitted in an ANDA. To assist the DMF holder in submitting such request, a list of elements FDA recommends to be included in the cover letter of the prior oral assessment request is provided in the guidance and is attached here. Here are some take-home messages from the talk that I will not read through, and links to guidances I have covered. Thank you for attending, and we welcome any questions.
Good afternoon. In this session, we will be talking about change in API supplier and associated drug product quality tips. We will discuss quality considerations for applications when changing API supplier, focusing the impact on drug product quality from drug product quality assessment perspective and facility assessment perspective. Learning objectives are: understand the common reasons for changing API source, identify required CMC information from the ANDA applicant, determine ANDA supplement reporting category for new API source, understand what facility information for API supplier is required in drug applications, understand the impact of data integrity violations on drug applications. In this session, I am going to present the drug product quality assessment perspective. My talk will include the common reasons for alternate API source in pending ANDAs and ANDA supplements. What is the supporting CMC information the applicant should provide to change the API source? What will be the supplement reporting category for changing API supplier? And my colleague, Dr. Bo Jiang, will discuss on facility assessment perspective. His talk will include facility review for API supplier change and data integrity assessment and impact on applications. Drug product quality assessment perspective. What are the common reasons for changing API source? ANDA was approvable. However, an unsatisfactory CGMP inspection status for the primary API supplier and new API is proposed. API supplier remain unacceptable or inadequate, and the application cannot be approved. Increase the capacity of the drug product manufacturing, and therefore additional API supplier is proposed. Withdraw the original API supplier and add a new supplier. Now this change in API source is applicable for both pending ANDAs and post-approval changes, meaning ANDA supplement. To change the API source, the applicant should provide following CMC information. The applicant should compare physicochemical properties and impurities of the API and provide the justification. Drug substance manufacturer certificate of analysis and drug product manufacturer certificates of the analysis for the drug substance from alternate API source. At least one batch of pilot or commercial scale drug product should be manufactured. Minimum of 3 months long-term and 3 months accelerated drug product stability data should be provided in the submission. In addition, in vitro testing of the drug product for new API source should be performed. Comparative dissolution data, depending on the dosage form of the drug product, should be provided. When the drug substance is highly soluble, comparative multi-point dissolution study using application or compendial medium and approved testing conditions should be conducted. When the drug substance solubility is low, comparative multi-point dissolution study using multi-pH media with approved testing conditions should be conducted. when the comparative dissolution profile for the drug product is similar, the new source of the API is supported. What will be the supplement reporting category for changing API supplier? Changing the drug substance supplier to another manufacturer involves more than simply a site change. There will be additional differences like new or modified route of synthesis, manufacturing process, solvents, or equipment in most cases. Because of the substantial risk involved, alternate drug substance source should be reported in a prior approval supplement. However, when new API source is approved as a PAS in another ANDA from the same applicant, the applicant can report the same alternate API source for their other drug products as a CV30. Let me discuss a case study where a supplement was submitted for an alternate or new API source. The supplement was submitted as CV30 reporting category, and the decision was the supplement denied to PAS by the agency. Alternate API source should be a PAS. As per the guidance for industry, changes to an approved NDA or ANDA questions and answers that was published in January 2001, Manufacturing Sites section, question 1, answer 1, and supported by the draft guidance for industry, post-approval changes to drug substances that was published in September 2018, section 11. This new source of the drug substance is considered to have a high potential to have an adverse effect on the drug substance's impurity profile and physical properties that may affect the quality of the drug product. Therefore, the CV30 supplement was denied to PAS. As I mentioned earlier in a slide about the exceptions for CV30 category, once the proposed alternate API source supplement is approved as a PAS for the same applicant, and if there are no other changes to be reported that would otherwise belong in a PAS type reporting category, the applicant can report the same change, that is the same alternate API source, for their other drug products as a CV30. However, the API manufacturing site or the testing sites should have a satisfactory CGMP inspection status and then can be considered as CV30. This will end my talk. The topics I covered today were taken from the following resources. These are available guidance in the agency websites. Please look into these guidance for additional information. Now, I will have a challenge question. The applicant should provide certificates of analysis for the alternate drug substance from the drug substance manufacturer, drug product manufacturer, both drug substance and drug product manufacturers. I will wait for a few seconds for the answer. And the answer is C. Both drug substance and drug product manufacturers. The applicant should provide certificates of analysis for the alternate drug substance from both drug substance and drug product manufacturers. Now, the facility assessment perspective will be discussed by my colleague, Dr. Bo Jiang. Thank you.
Thanks, Rajib. I will be providing some tips from facility assessment perspective and covering a simplified overview on data integrity assessment and its impact on applications. When a new API supplier is proposed, it normally involves multiple facilities, and we look for clear roles and responsibilities for each facility, whether it is for manufacturing, testing, or simply storage. or it is for development batches, exhibit batches, or commercial operations. And the roles and responsibilities need to be consistent between DMF 356H and Module 3 in the application. We also look for a complete list of all facilities, including all intermediate sites, from the DMF. For any reason, if some facilities will be excluded from commercial operations, a revised LOA later needs to be included in the application. OPMA takes a risk-based approach in assessing facilities based on the roles and responsibilities proposed for each facility. We look at the facility experience and any inspection history that are available to us. We may request a pre-approval inspection if necessary to further assess the facility on site. Keep in mind that facility CGMP compliance status may change any time before the application go date due to CGMP violations, which may or may not be related to data integrity issues. When the facility compliance status changes, it will prevent application approval. Additional API suppliers can be proposed in the applications, either before the application approval or post-approval. Most of the time, the original API supplier will be withdrawn due to CGMP compliance issues, and when that happens, we normally look for if there are any data integrity concerns at the original supplier sites. As any data integrity concerns, once confirmed to be data integrity violations, may impact the data reliability in the submission, and therefore impact the application approval. Data integrity issue is a very important issue because FDA must have confidence in the data in an application before it can be approved. Any breach in data integrity erodes or breaks the trust between the FDA and the industry. The data integrity violations are most discovered during inspections, normally as the tip of an iceberg, and then become larger and larger. Once it is confirmed, all the data in submission becomes questionable. Assessment of data integrity is a very lengthy process involving multiple FDA offices, including ORA, OMQ in the Office of Compliance, OPMA in OPQ, and other offices. We look at what types of GMP violations are, whether it is testing to compliance, or data deletion falsification, or inadequate OOS investigation, or inadequate data control, etc. We try to understand the scope of the data violations, such as any pattern or practice at the site, and how long the practice existed, and which GMP system may be impacted, and how many facilities may be involved, etc. Based on all information available, we assess the impact on pending and approval applications. The OPMA DI assessment is a collaborative effort with ORA and OMQ. While OMQ is performing its own assessment from compliance perspective, OPMA looked at data validity based on third-party forensic analysis and any data integrity audit report. OPMA makes materiality assessment on application data, focused on bio batch, exhibit batch, and registration batch data. The assessment outcome is application specific and could be no DI concern if not sufficient evidence available. It could be inconclusive, in which case we may send out a request for additional information or follow-up inspection. The outcome could be that the data are confirmed unreliable, in which case a complete response letter will be sent to the applicant. To remediate the data integrity issue in the application, we may request third-party independent test results. We may request to manufacture a new batch and conduct a new B study. As you can imagine, all those activities will delay the application approval. The drug substance facility will be put on OAI status, which prevents approval of all applications that using the subject API supplier. This case study is based on the real drug application with the name removed and the dates listed here are not real. The drug substance facility was inspected in September 2000. The 43 citations included data integrity violations, but not significant enough to support the OAI classification. 2 years later, another inspection was conducted, and this time more evidence became available, and the final classification was OAI. A warning letter was issued later on to the firm, and regulatory meeting and teleconference were conducted, and expectations were provided to the firm. Parallel to OMQ assessment, OPQ has identified the time period since September 2000, after which all data generated at the facility are impacted by the DI violations. For this application, the drug substance lot was manufactured in February 2003. Therefore, the drug substance data are considered unreliable and impacting the BE start outcome. A complete response letter was sent and requests the firm to choose one of the 3 options below. Option 1, to confirm the quality of the original drug substance lot and provide additional data that are listed here. Option 2, to manufacture a new drug product batch with a new drug substance lot from the original source and conduct a BE study on one condition that is specified here. Option 3, manufacturing a new drug product batch with a new drug substance source and conduct a new BE study. The firm responded to the CR letter, withdrawing the original drug substance site. However, no data integrity audit report was provided. The proposal is not acceptable, and an information request letter was sent to the firm to request the data listed in option 1 above. The firm responded to the IR letter, committing to options 3 above, and basically to repeat the BE study and replace the BE data in the application. The application was CR'd due to inadequate BE review. That concludes my talk. I included some additional resources here. The first guidance is about identifying facilities in applications, and the rest of the guidances are about data integrity and remediation, which may be helpful. I would like to thank Pei, Rapti, Adit, and Derek for their guidance and providing comments on my slides. And I also like to thank OPMA DI working group members for the very interesting discussion on this topic. Challenge question number 1. What facility information for API supplier is required in drug applications? A. Clear and consistent roles and responsibilities. B. Complete list of manufacturing, testing, and storage facilities for all intermediates and final drug substance. C. LOA letter should specify facilities for commercial operations if a subset of facilities are proposed. D. All of the above. The answer is D. Challenge question number 2. When data integrity issues are confirmed at a drug substance site, which of the following could happen? A. Request independent test data and additional information to assure quality of the drug substance data. B. Manufacture new batches using a new API supplier and conduct a new BE study. C. Facility OAI status prevents approval of all applications using the subject API supplier. D. All of the above. The answer is D. Thank you very much for your time, and I would like to take any questions you may have.
Good afternoon. In this session, we'll be talking about post-approval changes, how to correctly manage them, and we'll also be talking about updates to post-approval changes that were implemented in October of last year in GDUFA III. So our learning objectives for this session are to understand the reporting categories for post-approval submissions, how to avoid common deficiencies when you submit these changes, and we'll talk about the GDUFA III updates. So what are some of the common post-approval quality changes? What would a firm be changing after approval? It might be changing some issues with the drug substance, such as a site change or a source, or how the drug substance is synthesized and what equipment is used, for example. Also, there might be changes in the drug product, which would be new formulations, there might be new strengths, you may change excipients, of course the manufacturing site might change, and the process and the equipment, etc. may all change. There may be changes in the container closure system. You may be going from bottles to blisters and things like that. You want to add new types of containers. And one more type of change would be where the drug is actually tested and packaged. There might be changes in those sites. So when the supplement is submitted for these post-approval changes, sometimes a complete response letter is issued. And what would be the causes for those complete responses? Sometimes it's a change in the API source, but that source has not been accepted. The drug master file, which is referenced, is not in an acceptable state. Sometimes firms want to widen their impurity specifications or even the dissolution specifications, and these have to be appropriately justified. And when they're not, or not done, these changes aren't qualified, a CR letter would issue. If it's a change in the drug product site, and the site is not under good compliance right now, that would give a deficiency. And changes in the container closure do lead to deficiencies if they're not shown to be as protective as previously. So what are the type of reporting categories for changes? There are major, moderate, and minor. A major change would be considered a prior approval supplement, meaning that it needs approval from the FDA before that change to be effected. Moderate change would be, there are two types, what we call changes being affected 0 or changes being affected in 30 days. And a change being affected 0 means the change can be implemented immediately after the supplement is received at the FDA, and a change is being effective 30 means the change can only be effected 30 days after receipt. A minor change is a change that can be reported in the annual report, and in the annual report those changes are reported as what happened that year. And here are some examples that could be reported in the annual report. Reducing an overage that's in the original formulation. If you want to extend the expiration date based on the protocol that was approved in the application. If you're making a change to comply with a compendial, basically a change in the USP. If you want to go along with that change, that's a kind of reportable. A change in the supply of an excipient, if it's the same excipient. A change in how the drug is manufactured by changing the order of addition in solution products. Or certainly tightening of an acceptance criterion can be annual reportable. On the other extreme, these are examples of changes that require prior approval. So these would be adding a new API supplier, if you're changing the whole route of synthesis of the drug substance, if you're widening specifications, if you're changing the equipment and the manufacturing equipment for the different equipment that uses different operating principles. Adding a new flavor or color is a prior approval change. Adding a new strength is a prior approval change. And changing from a glass ampule to a glass vial with a closure is a prior approval change. So how do we, when we get these changes in the FDA, how do we process them? The supplement is received, it is then assigned to one of the reviewers, and the reviewer has to decide, well, the firm submitted that as a CB30, does it really belong in this CB30? If yes, it's reviewed, and then an action is taken. An information request might be requested before the final action letter. If, however, the reviewer decides that the change does not fit under changes being affected, the change is then elevated or denied to a prior approval supplement, and the applicant must resubmit the change as a prior approval supplement. One thing that's important, that if it's a special circumstance, the firm may request from the FDA that the prior approval supplement is reviewed in an expedited manner. And the cases would be in a drug shortage or extraordinary hardship to the firm. Could be a COVID drug, which we would give an expedited review in the last couple of years. And what are some reasons why when it's submitted as a CBE, it's denied to a prior approval? It could be an API process change, the drug product manufacturing site change is not acceptable, the container closure change is not acceptable, you're adding a new sterile filling line that's never been used before, and a change in the stability protocol. Those would be reasons that a CBE supplement could be overruled and changed to a prior approval supplement. Now, to make sure your supplement gets reviewed correctly and leads to an expedited review approval, follow these tips. Use all the regulations and guidances, and don't just rely on the data to say this is a CBE. It's the change itself, not the results of the change. All in the cover letter, all the changes need to be listed clearly so we don't miss anything. Make sure you know what's happening in the USP so you could make the changes appropriately. And make sure you work with your DMF holder closely in collaboration so that any changes in the DMF are reported to you and you're able to report those to the FDA. So here's a question that let's see if you picked up during my talk. Applicant wants to add an additional API supplier to increase the capacity for a drug shortage product. That change should be submitted as what? As a prior approval supplement, as a CBE30 supplement, as a prior approval supplement but requesting expedited review, or as a CB0? Well, the answer would be a new API supplier is usually a PAS, but if it's in a shortage situation, you could request, and you should request, an expedited review. Next, Benga will be talking about recent updates in GDUFA III. Thank you, and I'll hand this over to Benga.
Thanks, Paul, and thanks SBIA for giving me the opportunity to speak today. Continuing with the topic of managing quality post-approval supplement quality-related changes, I'm here today to speak more on GDUFA-related changes. So our learning objectives be here is to understand the go dates for prior approval supplements, the benefits of using a cover letter attachment with your various submission types for supplements, so PASs, your CPE 0s and 30s, the PEPFAR post-tentative approval amendment and other amendments as such, a description of the types of post-approval submissions that are accepted in GDUFA III as a controlled correspondence. In addition, some thoughts to when to submit a post-complete response letter clarification request, and also what to consider when submitting a reclassification of facility-based major complete response letter amendment for a prior approval supplement. And finally, what qualifies for a request for reconsideration for supplements? Even though this is not new to GDUFA III, I thought it'd be a great opportunity to highlight some of these as well for your benefit. So the goal dates, as you see on the screen here, remains the same actually from GDUFA II to GDUFA III. Our standard PASs, depending on if it requires a pre-approval inspection or not, will be 6 months and 10 months accordingly. And for the priority prior approval supplements, depending on if it requires a pre-approval inspection or if you've submitted a pre-facility correspondence for us to review and it qualifies, as noted in the commitment letter, the timelines are, as noted there, it's 4 months, 8 months, and 10 months accordingly. For the amendments that are received for the standard PAS major amendments, depending on if it requires a pre-approval inspection or not, it'll be 6 months if there's no inspection required and 10 months if there is. For the priority ones, again, the similar timelines of the 4, 8, and 10 months as applicable depending on if an inspection is needed and also based on if a PFC was submitted as noted in the commitment letter. The final part to this is nothing has changed with respect to the minor PS amendments. Both standard and priority will receive a 3-month submission date. Of course, the goal is to have at least 90% of these covered within the respective goal dates, and I believe for the most part you all can attest to we've done a great job with adhering to these commitments and goal timelines. So the cover letter attachment, the agency released a guidance document back in December 2021. And the intent of this was to use this attachment for cultural correspondences and the submissions. I will say that it has become very beneficial for those who have included this with their submissions with the PASs and for CBs as well. We note that it helps definitely in preparing the cover letter, but it helps us as the agency when we receive them to identify various aspects of the submission, which includes, of course, the application number, the dosage forms, the various disciplines that will be involved in the review that you believe, based on your assessment of the information, that will be involved in the review of the supplements. Also, I guess, pertinent information like facilities that will be involved as well and what the supplement provides for, among other pertinent things. The next 5 or 6 slides are just showing you basically a snapshot of the various parts of this form. It is in the appendix section of the guidance, so if you're unfamiliar with it, I'll encourage you to please look at it. It's a great tool, and as mentioned, it helps our process during triage. So what I'm showing you here is just one of the next series of slides that covers the different information included in that form. So if you can use it, we'll definitely appreciate it. PS submissions based on agency requests. So we oftentimes, when we review not often, I guess I should restate that, when we do review a CB0, CB30 that's elevated to a prior approval supplement, I guess as we call it internally when we deny to PAS, it often is, when the PS is submitted, is accompanied with a cover letter as well. So as covered in the previous section, of course, that attachment form would be great to include. But more importantly, in the cover letter, please include a reference to the date you had the communication with either the OGD-RPM or the OPQ-RBPM informing you of our decision to deny to PAS, and also who your point of contact was in that communication. This helps when we receive the PAS. This helps during our triage process, since there were designated people who looked at that submission. at the time of receipt, we oftentimes like to give the same review team once the PS comes in. So when we can identify this early on in the cover letter, it helps with the process. So we definitely appreciate that. As mentioned earlier, there is some changes to the control correspondences as it applies to post-approval submissions, specifically in this case, I guess to PASs alike, but applies to any one of them submissions. So we published our draft guidance in December of 2022. As I just indicated here, after issuance of a complete response letter or tentative approval letter, you can now submit a control correspondence if you're looking for clarification on some of the deficiencies or comments. After ANDA approval, general post approval inquiries for your prior to submitting things, you can also use that pathway to submit the inquiries for the response. Please note that number 2 here, that things that are not covered are things that are available in public guidances. We won't address those, but other types of things we would. So I definitely encourage you to have a look at the guidance document. The post-complete response letter clarification teleconferences between FDA and the applicants existed in the prior GDUFA period and is still here as well. GDUFA III, we published a guidance on this back in October of last year, and we talked more about the opportunity. This presents an opportunity to clarify deficiencies, either the major or minor deficiencies that may have been issued in a complete response letter. I noted here that questions requiring further agency review, disputes about classification of your CRL, or new information submitted would not be addressed. The intent of the post-complete response letter clarification teleconferences are really just to help clarify identified deficiencies. In addition to a GDUFA III that we didn't have in prior cycles, which is of great benefit, is the reclassification of facility-based major complete response letter amendments. So this reclassification is specific to when we issue a complete response letter and the only major deficiency is facility-based. So you have the opportunity now, if you have, based on the supporting information, if you believe we should reclassify your submission at the time of receipt from a major to a minor. And we have provided information out there in the public domain to give some form of guidance as to how you should go about making those submissions. At least it's documented in the commitment letter. But the key point here is you must make that request at the time of submission. So when you submit your CRL, you're submitting a reclassification request from major to minor with the necessary supporting information. If it qualifies, of course, we'll consider that and make that determination at the time. The last part here, again, is the request for reconsideration. Something that's not new to GDUFA III, but we found it to be worthwhile just as a reminder, is a pathway to handle FDA regulatory actions that relates to a PAS and has scientific significance. And this, however, other actions could be a refuse to receive decision, tentative approval letters, complete response letters, classification of a major amendment, classification of the standard review status, and also a CB0 denied to PAS. You have the opportunity for us to request, to ask us to reconsider our decision. So that pathway is available to you as well. All right, so wrapping this up, we have the challenge question. And this is, an applicant was issued a complete response letter for a prior approval supplement, which included the following major deficiencies: drug product, microbiology, and facilities. Which of the following pathways is appropriate for the reconsideration reclassification requests from major to minor? For this specific question, though it may seem tricky, but the pathway for this is a request for reconsideration. And again, this is if you're looking for the agency to consider reclassifying it from major to minor. You wouldn't submit this in a control correspondence. You wouldn't submit this in a post-serial teleconference. The reclassification D doesn't qualify because there are other deficiencies that were classified as major. So not just facilities. As you recall, that path is only for if the only major deficiency in your PAS is a facility. In this case, you may submit it as a request for reconsideration from major to minor. And of course, if it qualifies, we will reconsider it. We will reclassify. Well, I guess the reconsideration will be granted rather to change it from major to minor. I thank you for your time. I look forward to hearing your questions during the Q&A session. Thanks once again.
Good afternoon, everyone, and thanks so much for taking the time to join us today. My name is Michael Chasey, and I'm the Director of the Division of Pharmaceutical Quality Programs within ORA's Office of Pharmaceutical Quality Operations. My colleague, Lane Christensen, and I will be spending a few minutes discussing with you the best practices for remote interactive evaluations, or RIE, and other alternative inspection approaches. Previous SBIA talks have each covered the topics of 704A4 records requests and RIEs in detail. Our intent today is to update you on the changes that have been made, specifically looking at RRAs, or remote regulatory assessments, and the recent draft guidance that was issued. In addition, we wanted to provide you updates on the use of these alternative tools and other efforts that have and continue to be ongoing in our efforts to evaluate manufacturing facilities. I will discuss FDA's remote regulatory assessment guidance. Lane will then talk about the implementation of alternative tools, such as 704A4 requests and RIEs. He will also discuss other tools, such as mutual recognition agreements, and finally, some lessons learned and a direction forward. On July 25th, 2022, FDA published a draft guidance for industry titled Conducting Remote Regulatory Assessments, Questions and Answers. This document was intended to provide FDA's current thinking regarding the requesting, conducting, and use of remote regulatory assessments by FDA. The draft guidance document was for comment purpose only, with the comment period closing on September 23rd, 2022. FDA is currently reviewing stakeholder comments and will address them in the updated version of the guidance for industry. In addition, recent changes to the law have added mandatory remote record request authority for devices and bioresearch monitoring to Section 704A4 of the Food, Drug, and Cosmetic Act. So what exactly is an RRA? An RRA is an examination of an FDA-regulated establishment or importer and/or its records conducted entirely remotely to evaluate compliance with applicable FDA requirements. RRAs assist in protecting human and animal health, informing regulatory decisions, and verifying certain information submitted to the agency. RRAs are a tool FDA may use to support regulatory decisions and oversight activities. Currently, requests for records or other information from drug establishments under Section 704A4 of the FD&C Act is an RRA for which participation is mandatory, meaning they are not voluntary. However, RRAs that are not conducted under these authorities, such as a remote interactive evaluation, are voluntary in that an establishment can decline to participate or withdraw participation during the RRA, in which case the agency would consider other tools for evaluating compliance with FDA requirements. So why did FDA draft this guidance? Based on our experience during the COVID-19 public health emergency, FDA has noted the value of RRAs and concluded that they should be used for certain scenarios outside the current pandemic and for all types of FDA-regulated products. The draft guidance is intended to increase industry's understanding of RRAs and facilitate FDA's process for conducting RRAs. It is also intended to provide answers to frequently. So when may FDA initiate or request to conduct an RRA? FDA may initiate, or in the case of a voluntary RRA, request to conduct an RRA whenever we determine an RRA is appropriate to help fulfill the agency's regulatory responsibilities and protect human and animal health. For example, when FDA cannot conduct an inspection due to travel limitations brought on by pandemics, such as natural disasters or other unstable situations, making travel infeasible. In addition, when FDA determines that an RRA will assist FDA in conducting elements of establishment oversight or support regulatory decision. Examples include preparing for an already planned inspection, following up on a consumer complaint, assisting in verifying that an establishment has completed certain corrective activities, or supporting the review of a marketing submission. FDA will use a risk-based approach to determine whether to initiate or request an RRA. It is important to note that programs and centers within FDA may assess risk differently based on the product. The ultimate decision to initiate an RRA rests with FDA, as FDA retains discretion to employ RRAs as appropriate. At this time, we do not accept requests for FDA to perform an RRA. Is an RRA considered an inspection? No. Generally, an inspection, such as described in Section 704A1 of the FD&C Act, involves duly designated officers or employees of the FDA physically entering at a reasonable time and a reasonable manner an establishment subject to regulations under the FD&C Act to determine compliance with applicable FDA requirements. For this reason, we do not consider an RRA to satisfy statutory requirements that specify inspections under Section 704 of the FD&C Act. FDA will not display credentials during an RRA. FDA will not issue a Form 482 notice of inspection during a domestic RRA, nor will FDA issue a Form 483 inspectional observations during the RRA process. FDA's experiences so far have identified significant benefits using RRAs. For instance, RRAs have been used to help support and reduce delays of approval or authorization of marketing submissions for FDA-regulated products during the COVID-19 pandemic. They have also helped inform FDA decisions regarding whether to authorize multiple emergency use authorization requests for COVID therapies. RRAs have assisted FDA in verifying corrective actions taken in response to inspections of previously compliant manufacturers and in gaining compliance insight when it is not practical to inspect. RRAs have also provided information about deficient practices, which led FDA to take regulatory actions, conduct inspections, and has helped inform future inspection planning. Before I turn it over to Lane for his portion of the presentation, let's see if anyone was actually listening to me. Question to you is, what are some examples of a remote regulatory assessment? Note that more than one answer can apply. Is it A, a request for records and other information under Section 704A4 of the FD&C Act? Is it B, an on-site inspection? Is it C, a remote interactive evaluation? Or is it D, all of the above? The correct answers are A, mandatory request for records and other information under Section 704A4 of the Food, Drug, and Cosmetic Act, and C, a voluntary remote interactive evaluation. It is important to note that an RRA is a fully remote activity with no physical FDA investigator presence on site at the facility. This concludes my presentation. Thanks so much for spending time with me, and I'll be back at the panel session to answer any questions that you may have. Lane, go ahead and take it away.
Thanks, Mike, for setting this up and turning it over. Greetings, everyone. My intent today is to share with you how our RRAs are being applied to the work we do here at the FDA. So this is the workflow that was implemented for facility evaluation since the COVID-19 public health emergency began. When an original application or supplement comes in, it lists facilities with various manufacturing-related responsibilities. Based on the following risk factors, we make a determination if the listed site or sites are acceptable or whether a pre-approval inspection is needed. If not, then the facility is found adequate. Now, if yes, we will evaluate if we can use alternate tools. If when using any of these alternate tools, we find that a facility risk is low with respect to its ability to successfully complete their defined roles, then the facility will be recommended adequate for approval. And if we determine an on-site inspection is needed, then we work with our inspection colleagues to coordinate within the timelines established under various user fee programs. Now, I'm not going to give much of a background on our 704A for records request program, and we'll refer you to other information, including a presentation given previously by FDA colleagues, one noted here at the bottom of the slide. Although initially approved in 2012 under the FDASIA amendments for the Food, Drug, and Cosmetic Act, it wasn't until the pandemic that we really saw wide use of this provision. In just a matter of a few years, the agency has completed a large number of such requests, with over 1,000 as of the date when this data was pulled. We have seen a number of requests for both our surveillance and our pre-approval programs. For surveillance, over 35 import alerts have resulted in our efforts, divided between simple refusals for the Import Alert 9932 program and for issues with good manufacturing practice GMP operations under the 6640 Import Alert. For pre-approval assessments, we've had over 330 sites that were evaluated, which represents even more than that number of total applications. We've been able to approve over 330 applications based on the outcome of our records requests. During this time, over 75 applications were put on hold due to issues identified during the review of the record requests. We're looking into this data. It shows that these approval and withhold actions represent all across the different drug programs and include different dosage forms and locations across the globe. And as I put this presentation together, I was able to take a further look at pre-approval data and those specific applications that were withheld in the 2021 timeframe, after we completed records requests. I was interested in data from 2021 since it's been over a year since the evaluations were completed, and it allowed me to also see what happened subsequently to the review requests. What I found was these withheld sites were not location-specific, being across Europe and throughout Asia. Responsibilities of these sites that were initially withheld also varied. Simply due to the difference in volume of applications between programs, it made sense that the original ANDA sites were associated with more application and made up roughly 60% of those that were withheld. You're taking a further look at what happened next to these sites. Close to half of them were initially withheld as an outcome of a records request. They were able to resolve those issues through subsequent responses to the observations, and some of them were able to do so through on-site inspections. The rest of the sites, the other half, appear to still be pending over a year later. So in these cases, the agency issued a complete response letter. In these cases, due to the issues that included at a minimum the outcome of the record requests. Looking at the complete response letters, however, it suggested that there were more issues indicated than just these facilities. One particular site I noticed responded to the records request that they didn't conduct the listed testing responsibilities and weren't even doing business with the application holder. And so I'm sure that we can all agree in such a case that it was better to resolve this through a records request than it was to have somebody out doing an inspection of that site. Now for RIEs, I wish not to provide too much background information on this, as it has been covered in previous generic drug forums. If you're unfamiliar or wish for more background information on this topic, I refer you to our guidance and in presentations from last year highlighted here at the bottom of the slide. Now checking out some specific examples. Both of these here are for drug product manufacturers, both at foreign locations. Facility 1 was a drug product manufacturer for 2 pending ANDAs, both for delayed release tablets representing a new product profile for the facility. One of them included a unit operation that was new to the site, being hot melt extrusion. Now the second facility was a manufacturer for an aerosol foam combination product, which was a new type of product for the facility, new as being per the application history with the agency. They did, however, have experience manufacturing this type of product for other markets, as we discovered during the course of the RIE. At the conclusion of a records request for both sites, it was determined to conduct an RIE to go through the proposed operations. The opportunity allows to spend time discussing with subject matter experts and visualizing operations and following up with questions on testing and laboratory capabilities. In the end, both sites were found adequate and each application was recommended for approval. We now look at a couple of examples for foreign sterile manufacturers. Number 3 was for an NDA proposing to fill a solution into prefilled syringes. During the previous inspection, the site had been noted as having a number of significant issues that led to an OAI, an official action indicated outcome, where the site ended up even receiving a warning letter. During a subsequent meeting, when discussing the facility status of the approvability of the NDA, the site inquired about the possibility of conducting an RIE. In addition to not accepting requests for an RIE due to the significant risks identified during the previous on-site inspection and the outstanding concerns with the site, it was determined an on-site inspection was necessary and a satisfactory outcome required before the site could be approved for the NDA. For the next example, number 4, a high priority application was submitted under an emergency use authorization mechanism. Regarding the contract manufacturers, recognized the agency had previous history with the site and had approved a similar product for use in the United States. We decided to first pursue a 704 records request, leading to this RIE. During the course of the RIE itself, the assessment team identified multiple issues, one of which related to concerns with ensuring an aseptic environment during the compounding and in product fill steps. As such, the team was unable to recommend approval. The final decision was made that any fixes or corrections made in response to the observations issued needed to be verified and further evaluated during another on-site inspection. In each of these examples, I have made reference to outcomes and would like to go over the different outcomes for our RA, specifically for 704 record requests and RIEs. And when conducting either a record request or an RIE, if there are no observations made, it is very likely the site will be found adequate. In the case of a record request, you should expect to receive a Form 4003 stating the record request is complete and there are no further requests. For an RIE, a no observation memo will be sent during the closeout of the meeting. Now, if observations were made, the Form 4003 will include an attachment with an observations letter to communicate the outstanding issues along with instructions for responding. In the case of an RIE, the observations will be discussed at the closeout meeting, and an observation letter will be provided along with instructions for responding. In both cases, the agency encourages firms to submit responses within 15 days. After this round of communication, if issues remain unresolved, a facility may be found inadequate and the application would likely receive a complete response letter. In this case, a post-action letter will then be sent to the facility after the action date. The responses to the post-application letter will be evaluated at the next review cycle. During the next review, the facility is again evaluated based on similar factors as before, where responses to the RRA, including the PAL responses, will be included in the evaluation. So depending on these efforts, the facility may end up requiring an on-site inspection or use of alternate tools once again. So I'd like to go over some suggestions for enabling a successful RRA. And as you can tell, many of these interactions are very similar to an inspection, on-site inspection. As such, it's very important that you commit the same level of importance and attention as you would to an on-site inspection. Even though we keep saying that RIE is not an inspection, its outcome could affect the regulatory decision and the application and the timing of that decision. Anything that you don't understand or have doubts about, anything that seems unreasonable, please ask for clarification before committing to executing that request. Document organization is extremely important. It goes with both types of RRAs. If we really want to be efficient with your time in preparation as well as our time for reviews. Have subject matter experts on hand during the RIE sessions. You're ready to answer questions as you would during an on-site inspection. And finally, technology and platforms used for the RIEs can make or break our planned out interactions. Please check, please double check, please triple check for readiness. Are you able to have multiple breakout rooms? Can you zoom up on the line during production? Are there any dead spots in your facility that will drop a signal? This brings us to our second and final challenge question. The following statements about an RRA are true except A, virtual tour during an RIE doesn't need to be conducted all at once. B, records requests are made in advance or in lieu of an inspection within a reasonable time frame in either electronic or physical form. C, since they are not considered an inspection, it is not necessary to respond to any observations made during an RRA. Or D, generally, your records requested under FD&C 704A4 before an RIE. If you chose C, then you are correct. You are encouraged to respond within 15 business days to any observations that are made during an RRA. Your timeliness in responding can play a big part in the approvability of any referencing application or applications. So with that, I wish to summarize with a few closing remarks. The first is really a question. What will inspections and our interactions with manufacturers look like in the future as we get more comfortable with these tools? In order to get there, we are leveraging now the lessons learned from across applications and inspection programs to inform how to improve. For RIAs, they may be conducted in advance or in lieu of an inspection. An FDA may conduct an RRA following an inspection in order to conduct follow-up activities with the establishment or to assist in verifying corrective actions if appropriate. So really, the story is far from being over. There's a lot going on. Now is a great time as FDA continues to gather experience, seek feedback, work with stakeholders, hear what industry has to say, learn from best practices, learn from comments and feedback on our published guidances, and work together with regulatory counterparts and simply being able to use these tools more. We're currently reviewing feedback on the draft RRA guidance and hope to get an updated version out shortly. And then finally, expanding mandatory remote records request authority to be inclusive of both devices and by our research monitoring program. With that, thank you for your time. We look forward to any questions you may have during the panel discussion.
Thanks so much for another group of great presentations. As a reminder to our attendees, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many as time allows. It's our pleasure to welcome to our Q&A panel Dr. Derek Smith. He joined FDA in 2010 and served as a compliance officer, CMC assessor, quality assessment lead, branch chief, and division director prior to his current role. Derek is the Associate Director of Regulatory Affairs for the Office of Pharmaceutical Manufacturing Assessment within OPQ. He provides leadership and oversight for the assessment of the manufacturing process and facilities for biologics and small molecule drug applications with a focus on the integration of application assessment and inspection findings and data reliability assessments. He also serves as the co-chair for the New Inspection Protocol Project Initiative for the pre-approval inspections and is a member of the Knowledge Aided Assessment and Structured Application Initiative Steering Committee. Looks like we have some questions coming in, and we do have a couple questions already queued up for our first panelist, Kiduo Chan. And here is the first question. Please explain when a change in drug substance manufacturer would be a facility change. Can a facility change be submitted as a CBE 30?
Thanks. So a facility change involves changes in the location of the site of manufacture of intermediate, unfinished, or finished final drug substance. There are circumstances that facility change can be reported as a CBE 30. For example, a change to an alternate manufacturing facility within the same master file or within the same corporate ownership, or a change in the manufacturing for a drug substance intermediate. The change shouldn't involve any route of synthesis manufacturing changes. Usually, the quality of the intermediate or the drug substance and the impurity profile should be comparable between the existing and the proposed sites. Of course, this is assessed by case, and please refer to the draft guidance for more examples. Thank you.
Thank you for responding to that question. We do have another question for Kiduo Chan, and here is the question. Should a nitrosamine impurity assessment be part of any API assessment for a change in the API manufacturer for both pre- and post-approval submissions and be provided in the submission, or should be kept at the site to align with the nitrosamine guidance, which notes the assessment does not need to be provided in the marketing application?
So a nitrosamine impurity assessment should be submitted, especially if you have a change in the API manufacturer for both pre- and post-approval submissions. And per the FDA guidance for the nitrosamines in human drugs, all manufacturers of the chemical synthesized API should take appropriate actions to mitigate the risk. And you should review the manufacturing process and perform risk assessments to identify any potential nitrosamine impurities. And please remember to submit, especially if you have an API manufacturer change. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We've got a couple of questions that came in for Rajeeb Paul. And here is the first question. What if the API source did not change, but the release test facility changed due to closure? And if no specs and no change in specs, can this be a CBE 30?
This is a great question. If the API source did not change, however, testing facility changed due to closure, where no change in the specifications will be a CBE 30, provided that the post-testing facility has satisfactory cGMP status. However, if the new facility does not have satisfactory cGMP status, it should be a PS. Thank you.
Thank you for responding to that question. We have another question for Rajeeb Paul, and here's the question. If there is a change in the residual solvent and final drug substance stage made by the approved API manufacturer, then for introducing a new residual solvent in the ANDA, what should be the filing category and the documents required?
Thank you for the excellent question. If there is a change in the residual solvent in final drug substance stage made by the approved API manufacturer, then for introducing a new residual solvent in ANDA, it should be a prior approval supplement
per the guidance for industry, changes to an approved NDA or ANDA, Revision 1, published in April 2004, Section 7B5. The applicant should provide certificates of analysis for the new API manufactured with the new solvent, both from the API manufacturer and the drug product manufacturer, and manufacture a drug product batch and submit data to show that the new solvent does not affect the drug product quality. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a few questions that came in directed to Bo Jang, and here is the first question. What are the critical factors that may fail or cause the facility to fail an inspection?
That's a good question. This would be depending on what type of facility inspection that is. If it's a GMP inspection, any significant GMP violations that may result in the inspection failing. And if it is a PAI inspection, pre-approval inspection, so a couple of things that could happen. First of all, if that inspection found out the information at the site is not consistent with the data in the submission, that could be a major deficient. And then the second one could be the facility is not ready for commercial operation. And another reason could be any significant data integrity issue identified at the facility that could fail the facility inspection as well. Thank you.
Thank you for responding, and we do have one more question for Bo Jang, and here is the question. One drug substance manufacturing site is on OAI status. Do I need to repeat the bioequivalence study?
That's a good question. The answer could be it depends. If the GMP violations causing the facility OAI status include data integrity violations, and those data integrity violations impact the data reliability in the application, then a new B study is required to replace the data in the application. And also remember that the application will not be approved until that or GMP violation status is changed into compliance. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Paul Swartz, and here is the first question. I submitted a CBE supplement, but it was denied to a PAS. Can I appeal that decision?
If you get a denial from a CBE to a PAS, under the current way of doing things, you could submit a request for reconsideration, in a way, an appeal, and that will be acted on quickly. And if it's if the FDA agrees with your request for reconsideration, the supplement will be reinstated and continue as a CBE. But if not, and you'll find out a reason why, then it would be denied. The denial will hold, and it would be you would have to resubmit as a prior approval supplement. But you should definitely, if you feel that you didn't get it wasn't correct, submit that request for reconsideration.
Thank you for responding to that question. We do have one more question for Paul Swartz. This is the question. I need an additional site to package my drug product. What type of supplement should I submit?
Right. A change in site for packaging is typically a CBE 30, because it will require looking into the history of that particular site. If the site is acceptable, then it would be a CB30. But if it doesn't have a good compliance history, that again would be denied to a prior approval supplement.
Thank you for responding to that question. Moving on to our next panelist. We have a couple questions that came in for Binga Okubadejo. And here is the first question. What are the timelines for an expedited PAS for a drug shortage product?
Thanks for that question. So the timelines as stipulated in the commitment letter, if it were granted for review for drug shortage for a priority review, if a pre-approval inspection is not required, it'd be assigned a 4-month goal date. If a pre-approval inspection is required, it will receive an 8-month timeframe. Thank you.
Thank you for responding to that question. Moving on to our next panelist. I've got a few questions that came in for Lane Christensen, and here is the first question. How effective are the RIE inspections, and how do they compare to on-site inspections? Were the outcomes comparable and efficient?
So answering the second question first, I would say yes. How effective? Not sure about effectiveness, but how confident are we in these remote interactive evaluation inspections compared to on-site inspections? I would say the confidence is there in the decisions that we make. Otherwise, we wouldn't move forward with an approval determination. Those that I've been involved with, I think we were on a 12 plus or minus a couple hour time lag, and so we were up in the middle of the night, but we were able to focus 2-3 hours at a time, see what we needed to see, interview those that we needed to interview, and so it was a very directed approach each evening in our interactions with the site. We were able to see what all we wanted to see and take the time. And so at the end, when we closed them out, we felt good about what we had seen and any issues or any decisions that we wanted to make and recommendations for approval. So that confidence didn't seem any different than when going on site and spending 5 days, 7 full days. Doing an inspection. So I think for myself and those that I've interacted with, they worked out quite well.
Thank you for responding to that question. Moving on to our next panelist. We've got a question that appears to be from Mike Chasey, and here is the question. What is the estimated time frame between when a company responds to a records request and when a closeout or observation letter should be expected?
Thanks, Ray, for that question. Generally speaking, the estimated time frame will depend on the complexity of the documents that have been submitted to support the application. And for review time, because we do want to provide time to communicate any issues that we have found during our review and allow time for response to any of the issues to support the final application decision. At the end of the day, this will all definitely be accomplished by the user fee date. Thank you.
Thank you for responding to that question. We've got a question just came in for Dr. Derek Smith, and here is the question. Regarding data integrity, you mentioned using third-party forensics analysis. What tools will the agency use or recommend for industry to consider to enhance detection of data integrity?
Thanks, Ray. With respect to the third-party testing that Bo, or third-party audit that Bo discussed in his presentation, that is more about finding an independent analysis of the data integrity issues. So, what we're looking for when data integrity issues are found, we're looking for the facility, the applicants, to work with a third party to really assess the data. And so the tools, the assessment approach is really dependent upon what specific data integrity issues are observed, and how to then assess how deep those issues go and how much they impact either the facilities data from a manufacturing perspective or the DMF or application data. And so really it's a dependent situation based on what's found. And the third party audit is more about the independent analysis, right? So that it's someone that's providing a different, an outside perspective on what was going on in the facility to ensure that there is some, objectivity to the analysis. And with respect to data integrity controls, I think there's a lot of different guidances and presentations out there that we've given in various spaces, either from OMQ or OPQ. And I would encourage everybody to go look at the data integrity guidance and some of the other presentations that we've given with respect to what you can do in a facility, to help maintain a state of data control and ensure that operations aren't impacted by breaches in data integrity. Thanks, Ray.
Thank you for responding to that question. We have just under a minute left. We're going to try to squeeze one more question in. If Dr. Smith will, it looks like one just came in for him. And here's the question. As part of changing our API source, if the original source is later determined VAI after data integrity concerns are identified, does that resolve the concerns from the application and a DMF perspective?
Thanks, Ray. And yes, so I think as Bo said in his earlier response, it depends. The VAI CGMP classification may not necessarily reflect that the agency has found any retrospective analysis of data integrity violations cleared. In general, that just means that moving forward, the operations at the facility are considered VAI with respect to current good manufacturing practices. And so in those situations where there is a downgrade, from an initial or reclassification from OAI to VAI, the applicants and the DMF holders should work with their manufacturing facility to understand the impact of what those data integrity violations were, and how to, and as I said just a few moments ago, how to work with a third party to independently assess those, not just with is the data whole? Do we understand and trust the quality? But what does that mean from an overall review perspective in the data that the agency has reviewed? And so, the facility retrospective analysis that might be done by these independent third parties, they may say everything is okay, but it really comes down to you as the applicant or the DMF to also do your own assessment to what that means and how that fits into the data that the agency's reviewed to then further support ongoing review and hopefully eventually approval of high quality generic drugs. Thanks, Ray.
Well, we've all run out of time to answer questions in this session. A huge thank you to our panelists for answering a huge amount of questions that came in. We'll now enter into our break until 2:50 p.m. Eastern Time. Enjoy your break. Welcome back from the break. We're on the home stretch now. Our last session of presentation starts with submitting an ECTD, most common submission issues, and FDA plans for ECTD version 4.0. Our first presenter, Jonathan Resnick, is a member of CDER's Division of Data Management Services and Solutions. He focuses on electronic submissions, and he's been with the FDA since 2011. Prior to joining FDA, Jonathan spent 18 years working in IT to support federal and private sector clients. Our next presentation is on the CDER Next Gen portal. What's new? Our presenter for this topic, Mr. Sayum Saneh, leads FDA's mission-critical informatics initiatives in support of human drug regulatory review processes with a customer focus through analysis and pragmatic solutions that work to advance CDER's public health mission. Our third presentation is on the impact assessment of the 2021 data integrity notifications to sponsors. Our presenter, Dr. Nilofer Tampal, is the Associate Director for Scientific Quality and OB within OGD. In this role, Dr. Tampal develops strategies and oversees implementation of data quality and the scientific integrity of bioequivalence data submitted in ANDAs. She provides leadership and expertise in utilization of advanced analytic tools in the assessment of bioequivalence studies submitted in ANDAs. Next, we'll present on risk factors for benzene contamination. This presentation will be delivered by Dr. Pallavi Nithyanandan, who's the Director of the Compendial Operations and Standard Staff, Office of Policy for Pharmaceutical Quality, CDER. She's been with FDA since 2010, serves as one of CDER's experts on compendial issues, and is the CDER delegate to the United States Pharmacopeal Convention. Our final presentation is on approaches to mitigate the risk of nitrosamine impurities in pharmaceuticals. Our presenter, Dr. David Curie, is the Director of the OPQ Office of Testing and Research and has used his analytical skills to address issues that arise with complex drugs such as heparin, glatiramer acetate, transdermal systems, inhalers, modified release dosage forms, and protein-based therapeutics. Please join me to welcome our first presenter, Jonathan Resnick.
Hello, my name is Jonathan Resnick. I'm with FDA CDER in OBI in the Division of Data Management Services and Solutions. And today I'll be talking about submitting an ECTD, the most common submission issues in FDA plans for ECTD version 4.0. So in today's presentation, we'll be covering a bit about guidance, the most common reasons for technical rejection notices, ECTD validation as it pertains to submissions containing study data, frequently asked questions to our electronic submissions help desk, and FDA plans for implementations of the ECTD 4.0 standard. Let's take a quick look at ECTD guidance. We know that industry must follow FDA guidance when submitting an ANDA. The details are in the FDA 745A guidance called Providing Regulatory Submissions in Electronic Format, Certain Human Pharmaceutical Product Applications and Related Submissions Using ECTD Specifications. And we have a hyperlink right here to take you directly to that guidance. There are two main pieces of it. The first, submissions must be in ECTD format. And the second, submission size 10 gigabytes or less must be submitted through the FDA Electronic Submission Gateway. If these requirements are not followed, the submission will be rejected. I've included here 4 key pieces of information that you can use as resources related to requirements and guidance. So the first is that ECTD guidance currently on revision 7. The second is the ECTD Technical Conformance Guide. The third, the ECTD Submission Standards, and here I've provided a screen capture of a page on our FDA.gov website which contains the submission standards for ECTD version 3.2.2 and Module 1 version 3.3. And finally, there's a link for our ECTD website. So let's take a look at the most common reasons for technical rejection notices. Once the submission comes into the center, there are a few basic validation checks that happen prior to the main ECTD validation run. What we've noticed is by far and away the biggest failure in the validation check is for duplicate ECTD sequence number. We've noticed it accounts for nearly 50% of all ECTD rejections from the center. Passes these basic validation steps, we then use our ECTD validation tool. And what we find here are some of the top ECTD errors that result in an auto rejection. So let me go through these with you. The first one is that all the submitted files were not referenced in the backbone. And that's error code 1306. So what this is all about is if you provide a file in the ECTD sequence folder, but that file is not directly referenced in the index XML or the US regional backbone files, then it's not going to be displayed to the reviewer when they're using the ECTD viewer tool. That's why this is such a big problem and why a submission will be rejected if the file is not referenced properly. The second top reason for rejection is error code 1323. And in many ways, this is the reverse of error 1306. This is where the submitter has referenced a file in the backbone so that the reviewer, when they're using the ECTD viewer tool, they see a document listed on the screen, but when they click on the document, it says file not found, because the actual file itself wasn't included in the ECTD sequence folder. Then the third top error is unsupported DTD version used in a US regional XML. Not too surprising, since last year was when we stopped supporting that old Module 1 DTD 2.01 version. Then we have an error called ts.xpt with information on study start data for each study is not present. That's error code 1734. I'm actually going to get into quite a bit of detail on this particular one. This has to do with the technical rejection criteria for study data that FDA implemented back in September of 2021, and we're going to take a good look at that to help make sure everyone understands what's going on there. And then finally, in terms of auto-rejected errors, all files provided in the study section must be referenced by an STF or study tagging file, and that's error code 1789. I also have a couple of additional ones down here. These are top errors where manual review happens. So this one would be a submission type is invalid for an application type, or a submission subtype is invalid for a submission type and or application type. One thing to keep in mind when looking at these rejection numbers and when we're talking about top errors is that for 2022, FDA rejected less than 2% of all ECTD submissions. And just for kind of frame of reference, just in CEDAR alone, we received about more than 300,000 ECTD submissions. So when you see these numbers, although you might say, oh, 337 submissions, that's a lot, compared to the overall submission intake of close to 300,000, it kind of puts it in perspective. We also have this hyperlink down here to our specifications for ECTD validation criteria. That's where you can find out a lot more detail on these different codes. Now let's take a look at ECTD validations specific to study data. So, a few things. This past March 16th, we went live with a couple of updates to the FDA validation profile, and it relates to 4 validation codes. The first one is 1734. This is the one we had mentioned in that previous slide. And what's different about this code as of March 16th is it's no longer validating if the study ID in the trial summary file matches the study ID in the study tagging file. We moved that validation check to a new code, which is listed here, called 1738. Another change we did was with 1735. It's doing the same validation as always done in the past. The only difference is we've allowed for a condition where if the define XML file is tagged as data listing data definition, that it would no longer trigger 1735. So we allow that to be another valid file tag. Finally, on 1737, this is the same validation that's been in place for years. No change to the logic. The only thing is the validation is now looking across all study data sections in modules 4 and 5, with the exception of these five sections here. Previously, it was only looking at the TRC sections. Bit more on guidance here. There is a separate study data guidance called Providing Regulatory Submissions in Electronic Format, Standardized Study Data, last updated June of 2021. And here it says sponsors must conform to the standards in FDA Data Standards Catalog, and that applies to the NDAs, BLAs, ANDAs, and INDs. And here, in this conference, we're talking about ANDAs. Now, FDA is using ECTD validation 1734, 35, and 36 to confirm that sponsors are conforming to the FDA Data Standards Catalog. And FDA began using these validations and rejecting if they were to fail back in September of 2021. And this subset of ECTD validations are commonly described in detail in the technical rejection criteria for study data, also referred to as the TRC. And just a little note here, the technical rejection criteria for study data, you can find this within the Study Data Technical Conformance Guide. And you can find the Study Data Technical Conformance Guide along with the Data Standards Catalog under the Study Data Standards Resources page. And we've got a link here to make it really easy for you to find it. And then we have a couple of other links here, just helpful for you. If you needed to get some information in general about ECTD, here's the link to our ECTD website. And we also have a page for study data for submissions to CDER and CBER. Just some additional information about study data validations are located on here as well. On this slide, we took a look specifically at ANDAs during the years of 2021 through 2022. And we looked at ANDA submissions where study data was contained in those submissions and provided under the TRC applicable sections. And what we found is through the first half of 2021, there was quite a big failure rate. About 40% of ANDA submissions which contained study data in these TRC applicable sections were failing the technical rejection criteria. But as we got closer to September of 2021, which is when we had communicated that FDA would start rejecting submissions that failed this validation, we found that the conformance rate actually went way up. And so inversely, what you're seeing here is the failure rate of these validations went way down. So this red vertical line is when enforcement began, meaning when we started to reject if the TRC criteria failed. And you'll see that starting in 2022, the failure rates dropped dramatically. They're really just between 2 and maybe 5%, which conversely means that we're seeing a conformance rate, a successful conformance rate to the technical rejection criteria in the 95 to 98% range, which is really good. What we do here on this, we're taking a look now at that 2 to 5% of ANDA submissions last year that did not conform, that failed these TRC validations. And what we found is the TRC validations, number one, it's 3 validations, 1734, 1735, and 1736. And what we found here is it's 1734 overwhelmingly is the one that was giving people problems. It accounted for 58% of the errors, and the most common reason why 1734 was triggering was because the ts.xpt, or otherwise known as the trial summary file, was missing from the study. So we've summarized this information here in this slide. So to begin, let's just take a look again at what is the 1734 validation. It's a data set named ts.xpt with information on study start date must be present for each study in required sections. And the validation is looking, number one, is the trial summary data set present? If it is, study start date is provided. Study start date is in a valid format, and does the study ID in this trial summary data set match up with the study ID in the STF to which it's referenced under? And what we found is 54% of the cases where 1734 triggered is because there just was no ts.xpt file at all submitted for the study. The second biggest reason for 1734 to trigger is that if a TS file was submitted, it didn't have an accurate value here for the study start date, a valid value. And then finally, the third most common reason was that the study ID in the trial summary file didn't match up with the study ID that file was referenced to with a study tagging file. So now we're going to switch gears and we're going to look at a few frequently asked questions to our ESA help desk. We think these conferences are a really good forum to share some of the most common questions our help desk gets. So we've got 3 to go through. So the first one is, where should my document go within the ECTD structure? And what the team will do is respond back with 4 key reference documents. The first being the ECTD Technical Conformance Guide, which is a high-level guide on putting together an ECTD submission and how to organize it. And then the Comprehensive Table of Contents, Headings, and Hierarchy Specification. This contains a list of all the predefined headings and subheadings in the ECTD. And then 2 ICH M4 guidelines, one called the Organization of the CTD, and the other one, the CTD Quality Question and Answers. The next question the help desk gets that we'd like to share is, why did my submission fail ECTD validation 1734? And the typical answer would be, well, a study data was submitted to modules 4 or 5, and there's no valid trial summary reference under the STF for that study. And to see 8221 ECTD technical rejection criteria for study data within the study data technical conformance guide for details. So the study data technical conformance guide is the place to go to really understand 1734 and 1735 and 36, to understand how FDA is validating your submission that's contained study data with the ECTD validation criteria. And the last question we wanted to share with you is, will a validation error cause a rejection? And so here the help desk will respond back with a link to the specifications for ECTD validation criteria, so they can first provide that person with a list of all the validations. And then also point out that in that list, each validation will contain a severity description, and only the ones that are marked high are the ones that would result in a technical rejection notice. So now with the time remaining, I'm going to talk about FDA's plans for implementation of the next version of ECTD, that's version 4.0. So we begin by talking about the ICH level activities, right? Version 4.0 of ECTD was developed through ICH. ICH has published the version 4.0 implementation package. The latest version is 1.5 that was posted in May of last year. On the right-hand side is a screenshot of the ICH webpage for ECTD 4.0. So you can see that all of these documents are currently listed and available for download. There's also a Q&A change request document available as well. And you should also know that the different regions in ICH, so the different regulators, they have been posting information in a table on this webpage that will tell you when they plan to start accepting ECTD4 and when they plan to do technical pilots and so forth. As far as FDA-specific activities, FDA has published the ECTD4 Technical Conformance Guide and the FDA 4.0 Module 1 Implementation Package. The package was originally posted in February of 2020 with an FR notice asking for public comment. Since then, it's been updated a few times, and the latest version was posted in September of last year. FDA has also posted the specifications for the 4.0 validation criteria, comprehensive table of contents, headings, and hierarchy. And I've posted here on the right-hand side just a screenshot of a page that we've set up on our fda.gov website, which is dedicated to the ECTD 4.0 implementation, where you can find all of these documents. FDA is also continuously receiving software updates and testing to make sure that we're working closely with our own tool vendor to make sure that we can receive, validate, and view the ECTD4 submissions. And we're also in the later stages of a technical pilot to make sure that we're set up to meet the ECTD4 specifications. As far as our FDA implementation strategy, our plan is this. Our initial release is going to be to accept new applications in ECTD 4.0. It'll be voluntary. So the technical pilot that we're engaged in right now, that we're wrapping up this month, it's a small group and it's focused on just new applications submitted in 4.0. We're not talking about transitioning yet. We're also planning to provide the ability for industry to submit sample submissions in 4.0 for technical feedback. That would be available to everybody. We're planning to open the door to that later this year. We'll leverage our existing sample submission process that we have open today for 322 submissions. And then we're looking to accept these new applications in 4.0 either late this year or probably more likely early next year. Once we are accepting 4.0 submissions for new application types, we want to make sure everything is stable. So likely about a year after that, we'll look to begin accepting the transition of current applications. So applications that are in 3.2.2 today that you want to switch over to 4.0. And then once we have that implemented and stabilized, we'll then turn our attention to implementing two-way communication. A little bit of information here on the technical pilot. The objective is to determine if the implementation satisfies the requirements in the technical specifications and then make any changes prior to accepting 4.0 submissions in our production environment. And the testing started last June, and it is wrapping up now. So how can you prepare for ECTD 4.0? We've put together 4 bullets here to really help give you a pathway. So first, discuss 4.0 development plans with your vendor or IT organization. Look to make sure you can understand the specifications. Is there a plan for transitioning? You can always send questions to ICH or FDA. Then become familiar with the ECTD 4.0 concepts and enhancements. ICH has put out a couple of PowerPoint slide decks to help you orient and understand the ECTD 4.0 implementation package. Take a look at our FDA 4.0 technical conformance guide too. Then know where to find the ECTD 4.0 information, and I'm hoping this presentation here is going to go a long ways in helping you check that box. And then being able to submit a 4.0 sample submission for technical feedback. Later this year, make sure to check in on our ECTD sample submission process webpage because we plan to update that page maybe in the fall with instructions for how you can submit a sample 4.0 submission for technical feedback. And here I've included the particular link for the ICH 4.0 webpage and the link for the FDA's 4.0 webpage, so it's easy for you to find and bookmark. So in summary, we talked about common submission issues. Duplicate ECTD sequence numbers, they're the #1 issue. We talked about the frequently asked questions to ESUB. ESUB at FDA HHS is a great resource. We talked about the ANDA study data conformance, how it's working out really well, that we're seeing 95 to 98% conformance in 2022 to these TRC validations. And when there is a problem with conformance, it's mostly related to validation 1734, and we looked at what are the top trigger reasons for that. And then finally, we talked about FDA plans for implementation of ECTD 4.0. The publications are out there. The specifications are published, rather. Testing is underway. And our sites are set to start accepting these 4.0 submissions either at the end of this year or more than likely early next year. That's the end of my presentation. Thank you very much for your time. I look forward to taking questions. Thank you.
Thank you for introduction. Hello, my name is Seyum Sanai. I am leading the FDA CDR Informatics initiatives in support of human drug application review and approval process with a customer-focused analysis and pragmatic solution that work to advance CDR's public health mission. Today, I will have the opportunity to speak with our stakeholders about the CDR NextGen portal. As many of you may be active participants in CDR NextGen portal, I will share how the portal has been leveraged to improve collaboration and operational efficiency the last year or so. So, agenda today, we will have overview of CDR NextGen portal. What's CDR NextGen portal? Before and after CDR NextGen portal, our digital transformation, and then what's new really during the last year or so. And we'll share our users' adoption success stories. So, what's CDR NextGen portal? CDR NextGen for the purpose of non-ECTD submission collaboration and reporting. With that, the FDA digital transformation is continue and provide value to receive clean, complete, and validated data to support CDR's mission. As you see, in the past, CDR used to receive paper and media submission through the document room. After CDR NextGen portal established, stakeholders come through the CDR NextGen portal, provide their submission, and that submission directly routed to the review division or internal workflow, and the reviewers can start their work as soon as the submission happened. We have Cedar NextGen, we have about 20 portal products. Today, we'll go over the recent updates regarding CARES volume, control correspondence, and pre-ANDA meeting requests. So what's new? Cedar NextGen portal recently released new product enhancements to support collaboration. The CARES Act being one, control correspondence and pre-ANDA meeting another. And then we also launched a pre-launch activities importation request. We call it Player. It's a new product supporting the Office of Compliance. CARES, we launched in March. The following updates were the CARES Act amount information reporting, processing. The updates include we have a new process to submit CSV files, separated files, which will address the previous limitation on the number of records in CVS file, and also addressed common file validation errors. We also added a new process to replace existing submissions and new functionality to view previously submitted reports. Moving on, for Generic Drug User Fee Act III, we have a couple of enhancements in the control correspondence area. As you see in the screen, we have additional status type for pending for ANDAs. We also added screening questions to product details to align to GDUFA III commitments. We also updated certification text in alignment with Generic Drug User Fee Act III enhancement. In addition, on the right-hand side, we have a pre-ANDA meeting request as part of GDUFA III enhancement. We added to the meeting request addition of meeting types, product-specified guidance in alignment to the GDUFA III guidance. We also added additional questions added to the meeting request to align to the guidance. Next step, moving on to the pre-launch activities importation request or Player, launched in a February time frame. Now with Player portal product, stakeholders be able to submit a Player in preparation for market launch within 60 days of the anticipated decision date for pending initial original marketing applications. And this Player application or portal application is also enabled with two-way communication capability, so stakeholders and the FDA can correspond digitally, such as information requests, clarifications, final response letters. All this can be communicated in a two-way communication capability. So for this, the Division of Global Drug Distribution and Policy Import-Export Compliance Branch manages the Player requests and provide complete response to stakeholders' requests with review decision input from Office of Generic Drug, Office of Pharmaceutical Quality, and Office of New Drugs. Moving on, we have continued to see a growth in our user base. The CEDAR NextGen portal team can continue to make enhancements to improve user experience. As you see, we have 12,000 plus users on a daily basis accessing the CEDAR NextGen portal. So far, we have 100,000 plus submissions came through this pipeline. In summary, we have covered so far what's CEDAR NextGen portal, what's new, and user experience and adoption. So with this, we are continuing to provide you more updates, what's happening in CEDAR NextGen portal. And if you are looking forward to access the CEDAR NextGen portal, please go to cedarnextgenportal.fda.gov and get your access and let us know your feedback. With that, that concludes my presentation for today, and thank you. You have a wonderful day.
Hello, everyone. I'm Nilofer Tempal, and I'm the Associate Director in the Office of Bioequivalence, Office of Generic Drugs. Today, I will be presenting our assessment of the impact on the stakeholders following 2 data integrity notifications from the FDA in September 2021. And what I'd like for you to take away from this presentation is the understanding that both sponsors and personnel at the testing site have a critical role in ensuring the integrity of the data. And both entities, therefore, need to consider appropriate risk mitigation strategies to address falsification of data in bioequivalence studies. Here's the outline of my presentation. I'll start with the background of the events leading up to the notification actions, and then we'll see how these actions impacted the patients and sponsors, and what were some of the lessons learned along the way. I'll also talk about the steps that can be taken to avert future problems of similar nature. So let's go over the background. In November 2019, FDA conducted inspections at the Panaxel and Synchron sites. One of the inspections was a surveillance inspection, and the other was a for-cause because the assessors had raised flags about the integrity of the study data in at least 2 applications. Following the inspections, the Office of Bioequivalence undertook extensive investigative analysis. And this was a very different kind of analysis, not something that we do on a routine basis. And with this analysis, we identified anomalous and unreliable study data from these 2 CRO facilities. We found instances of significant misconduct and violations of the federal regulations spanning multiple studies over a number of years across several drug products and involving multiple applicants. Consequently, general letters had to be sent to the CROs asking them to explain the observed anomalies, including the anomalies in the PK trend, as well as multiple instances of subject pairs with overlapping concentration profiles that we had seen during our investigations. And neither of the CROs were able to provide valid scientific reasons to rule out that the data were falsified. So then in September 2021, FDA posted the notification of actions to inform the stakeholders about the findings at these 2 CROs and also the action that the FDA was taking. The sponsors were informed to repeat the studies from the CRO facilities at alternate sites, and this included approved, tentatively approved, and pending applications for both the generic drugs and the new drugs. Also, since potential bioequivalence problems were identified, the approved products on the market were not considered to be therapeutically equivalent any longer, and meaning they were not interchangeable with the brand product. Therefore, FDA went ahead and changed the therapeutic equivalence rating of the affected generic products from AB to BX. Now, this notice also advised patients to continue with their treatment or at least not make changes to their treatment without consulting their healthcare professionals. And then the CROs, they were notified separately to rectify their system-wide failures with appropriate CAPA, the corrective and preventive action plans, so that the agency is assured of sustained compliance with the regulations from these two CROs going forward. This was not the first time that the FDA had asked applicants to supplement or amend their applications due to integrity issues, and also not the first time that the FDA had changed the TE code of the affected applications due to the integrity issues. However, this was a pivot from the past cases, in that here the driver of the action was extensive investigative analysis by the assessors. It was not solely based on the observations during the inspections. And that's understandable, because when we have intentionally compromised data, they will not always be apparent. They are very challenging to uncover. And site inspections may not provide evidence of intentionally compromised data. For example, when a CRO may be switching the study samples or analyzing the same set of samples and then swapping the data. This type of falsified data can sometimes become apparent when the assessors in the Office of Bioequivalence evaluate the totality of the information and all the evidence before them. Another pivot in this case was that all study data ever submitted from these two CRO facilities were rejected. This was in contrast to only the study data from specific time period in earlier cases involving integrity issues. Now, having covered the background, I want to talk about how the applicants responded to the information request letters and FDA's assessment of their responses. And you will see few common themes becoming apparent in the responses from the applicants. So first, many applicants, they responded with their plan to submit repeat studies. Some applicants, they asked if they could reanalyze the study samples at another CRO because subject samples were still available and stored at the impacted CROs. Unfortunately, this proposal was not acceptable to the FDA. That's because the integrity of the stored samples could not be assured since the CROs had engaged in creating false data in their studies to start with. Then there were another set of applicants. They disagreed with the FDA's position that the studies were inadequate, and they did not think that the studies needed to be repeated. And because they indicated that the sites or the studies were inspected earlier by the FDA, and no concerns were identified and no issues were raised either at the clinical or the analytical sites. And in some cases, these sites were inspected on more than one occasion. And that's correct, that's a fact. However, FDA's assessment was that when data are intentionally manipulated, past inspections cannot provide us sufficient reassurance. And I already alluded to this point in my previous slide. So then during inspections, if there is absence of evidence, it does not necessarily mean evidence of absence when we are dealing with fabricated data. Now then, there were also some other applicants who conducted their own investigations or they engaged third-party auditors. And these investigations and the third-party audits did not find any issues with their specific studies. And they indicated that they had verified the clinical documents and also the analytical and the statistical data. However, here again, FDA had no confidence in the conclusions of these investigations and the audits. And I say this because for the same applications, our analysis had identified data anomalies. The issue here was that rather than doing a systematic assessment, only random checking of the data anomalies were performed during the audits. At least that's what was reported to us. So then none of the subjects identified by us as being falsified or pairs of subjects with overlapping profiles were included in their investigations or their audit reports. And then the next two themes, they are related to the BX rating. So some applicants, they agreed to repeat the studies, but they requested we change the therapeutic code back from BX to AB. And others, they made the same request, but they did not want to supplement their ANDAs with repeat studies. And then there were another set of applicants who cited concerns about the BX rating causing a potential drug shortage for their drug products because they held the major market shares. Now, in all these cases, FDA explained that removing the BX code could only be appropriate if we had made an error in identifying the application as relying on the data from these impacted CROs. Otherwise, the agency needed to treat all approved applications similarly, irrespective of their market share. So now let's look at this slide. It shows a tally for the status of the impacted ANDAs as of February 2023. And you may be interested in knowing how things turned out. So here we can put the impacted applications into 4 buckets. The first bucket includes percentage of the applications, both approved and pending, for which the studies were repeated and submitted for review. The next one shows the applications that were withdrawn. The third bucket includes applications for which the applicants told us, they informed us that the studies had failed to meet the bioequivalence when they were repeated. And then the last one is a miscellaneous bucket. It includes applications for which extensions were granted with adequate justifications and documentation. For example, some applicants, they may have cited challenges with procuring the reference product, or some other applicants, they had to go through the process of requesting the agency to assign a new reference standard because it was not available, no reference standards were available on the market. And this would have caused some delays for them to start the studies. In addition, we also have to remember that this was all happening during the COVID period. Now I want to pivot to the impact of these actions on the key stakeholders. First, the patients, and then the applicants. As far as our knowledge goes, there was no additional burden for the patients. No drug shortages were identified. And all throughout, including the time period of the announcement of the actions, the agency had remained vigilant and monitored the potential for drug shortages for these impacted drugs. And we had also continued to monitor and perform post-marketing safety analysis and evaluate any safety signals or lack of effect signals for these approved drugs. And we did not note any such signals. Now, you can imagine all this requiring massive collaborative efforts and cross-office coordination, and it caused a tremendous increase in our workload, including the work to be done to assess large volumes of new data of the supplements and the amendments. For the applicants, when we did the market share impact analysis, we did indeed find the decrease in the market share for many generic drugs. And also, a substantial number of ANDAs were withdrawn by the applicants, either for business reasons or a non-bioequivalent product. Since we don't have all the information, we cannot tease out what was the reason for the withdrawing. Thinking about the key lessons from this experience, one lesson that is very clear and it stands out is that there is no place for falsified data in FDA-regulated products. FDA will take action against, within its authority once it becomes aware of the falsified data or information in the submissions. And also, depending on the nature of the violations, it may impact all work ever done at the testing facility site, as was in this case. The other lesson we can think about is that the cost at the back end can be costlier than the cost at the front end, because when we have to repeat large number of studies, it can be a huge financial burden. So sponsors need to provide a greater oversight for the outsourced portions of the applications. Also going forward, the agency and applicants need to stay vigilant, at least to the known patterns of data manipulation. And I have provided here a few of them. For example, preferential chromatographic reintegration. I will say we used to see a lot of manual reintegrations in the past, specifically for failing QCs to bring them into passing. And also samples, which were the CMAX samples, would be preferentially be reintegrated so that the ME study would be met. However, we remain vigilant on this aspect, and more recently, we do not see as many manually reintegrated chromatograms. Some other examples would include preferential sample reanalysis, falsification of lab date records, sample substitutions, or slipping in fabricated values for a couple of samples in the statistical analysis, especially the CMAX samples. As for the next steps to mitigate the risk, I will first share with you at least two measures that the Office of Generic Drugs is undertaking to identify unreliable data. So the first one is enhancing data analytics and visualization tools to identify data integrity issues. Here, we are leveraging the functionality of the software tools to automate detection of discrepant and atypical data in the submissions. And we have already adopted some of these tools as an integral part of our study assessment now. The other is working collaboratively and in partnership with our internal and external stakeholders. Internally, this includes cross-office multidisciplinary team approach between our assessors, the OSIS investigators, and our statistical experts. External collaboration includes global health authorities, the counterparts of the global health authorities. And we have established a process, and we hope that sharing the information and the data about the concerns during the assessment among the regulators at an early stage would bring more awareness and also more data points for the agency's assessment, and this could potentially shorten the time of taking the action. What are some of the steps that sponsors and CROs could take to mitigate the risk of compromised data? For the sponsors, there are a number of guidance documents from regulatory agencies and also non-regulatory bodies, all providing very similar recommendations and laying out the expectations of the sponsor's responsibilities. By being diligent about site selection, monitoring the study conduct, and confirming the reliability of the study data for the outsourced portions, sponsors can meet their responsibilities for assuring the integrity of the data in the submissions. For the CROs, to ensure the integrity of the data that they're generating at their facilities, they would need to establish a robust quality management system And more importantly, the leadership would need to support and promote a culture of quality within the organization. And then lastly, sponsors and site personnel should consider reporting fraud and data manipulation in the study conduct. Sponsors may come across this type of information either during their monitoring or study audits. And you can send this information through the mailboxes noted here, or include the information in the submission. More recently, some sponsors have reported to us unreliable data that were identified during their audits. And we greatly appreciate that type of transparency. On this note, I will conclude my presentation. Thank you for listening. Before I go, I have 2 challenge questions. So here's question 1. The market share of generic drugs were not impacted by FDA's actions. True or false? The answer is false. As I mentioned earlier, when we did the impact analysis, the market shares of many of the ANDAs, many of the drug products, the generic products, were impacted. Question 2. Sponsors are expected to verify the accuracy of reports submitted by a CRO in support of marketing applications. True or false? The answer is true. The sponsors are responsible for assuring the accuracy of the data and the information that they include in the submissions to the agency. With that, I thank you again, and I will be joining the panel where I'll be happy to answer any questions for you. Thank you.
Hello, my name is Pallavi Nityanandan and I serve as the Director for the Compendial Operations and Standard Staff, and today I will be presenting on the risk factors for benzene contamination in drug products and will be specifically talking about the excipient carbomer. This talk will begin with providing background on the topic, clarifying interpretation of ICH Q3C guidelines on residual solvents, discussing USP carbomer monographs, as well as FDA recommendations to USP on the revision of carbomer monographs and recent USP revision proposals for carbomer monographs. As many of you know already, benzene is a known human carcinogen that causes leukemia and other blood disorders. Recently, FDA has posted information on our website to inform stakeholders of recent recalls due to benzene contamination. The link to the information on the website is provided at the bottom of the slide. The benzene contamination may be related to inactive ingredients such as carbomers, which are used as thickening agents in drug products, isobutane, which is a spray propellant, or any other drug component made from hydrocarbons. The information on the website also reminds drug manufacturers of the cGMP requirements based on which they must establish scientifically sound and appropriate specifications and test procedures to assure drug components, both active and inactive ingredients, and finished drug products conform to appropriate quality specifications as outlined in 21 CFR 211.84 and 211.160. The manufacturers are also required to test their raw materials and finished drug product batches according to 21 CFR 165 prior to release to ensure that they meet appropriate specifications for identity, strength, quality, and purity. FDA also recommends using risk assessments as a tool to determine if manufacturers have appropriate specifications, test methods, and controls to ensure drugs are free from contamination, and also evaluate the possible presence of benzene in their drug products and components, including active ingredients and inactive ingredients. So USP-NF currently has 8 monographs for various grades of carbomers. The numbered carbomer monographs indicated in the portion of the table with the orange shading which is namely the 934, 934P, 940, 941, and the 1342, are the ones which are manufactured using benzene, and the individual monographs have benzene limits that significantly exceed 2 ppm as compared to the other 3 grades of the carbomers. Now we will discuss the interpretation of ICH-Q3C residual solvents guidelines, regarding which there appears to be some confusion. As per ICH-Q3C guidelines and USP General Chapter 467, Class 1 solvents should not be employed in the manufacture of drug substances, excipients, and drug products because of their unacceptable toxicity and their deleterious environmental effect. Exception for drugs considered as a significant therapeutic in advance, and this is a very important concept, so please make note. Only if a class 1 solvent's use is unavoidable in order to produce a drug product with a significant therapeutic advance, then their level should be restricted as shown in the table 1 of the ICH-Q3C guideline, unless otherwise justified, which happens to be 2 ppm, which is listed for benzene in the guideline. Just restating that again, only if a drug product is deemed to be significant therapeutic advance and the use of a class 1 solvent is unavoidable in its manufacture, then the level should be restricted as listed in the guideline. Please note that sunscreens and hand sanitizers, which are the subject of the recent recalls, these are not considered as significant therapeutic advance, nor is the use of benzene as a solvent unavoidable in their manufacture. Continuing the discussion on ICH-Q3C, even if it meets the criteria we have just discussed, when the use of the class 1 solvent is justified, then these carbomer monographs, which are the numbered carbomer monographs 934, 934P, 940, 941, and the 1342, allow residual benzene levels that are significantly higher than the 2 ppm permitted by Table 1 of the ICH-Q3C and the USP 467 General Chapter on Residual Solvents. So very important point, please make note that the ICH-Q3C guideline and USP 467 are not to be interpreted as recommending that controlling benzene at not more than 2 ppm at the drug product alone is sufficient in the absence of significant therapeutic advance. So that seems to be a point of major confusion, so hope you will take that away from this presentation. So FDA has had conversations with USP and made the initial recommendation to omit these monographs as early as February 2020, and we really recommended the deletion or omission of the numbered carbomer monographs beginning in 2020, and we have had several conversations on that. But an official letter was sent in November 2022, summarizing the previous discussions, reiterating the known health and safety risks associated with benzene, and also we recommended that USP make the monograph omissions effective no later than 2025, which would also provide sufficient time for the implementation of this change for stakeholders. So following our communication and recommendations to USP, they have posted a notice of intent to revise on November 18th, 2022 on their website, informing stakeholders that they intend to omit the numbered carbomer monographs with a targeted official date of August 1st, 2025. So FDA recognizes that industry stakeholders may have questions on the reformulation and how to report the changes to the agency. So we plan to publish a guidance for industry on products with benzene containing carbomers, recommendations for reformulation, which will advise on reporting categories for various dosage forms based on risk considerations for switching to another carbomer with similar physicochemical properties. So Hopefully you will get to see that soon. And FDA encourages early implementation of reformulation if using carbomers 934, 934P, 940, 941 or 1342 prior to the proposed monograph omission date of August 1st, 2025, which means you don't need to wait until the monographs are going to be omitted. You know that the change is coming, so you can implement the change reformulation as soon as your company is able to. That brings us to the end of this presentation. Thank you so much for your attention.
Hello, thank you for that kind introduction. My name is David Curie. I am the Office Director of the Office of Testing and Research, which is in the Office of Pharmaceutical Quality. And I'm going to be speaking about some studies we did in the lab around mitigation of nitrosamine formation in pharmaceutical formulations. Before I begin, I just want to say that patients expect safe and effective medicines with every dose they take. And pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It's what gives patients confidence in their next dose of medicine and what we strive to provide. So in 1978, a group of World Health Organization experts suggested something called NAP, or nitrosation assay procedure, as a general in vitro test under some standard conditions, which is 4:1 nitrate to drug at 37 degrees and pH 3 to 4 under aqueous conditions, and to study the nitrosation ability of drug substances. And this was really kind of targeted toward conversion within humans. And the conditions they settled on are really quite optimized for the formation of nitrosamines, quite favorable for the formation of nitrosamines. And the two drugs in our study, drug A and B, the first one of these formed a nitroso drug substance-related impurity pretty fastly under NAP conditions. And the second one, which had a process-related impurity, which was dimethylamine, under NAP conditions formed NDMA. An important note about using these NAP conditions is that they're favorable, as I stated, for nitrosamine formation. But drugs containing reactive amines may never experience the conditions, these optimal conditions. So it doesn't necessarily mean if you get a positive result with or nitrosamine formation under NAP conditions with a given drug that will actually happen to the drug. So so to form nitrosamines, you need a source of a nitrization. You need a source of nitrite. And nitrite is one of those sources. And then you need a vulnerable secondary amine. And there's a recent publication about the LASA database, which is in the Journal of Pharmaceutical Sciences, published in 2022, where they measured the level of nitrite in parts per million in a number of different excipients that are commonly used to formulate drug products. There's a list. Some of them are listed here on this chart. Crosspovidone, magnesium stearate, povidone, microcrystalline cellulose, lactose, hypermellose, crosscaramellose, and cornstarch. And you can see each of them has varying amounts across all the lots and batches that they tested, with crosspovidone having the highest amounts, close to 10 part per million. And of course, when you would make a given formulation, given solid oral dosage form, you would mix these together in some proportion. And in a typical formulation, an average amount of nitrite present is about 1 part per million. So in most drug product formulations, you're going to have some amount of nitrite present. And to form small amounts, trace amounts of nitrosamines in a drug product, you really, of course, don't need much of the nitrite around. And so this is the excipients are a source of potential nitrosation progenitors. And so in this study, we have the two drugs, and we know that in marketed drug products, these two drugs form nitrosamines, and that formation was dependent on, was in some formulations and not others. And so in the lab, we mimicked drug product manufacturing steps, and we also spiked in excess nitrite. We exposed them to heat and humidity. And we manufactured tablets in-house that either contain or did not contain antioxidants, or contain or controlled or had their pH of the formulation adjusted or not. And our goals from this study are to widely share the findings, and I'm kind of doing that today here, and hopefully to provide some examples of strategies that could be used to mitigate the presence of these in drug products. So this is the OTR manufacturing process. We have one of the two drugs. It goes through a sieving step, a mixing step, and granulation, and this is the step where we could spike in nitrite or dimethylamine if we wanted to. It goes through semi-drying and drying using a fluidized bed drying method. We have a granule formation step, and then ultimately the tableting. So here's some of the results with the first drug, which formed an NDSRI. And these drugs that were made were put on stability for 6 months. And we made measurements across these 6 months. This particular set of experiments was done at 25 degrees, held at 25 degrees C and 60% relative humidity, and then the amounts of the NDSRI that formed over that time was monitored. And there are 3 different antioxidants: ascorbic acid, caffeic acid, and ferulic acid. And then also we applied either acidifying or using sodium bicarb to make the formulation basic in this panel here. In each case, the blue line is the control, where we have added no amount of antioxidant or no change. It's the standard formulation. And you can see that if you do nothing under these conditions, that you get an increase in the amount of NDSRI over a 6-month time period up to about 200 part per million. But if you have ascorbic acid, either at 1%, 0.5%, or 0.1%, you get a big inhibition of the formation of that NDSRI. Caffeic and ferulic also have impact, but not as much as ascorbic acid in this case. And actually, the most dramatic difference was using the sodium bicarb, which basically didn't show any formation. In the other drug, and here we were getting NDMA formation. This is this is color coded through the 3 antioxidants: ascorbic acid, caffeic acid, and ferulic acid. And here, the blue is the initial measurement. The orange is day 30. The light blue is day 60. The darker blue is day 90, and the purple is day 180. And you can see in this case, actually, the ferulic acid had the this one formulation that showed the most formation of NDMA was spiked with 100 part per million of nitrite. So a really big excess of nitrite was added to this formulation. But what you can see that's from these data, if you look carefully, is that when we spike in a lot of nitrite, the ferulic acid in this case seemed to have the most inhibitory activity compared to caffeic and ascorbic acid, which had kind of a similar level of inhibition. The other thing you can see is that even when it is formed, like at day 0, when it was formed in the manufacturing step, that it decreases over time, towards 6 months. So a lot of interesting data in here. Of course, I don't have time to break it all down. Hopefully, we'll be sharing this in more detail soon. But I did want to kind of show these preliminary data, and then some of the preliminary observations that we can make from them. In terms of antioxidants for the first drug substance, which the highest inhibition of NDSRI formation among the antioxidants was observed as ascorbic acid, which was greater than caffeic acid, which is greater than ferulic acid. For the second drug, however, the rank order potency was reversed. Ferulic acid was greater than caffeic, which was about the same as ascorbic. And so the antioxidants need to be fit for purpose, and their effectiveness depends on the drug substance and the manufacturing and the formulation. An increase in antioxidant concentration improved the NDSRI mitigation. So for pH manipulation, a set of conditions, maybe to no one's surprise, facilitated nitrization reactions. Maintaining a neutral pH of the drug product served as a protective strategy against nitrosamine formation. And so an alkaline modifier, sodium bicarb, had the most effective inhibition of NDSRI formation and NDMA formation for the two drug substances tested. And also heat and moisture control would be an important factor. The data suggests that formation of nitrosamines was greater under conditions of elevated heat and moisture, and that the main nitrosamines, or the main progenitors for making them, were introduced or formed during the drying step of wet granulation. This might be due to the presence of secondary amine and oxides of nitrogen in the wet mass, which was then converted to a nitrosamine impurity during drying. So in general, antioxidants and pH mitigate the formation of nitrosamines in these two drug formulations that we studied, and the effectiveness of that mitigation strategy was product dependent. I want to acknowledge a huge number of people that are involved in this, particularly Jan Yang and Dia Shaklia. Most of this work was done in the White Oak labs of OTR, and then the St. Louis lab helped in terms of the testing of all these samples. And of course, my colleagues in various discussions around all things nitrosamines. And thank you for your attention.
Thank you all of our presenters for another group of great presentations. We'll move into our QA panel session at this time. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We've already got some questions coming in, and the first group of questions, the first two questions, will be addressed to Jonathan Resnick. And here is the first question. When will new Type 3 and Type 5 DMFs be considered for pilot in version 4.0?
Great question. So once FDA starts accepting version 4.0 submissions, you will be able to send in a new Type 3 or Type 5. Really, you can send in any application type as long as it's a new application. So it could be an ANDA if it's a new one, or it could be a DMF. It could be a Type 2, 3, 4, or 5. The key is you'll need to wait until FDA announces that we are accepting those voluntary 4.0 submissions, which in all likelihood probably won't be until early next year. We will update the FDA data standards catalog with the ECTD 4.0 standard, and in that catalog it will say the date that we start supporting 4.0. So you'll be able to look there as well as our ECTD website. And I noticed you have the word pilot in your question. So you might be having in mind maybe sending in a test DMF submission to make sure everything's working okay. We do plan to open the door to submit test submissions using 4.0 later this year, and we'll have information about that on our ECTD site.
Thanks for responding to that question. We do have another similar question about testing out ECD version 4.0 capability before sending in a real submission. Are we able to submit a sample to FDA for feedback?
Absolutely. Our plan is definitely to allow industry to submit sample 4.0 submissions so that everyone can make sure that these submissions are being published properly into the standard before an actual real live submission comes in. So we have a current sample submission process set up today for ECTD 3.2.2, our current version. You can learn about that process on our ECTD webpage. For those of you that don't know how to get to our ECTD webpage, it's quite easy. It's fda.gov/ECTD. Now, what we're going to do, and I just kind of alluded to that in the last question, is later this year, we're going to update our webpage that pertains to how to submit a sample ECTD submission. So it includes specific information about how to do the 4.0 sample. I don't have a specific date yet, but look for it to happen around the September timeframe of this year.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Sayum Sane, and here is the first question. Does the CEDAR NextGen portal replace the need to use the old ESG gateway and the need to have security keys?
Great question. So does the CEDAR NextGen portal replace the need to use ESG? The answer will be no. The CEDAR NextGen portal does not replace the electronic submission gateway. The portal is used to submit non-ECTD submissions, which are not required to have an ECTD format. Except in some cases, if, for example, for research IND, yes, we can accept the submission via portal. Hope that helps. Thank you.
Thank you for responding to that question. We have another CEDAR NextGen portal question that came in for Sayum Sane, and here's the question. Can CEDAR NextGen portal be used for requesting IND application number as well?
Another great question. I'm going to repeat the question one more time. Can the CEDAR NextGen portal be used for requesting IND application number as well? The answer is yes. An IND sponsor or a U.S. agent can register and submit a request through the CEDAR NextGen portal to obtain an IND application number. If you have any question, please let us know. But the answer is, in short, it's possible to request IND application number via portal. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a couple questions that came in for Dr. Nilüfer Tamper. And here is the question. If the CRO is identified swapping the data to fabricate the BE results, will the CRO be barred from other ongoing studies with NDAs and ANDA applications?
Yeah, hi, thank you for the question. I think to the best of my knowledge, we don't have regulations that speak to the debarment of a CRO. Maybe you may be thinking about our regulations for debarring a clinical investigator, but the same similar regulations for the CROs we don't have. Thank you.
Thank you for responding to that question. We have another question for Dr. Nullifer Tample, and here's the question. Does FDA perform the reanalysis to conclude the CRO's fabrication data, or is it based on statistical analysis of raw data, or just a review of the data?
Yeah, here I think what we do conduct the statistical analysis of the submitted data, and that's a routine part of our assessment. And in that process, say we observe some concerns. For example, sometimes the statistical output of our analysis may not match the applicant's analysis. Or the assessors may notice some discrepancies in the values in the data sets in the submissions. Then additional analysis or the investigation would be undertaken. And the steps which an assessor may consider will all be driven by the nature of the concern or the issue before them. Thank you.
Thank you for responding to that question. Moving on to the next panelist. Our next group of questions is directed to Pallavi Nithyanandan, and here is the first question. Is it sufficient to restrict the level of benzene at 2 parts per million in the drug product and continue using ingredients and excipients that contain benzene?
Hello, thank you for that question. So the answer would be no, and as explained in the slide 6 of the presentation, only under narrow circumstances, if a product meets the 2 criteria, one of which is that the use of a class 1 solvent is unavoidable in order to produce the drug product with a significant therapeutic advance, then that's only when those 2 conditions are met, then you may be allowed to use an ingredient containing benzene. Otherwise, I think they should be avoided. That's what the ICH-Q3C guidelines specify. Thank you.
Thank you for responding to that question. We do have another question for Dr. Nithyanandan, and here's the question. Does industry need to wait until USP's proposed monograph omission date of 2025 to switch to carbomer that's not manufactured using benzene?
So the answer is you don't have to wait because this is a change that is for the improvement of public health, so you don't need to wait, and early adoption is very much encouraged as soon as you're possible to switch from, it is possible for your firm to switch from the benzene containing carbomer grade to the non-benzene containing carbomer grade. You should go ahead and implement the change. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We just got a few questions in for David Curie, and here is the first question. What is the chemical mechanism for the antioxidants to inhibit the nitrosamine formation?
Well, it's a very good question. I think if you were talking about these reactions in the in solution, these things like ascorbic acid are known to be free radical scavengers. So that's potentially a route, and also in terms of neutralizing nitrite that's present, that's a known reaction where you pretty fastly form, dehydroascorbic acid. And so, but, of course, you realize that what's going on is in the solid state in these tablets. And so it's, what the real mechanism is still a question. And how the antioxidants are doing the inhibition is also kind of an open question, but it's likely either by impacting the nitrite that's present, giving it another route to which to react, or by some sort of free radical scavenging mechanism. Thank you for the question.
Thank you for responding to that question. We've got another question just came in from Dr. Curie, and here's the question. We see that you use the likes of caffeic acid and ferulic acid in the studies. These additives are not in the IID. Is there a possibility that sponsors can start thinking of using these based on the safety data in the public domain?
Thank you for that question. So we chose the antioxidants for the study because they were kind of in series, ascorbic, ferulic, caffeic, and ferulic, and so and they had been used in other published work, and so but we did realize that two of them are not IID, and you'd probably be better off sticking with those, at least in initial work, until there's some sort of agreement otherwise. Thank you for the question.
Thank you for responding to that question. We'd also like to welcome to our Q&A panel Dr. Liken Liang, who's the branch chief in the Division of Liquid-Based Products 2, the Office of Life Cycle Drug Products, and we just had a few questions come in for Dr. Liang, and here is the first question. If the residual benzene level in a carbomer excipient conforms to the 2 ppm limit specified in the USP monographs for carbomer polymers, can I use it for any products without any additional considerations in terms of residual benzene?
Thank you for the question. My colleague Pallavi has already provided answer to a similar question earlier. I'd like to further comment that if the use of such a carbomer is justified from drug product quality perspective, USP tests and limits are considered minimum requirements. Additional consideration may need to be taken depending on the drug product involved. For example, if the drug product has a very high dose, you will have to take the dose level into consideration in assessing the impact of the benzene level in excipients and in other components of the product. And you may need to implement proper controls that may be stricter than what the USP monographs describes, and provide justification for your control strategy. Thank you.
Thank you for responding to that question. We do have another question for Dr. Liang, and here is the question. Where can we get more guidance regarding the expectations when changing a carbomer manufactured using benzene as a solvent to a carbomer manufactured without the use of benzene?
Thank you for that question. As Pallavi mentioned earlier, there will be a guidance published to provide recommendations for various scenarios for carbomer change. In addition, a firm can engage the agency for discussion and advice. For example, a generic firm can submit a control correspondence, contact the project manager, or submit a formal meeting request for a complex drug product, depending on the stage of your application or the product development. Thank you.
Thank you for responding to that question. Moving back up to the top of our panel, we've got a couple more questions that came in for Jonathan Resnick. And here's the next question for Jonathan. I am new to ECD and would like to know if FDA publishes a listing of all headings available in the ECTD?
So the answer to that is yes. On our fda.gov/ECTD webpage, we have a ECTD submission standards catalog. And one of the documents there is called the Comprehensive Table of Contents, Headings, and Hierarchy. And that provides a listing of all of the predefined standardized headings in modules 1 through 5. So all those harmonized headings, especially between modules 2 and 5 through ICH, are all listed there in that specification document for you to be able to access. So hopefully that helps answer that question.
Thank you for responding to that question. One more question just came in for Jonathan Resnick, and here is the question. You mentioned that FDA plans to accept voluntary ECTD version 4.0 submissions within the next year, but when will version 4.0 be required?
So short answer to that question is it's too early to say. So we first need to open the doors and start receiving those voluntary 4.0 submissions for new applications and make sure that the systems are running smoothly and that there are no major issues. After we find that things are working well, we will then open the door for people to transition older applications. So if you have maybe a drug master file that you originally opened up using ECTD 322, and now you want to start making submissions in 4.0 to it, that's something we're looking to allow industry to do probably about a year after we initially open the doors to providing a new application in 4.0. And once we're at that point, then FDA will determine the date that the 4.0 standard will be required, and then we will publish an FR notice to alert everyone of when that will be, and we'll put that date in the FDA data standards catalog and also make it prominently displayed on our FDA ECTD webpage.
Thank you for responding to that question. We've got just over a minute and a half left. I have another question that came in for Dr. Nullifer Tample, and here's the question. Will FDA honor a sponsor's monitoring report as a justification that there were no data integrity issues for our study?
Yeah, that's a challenging question, and I thank you for even bringing it up. So, I'm going to assume here that this question is related to a study that is outsourced to a CRO, and the sponsor is providing the oversight through the monitoring. So now whether a monitoring report will be accepted as a justification, that would boil down to two things. First is the nature of the integrity issue. What I mean is, like, the manner in which the data may be manipulated, whether that's very systematically spread it out, how egregious it was. So that's the first thing that needs to go into the consideration. And then the second is our level of confidence in the monitoring report. So we may need to be able to see and assess the thoroughness of the monitoring, how encompassing it was, whether it covered all the relevant aspects. So, but then in your judgment, if the monitoring report can serve as a supporting document, then you should submit it. And it may add to our overall assessment as we go through all the available information and evidence. It can be considered as a supporting piece of information, but because of the reasons I outlined, it is unlikely that it will serve as a standalone or a sole piece of evidence. Thank you.
Thank you for responding to that question. We are just about out of time, but we did have one question that just came in for Dr. David Curie about nitrosamines. And here's the question. What formulation strategies can be used to mitigate nitrosamine formation in drugs?
Well, I think the pH adjustments were the most impactful for our studies with these two drugs, and of course the addition of antioxidants. And you know, one of the things I want to point out is we chose an acidic form of ascorbic acid and these other antioxidants to use. You know, if you use the sodium salt of it, you might do better, right? You know, so we learned something in these studies about, you know, using the sodium salts might be a better approach because it combines both antioxidant capacity and pH control. So that's kind of seems to be the two most important things. Thank you for the question.
Well, that's all the time we have for our questions and answers in this session. We certainly a huge thank you to all of our panelists and presenters over the past 2 days for the great presentation and answering a huge number of questions that came in. Just a few bit of housekeeping items. That recordings will be posted in approximately 5 to 7 business days. So look for those recordings on fda.gov/CEDAR-SBIA. Remember again that certificate of attendance to support continuing education credits for SOCA RAPS and SQA, and the CE claim codes for continuing education for healthcare providers, physicians, pharmacists, and nurses are available for 2 weeks. And remember, that deadline is April the 27th. Now it's our pleasure to welcome back Dr. Lei Zhang to share some closing thoughts. Welcome, Dr. Zhang.
Thank you, Ray. Good afternoon, good evening, and good morning to all the participants across the world. It is at the end of this year's Generic Drug Forum. I hope that you, like me, enjoyed every session of the past 2 days of the forum. In closing, I would like to thank all the participants for joining us from across the globe and making this 10th anniversary of the Generic Drug Forum a success. The past 2 days have been filled with valuable insights and practical advice on the ANDA assessment process, as well as updates on timely topics such as GDUFA III enhancements, product-specific guidances, industry meetings throughout the product development and life cycle, best practices and tips for ANDA submissions, policy, and information technology, just to name a few. It is encouraging to see the continued interest from our stakeholders in this forum, and we are committed to providing clarity and fostering collaboration with the generic drug industry. We had more than 3,600 people registered from more than 100 countries, and nearly 2,000 people participated via Adobe in the past 2 days. It took a village to organize and run this forum. I appreciate the efforts of the speakers, panelists, and the subject matter experts who worked tirelessly to deliver informative and engaging sessions. I also would like to extend my gratitude to our SBIA colleagues, Brenda, Ray, and others who moderated the sessions and coordinated behind-the-scene efforts. Finally, I would like to invite you all to join us for the upcoming SBIA webinars on the M13A draft guideline on May 2nd and on GDUFA III scientific meetings on May 15th to learn more about these topics. In addition, FDA also going to host 2 hybrid workshops with the Center for Research and Complex Generics, CRCG, on next week, April 20th to 21st, and also on May 10th, respectively, on alternatives to comparative clinical endpoints and pharmacodynamic bioequivalence studies for generic orally inhaled drug products and drug device combination training. I believe those topics will be of interest to this audience. We are also hosting our annual GDUFA Science Research Workshop, public workshop, on May 11 to 12 at the White Oak campus. It will be in a hybrid mode, have both in-person as well as online option for people to attend. We hope you can join us for these informative events. Follow us on the generic science research website or FDA meetings, conferences, and workshops website for future events that may be of interest to you. With that, I thank you all for your time and participation, and I look forward to seeing you again in the future. Thank you.